unaccepted theory is the mantle plume hypothesis. Mantle plume that generally comes from the core mantle boundary and it comes to the asthenospheric mantle and then interacts with the lithospheric mantle. As I was discussing about the different components of large igneous province, you can see how the different components of large igneous province are forming in a mantle plume setting. These are radiating spectrum, these are transversal terminals, these are surface hierarchical tiles, these are pragmatic outer plates, these are sigma crystals and automatic cells. These are the different components of large igneous province that basically form due to the interaction of the mantle plume with the lithospheric mantle or asthenospheric. So, why the large igneous province research is important? Because we are dealing with the several important hypotheses that we are going to resolve by the study of large igneous. One of the important hypothesis that is the evolution of Earth mantle. How the Earth mantle, whether it is has changed its time. As I was saying, the magma from lithosphere and asthenospheric mantle. If we study different large igneous province, we collect large igneous province from Archean time, we collect large igneous province from Proterozoic time, and from recent time. We study its geochemical evolution, and we can find out how the Earth mantle has changed its time. Second thing is metallurgy. As I was saying, large igneous province has generated huge amount of mafic and ultramafic. Obviously, if we are dealing with mafic and ultramafic magma, they must associate some amount of metallurgy, whether it is nickel, whether it is cobalt, whether it is different Pg, whether it is copper. So, defining the association of different metallogeny with large igneous province is important. In the global platform, some metallogeny is associated with LIP and some LIPs are completely parallel. So it is necessary to define the geochemical constant on the association of metallogenic with LIP. Third point is the environmental changes that it is going to affect when it is erupting large amount of magma. As I was saying in the Deccan LIP, it is generated a magma at 65 million years. And we all know the 65 million year magmatic activity is supposed to be the extinction of dinosaurs. So when it is erupting large amount of magma, along with the magma, several gaseous forms like carbon dioxide and sulfur dioxide is coming into the atmosphere. So it is going to affect our environment change and also catastrophes like dinosaur. And the most important point is the significance of large igneous province, that is paleocontinental region. During the generation of large igneous province, large amount of what the continental location we are observing now. During Archean and Proterozoic time, the position of the continents were not like that. So by observing, by studying the synchronous magmatic or contemporaneous magmatic activity in different provinces worldwide, we can define which platon or which continent were neighbors during the geological time. So paleocontinental reconstruction is one of the important parameters that we are going to define by studying. In addition to these four important research frontiers, I am basically working on other four parameters that I have mentioned here. We are dealing with the Archean LIP. In the Phanerozoic LIP or recent LIP, the flood methods are present. But in the Archean LIP, we are mostly focusing the identification of large igneous province through study of mafic dikes. So the mapping of the large igneous province is one of the important component of large igneous province study. So we are doing the mapping of large igneous province by several fieldwork through Google Earth image study and geochronological data. In addition to that, we are also evaluating the economic potential of different large igneous provinces of the Indian. In addition to that, we are also doing geochemical study by studying Holberg geochemical analysis, isotope geochemical analysis, by which we can evaluate the nature of limestone from which these magmas are coming. We are also studying about the emplacement systematics of these large igneous provinces. In terms of emplacement systematic, we are studying about the mechanism of emplacement, whether the magma is coming in a vertical setting or the magma is coming in a horizontal. That can be. In the next discussions, I am going to discuss these four important frontiers on which we are working. As I was saying about the mapping of architecture, Indian fields hold several generations of spectacular massive diagrams which are exposed in the different cratons of the India. Whether you take Dharwar craton, whether you take Singhbum craton, whether you take Bundelkhand, whether you take Aravalli, all these cratons hold spectacular cross-cutting, large and huge scale mapping diagrams. Based on the mapping of mapping diagrams, we have identified three Archean and 12 Proterozoic LIP events in the Indian field. And also the example of Dharwar and Singhbum craton, how this mapping has been done. In the Dharwar craton, this is the map of, I will precisely say that is the diagram map of the Dharwar and Singhbum. You can see several colors are marked. That is for the different generations of mapping. As I was saying, we are basically dealing with the medium diagram. The Dharwar craton from 2.37 GA, 2.37 giganum to 189 or 1.73 giganum, 9 generations of mafic magma tissue has been. And these 9 generations of mafic has been marked in this map different colors. Before that, these this classification of morphic diagram, these different events have not been done. By using Google Earth images, we saw how these morphic diagrams are identified by using Google Earth images. And also conducting several field tests, we have prepared this map from for this is for Dharwar crater and for Singhbum. These are the direct lines that we generally observe how it looks like in the field. You can see boulder structure, three black color outlines, how huge and how large. In the Google Earth images, in the Dharwar craton and Singhbum craton, the country rock is granite. That is, by tonal difference, it can be easily distinguished from the country rock. The country rock is linear bodies, generally it's linear bodies, and these cross-cutting bodies, these are mafic dikes. And as this mafic and ultramafic is composition, obviously their color will be darker to compare. Therefore, by using image, we can identify how or what generations of diagram. This is all about the mapping of diagram that we are working. And second component is the economic potential of large scale. As I was saying, huge amount of mapping magma has been generated during the earth different times. So it constrains its geochemical character, whether it is geochemically essential to hold different metals or other economic units. So it can be done by using several that you by using trace element and rare earth element studies, we can characterize whether when a magma is coming, must interact with the and we need to establish significant interact or it is less interact with the by using several trace like the proxies from trace element from crust and the proxies for trace element for mantle. That is thorium, one of the important element that is considered as crustal element, and the niobium is one of the mantle element. By taking the ratio of thorium and niobium, we can establish whether the magma that has been generated in LIP contain significant amount of crust or not. By evaluating these parameters, we have suggested the Indian LIPs has potential for future study in respect to the economic potential. So I have marked several large igneous province, but with respect to general message, I want to say the Indian LIPs has economic potential for future exploration. And third important parameter that we are working about the nature of mantle source. In terms of research frontier, it is important because in the igneous petrology, all research lies. What is the nature of several people are working on that domain, igneous petrology and geochemistry. People are using different proxies. But at the end of the at the end of the research, we need to define what is the composition, how much percentage of melting has happened, what is the source rock. These are the different parameters. By using geochemical proxies, we have defined the nature of mantle source for the mafic diagrams of the Harwar craton. For the first time, we have reported that apart from peridotite source, that we generally believe mantle is of peridotite composition, that is ultramafic in nature, olivine and pyroxene in composition. But in the Harwar craton, we have defined a specific type of mantle composition, which is peridotite plus pyroxenite in composition. Similarly, as I was saying, another important frontier of large igneous province to define the cratonic neighbor. We know the neighbor of our country during the recent time. But in the ancient time, in the Archean time, in the Proterozoic time, who are the cratonic neighbor of Indian silt? So for that purpose, we define the barcode. We all know about the barcode. But every craton has its own barcode. Barcode is nothing, it defines the magmatic event that has happened in the geological past. That can be prepared by using available geochronological data. We date the rock and we define at which time this rock has been emplaced. So you can see by comparing the LIP barcode of different cratons, you can see craton A and D. These are T1, T2, T3, T4. These are different magmatic events. And A and D has similarity in terms of magmatic event. Similarly, C and D has some similarity in terms of their magmatic event. By comparing these geochronologically dated mafic magmatic events, we can say these are cratonic neighbors. By using this principle, we have defined the cratonic neighbor of our different cratons, that is Dharwar, Bastar, and Singhbhum craton. You can see several matching events are there in terms of the magmatic activity. As I was saying, the magmatic events of LIP scale can help to define the paleocontinental reconstruction. During 90 million years, the Indian silt has been separated from the Madagascar by Marian plume. During 117 million years, the Kerguelen plume has played significant role in the separation of the India on the eastern side. Similarly, the Deccan Plateau that is formed due to the reunion mantle plume that you can see the Deccan large igneous province that is due to the reunion plume. That is the present location of different plume centers in the Arabian Sea or Indian Ocean. So I was discussing about the different research frontiers of large igneous province. So key takeaways from this LIP research, LIP has significant potential with respect to metallogeny. So country's economy is also dependent on the LIP research. So at the last, or at the end, I can say LIP is always important, whether it is human body or in the geological history. Thank you so much.
Thank you very much, Dr. Samal. I think we can have. Yes, sir, no problem. Professor Raghavendra.
Completely different, these two plates. But what will happen in the movement of continents? These two move independently or how?
Every plate has its own independent motion.
No, but in the plate tectonics, what we study, plate tectonics are huge land masses, including this part, ocean part also. Along with that, it moves. But you said these two are two independent.
Because I am saying independent with respect to their composition. If you are talking about continental
But both
fluids,
are moving.
both are moving,
And then both are actually interacting.
interacting with each other.
Okay.
Both are moving, both are interacting with each other.
Okay, okay. Thank you. Yeah.
Any other questions?
Any isotopic age support?
Sir, for all these 9 generation of mafic dioxins, isotopic studies are not available. For
Yeah,
some generations of dikes, isotopic studies are available.
yes, but then because the directions and based on that, always it may not be very correct, I think.
Yeah, sir, I completely agree. The preliminary study is done through the whole rock and geochemical analysis. For most confirmed association, we need to study their isotopic characterization. Whether it can be radiogenic isotope, stable isotope, there are different isotopic systematics are there. So it is required.
Yeah, if there are no further questions, we thank Dr. Samal for his wonderful talk. Our next speaker is the elected fellow for 2023. He is Dr. Professor Ganesh Nagaraju from Department of Biochemistry, Indian Institute of Science. He specialized in DNA repair, genome stability, and cancer biology. And he has coveted honors starting from his PhD thesis. He got the best PhD thesis award, BM Birla Science Award Prize, National Bioscience Award, Sir C.B. Raman Young Scientist Award from University of the Government of Karnataka, J.C. Bose Fellowship, and now he is with us as a fellow of the Academy to give his talk. Professor Ganesh.
Thank you, Professor Apparao, for the kind introduction. And it is my pleasure and then honor to be here. And then I thank Bangalore Academy for this wonderful opportunity. So we work on DNA damage responses in the mammalian cells. And then today's title is RTL1 helicase regulates homologous recombination in the mammalian cells during DNA replication. So the genetic material which is stored in the form of DNA and then chromosomes in all our cells is susceptible to various types of damage. And among different types of DNA damage, DNA double-strand breaks are the most dangerous types of lesions. So which can arise by, endogenously by free radicals and then other stalled or collapsed replication forks mechanisms. And even exogenously by radiations or chemicals, it can induce DNA double-strand breaks. The first thing that cell responds to DNA double-strand breaks is activation of the cell cycle checkpoint, which actually arrests the cells in the, at a particular stage, either in the G1 or in the S phase, and then accordingly facilitate the repair by the repair pathways. There is very good coordination between cell cycle checkpoints as well as repair pathways in order to take care of repairing these breaks and then maintaining the genome integrity. If there are unrepaired breaks or misrepaired breaks, basically it can lead to genome instability, which is the hallmark of the cancer cells and then induce tumorigenesis. There are two major pathways to take care of repair of the DNA double-strand breaks. One is the DNA non-homologous end joining, which is generally often error-prone because of the microinsertions or deletions basically at the break points. So as opposed to NHJ pathway, there is another classical homologous recombination pathway, which always requires homologous template DNA to repair the breaks. And then, so this is considered as an error-free pathway in the repair of the DNA double-strand breaks. And it always requires processing of the DNA ends to generate a 3' single-strand DNA, to which the Recce protein in the bacterial system and its functional ortholog in the mammalian system is the RAT51 protein, which actually polymerizes onto the single-strand DNA, which actually searches for the homology, invades the homologous duplex DNA to generate a D-loop structure, and this recombination intermediate is resolved in two alternative pathways to complete the repair in a error-free fashion. Many genes are known to participate in the homologous recombination mediated repair of the DSPs. So mutations in some of these genes are known to cause various genetic diseases as well as cancer. Some are examples are here basically. The BRCA1 as well as BRCA2 germline mutations leads to breast as well as ovarian cancer predominantly in women. And there are 22 FANG genes basically. Any one FANG gene mutation leads to Fanconi anemia, and helicase mutations like the BLM gene and then Werner gene leads to Bloom syndrome, Werner syndrome. And protein kinase mutations leads to ATM mutation leads to ataxia telangiectasia, and then ATR kinase mutation leads to ATR sickle syndrome. and mRNA complex protein NBS1 gene mutation leads to Niemann-Pick syndrome. These are all chromosome instability and then cancer susceptibility genetic diseases. So that shows the importance of the homologous recombination in taking care of the repair of the DSPs as well as in maintaining the genome maintenance. The RAF51 is the key player, which is the central key player in the homologous recombination mediated repair of the DSPs, which actually polymerizes onto the single-strand DNA to form a nucleoprotein filament, so which actually searches for the homology, invades the homologous duplex DNA, and then initiates the homologous recombination. But the RAF51 filament formation is actually rather inefficient in the cell because, so there is a RPA protein, which is a single-strand DNA binding protein, which actually binds to the single-strand DNA with a higher affinity and is also present abundantly in the cell basically. To overcome this barrier from the RPA protein, there are recombination mediators. Those are like the BRCA1, BRCA2, as well as RAF51 paralogs basically. In mammalian cells, there are 5 RAF51 paralogs. These are named as RAF51B, RAF51C, RAF51D, XRCC2, and then XRCC3. If you look at the protein sequence, it doesn't show high degree of conservation to the RAF51 except for the 2 motifs which are shown in the red boxes. These are Walker A as well as Walker B motifs which are required for the ATP binding as well as ATP hydrolysis. If we make a mouse knockout of any one of the RAF51 paralogs, that actually leads to embryonic lethality, indicating their essential genes. But the bigger question is that the essential functions in the genome maintenance is largely still unclear. And any one gene mutations leads to different types of DNA damage sensitivity in the context of different chemical agents basically, and it shows spontaneous chromosomal aberrations in the form of chromatid breaks, radials, and then gaps basically, and then reduction in the homologous recombination. So germinal mutations in the paralogs are also known to now in the recent times cause basically breast as well as ovarian cancer in the in the females basically. And 2 of the paralog mutations, that is RAF51C as well as XRCC2, so leads to Fanconi anemia. And biochemical studies from a couple of groups basically shows that these paralogs exist majorly in 2 complexes. One is the BCDX2 and other is the CX3 complex. So in our lab, so we showed a new functions basically. These are repair independent functions in taking care of the replication problems in the genome. So what happens is that if there is a DNA lesion basically, so the folks actually undergo remodeling by the helicases as well as even the RAF51 as well as RAF51 paralogs. And these RAF51 as well as RAF51 paralogs actually binds to these reversed forks basically and stabilizes them from degradation by the nucleases. That's how it actually maintains the integrity of the genome. So in addition to taking care of the stabilization of the stalled forks, these paralogs are also involved in the restart of the stalled replication forks. So by homologous recombination pathway, and even if there is a lesion and then that gets converted to replication associated DSPs, so that also is taken care in a recombination mediated replication restart, and then accordingly complete the genome duplication and then maintain the integrity of the genome. So all the homologous recombination and then so the factors which are involved in the homologous recombination is actually important for taking care of the repair of the DSPs as well as replication problems in the genome basically. Their activity has to be controlled, regulated. Otherwise, it can lead to various types of genome rearrangements basically, like the gene amplification, loss of genetic information, loss of heterozygosity, and then different types of chromosomal rearrangements. So there has to be a proper balance between the homologous recombination pathway and then the homologous recombination proteins like the RAF51, BRCA2 as well as RAF51 paralogs, and along with the anti-recombinases. These are all the helicases like the RTL1, PARI, FPH1, BLM, RECUL5. They are all helicases basically, which their functions has been implicated as anti-recombinases basically in trying to regulate the homologous recombination in our cells. So 2 papers basically from the Simon Bolton group showed that RTL1 helicase actually is important for suppressing the homologous recombination during DSP repair. So subsequently, they also showed that RTL1 helicase is a PCN interacting helicase, which is actually required for the genome-wide replication and then taking care of genome duplication as well as genome maintenance basically. So we speculated that possibly RTL1 helicase may be regulating the homologous recombination during DNA replication. So there are multiple factors which are actually required for the DNA replication in the mammalian cells. So the most important ones are the MCM helicases, and then the Pol epsilon, which is required for the leading strand synthesis, Pol delta for the lagging strand DNA synthesis, PCN is the the clap basically, and you have the RPA protein. So, this replication is actually not a very, a smooth process basically in our cells, because, it actually goes through, so with the template damage in the DNA, so if there is a DNTP alterations in the cells, that actually leads to replication stress. If there is a secondary structures like the G4 structures, which actually forms a roadblock for the DNA replication, there is a transcription, replication, collisions basically, all this actually leads to replication stress in the genome. So that is where that the homologous recombination proteins actually come into the picture in taking care of dealing with the replication problems in the cells, majorly in taking care of stabilization of the stall DNA replication forks, and as well as in resuming the DNA replication. We speculated that possibly the HARTL1 helicase may be regulating the homologous recombination during DNA replication. To study this basically, Kumar, who started this work, so we carried out I-pond experiment, that is isolation of proteins on the nascent DNA. So what you need to do is that label the replicating sites with IDU, which is a halogenated thymidine analog, and then treat it with the hydroxyurea, which actually causes genome-wide replication stress, and then later do the pull down with antibody specific to the IDU, and then later do the Western blotting with all these known factors basically. You can see that RTL1 is actually enriched in the recovery forks basically. In addition to RTL1, interestingly, we also see accumulations of the HR factors like the RAF51 and even the RAF51 paralogs. Later, to understand the role of the RTL1 helicase in the replication context, basically, we subjected the cells to hydroxyurea as well as afidicolin. Both actually causes replication stress. And when we measure the cell survival, basically, upon depletion of the RTL, there is a high degree of cell death. But we don't see the cell survival sensitivity in the context of the zeosine, which actually induces DSPs, this actually gives us the separation of some functions basically, its role in the replication stress context, but not in the DSP repair context. So then subsequently, Suruchi carried out further experiments basically. We measured the global DNA replication by the BRD incorporation compared to the control cells. As you can see that the flow cytometry studies basically, here is the quantification, you can clearly see that there is about 50-fold reduction in the global DNA replication compared to the control upon depletion of the RTL1 with the 2 shRNAs. That shows that RTL1 is important for DNA replication in the human genome. So because RTL was already implicated as a anti-recombinase, we wanted to measure the homologous recombination. For that, we used a reporter system wherein this contains 2 copies of the gene coding for the green fluorescent protein. Both is actually mutated. First one has a 5' truncation, and second one is full length actually, but is interrupted with ISC1 recognition endonuclease. So this reporter has been integrated in the human chromosomes basically, in the U2S cells. And then, so we measured after inducing the break with ISC1 transfection with a plasmid encoding ISC1, so endonuclease, which actually makes a break here. And if the repair occurs by the homologous recombination pathway, then you see the generation of the GFP positive cells, which can be measured by the flow cytometry. And here is the data. You can clearly see that compared to the control cells, upon depletion of the RTL1, you can clearly see that there is 4 to 5-fold increase in the homologous recombination. Now that can be rescued upon co-depletions of the RAF51 paralogs like the RAF51C, XRCC2, and then XRCC3. So this is in the context of the DSP repair. Now we wanted to study homologous recombination in the context of the whole genome during DNA replication basically. So what we did is that we subjected the cells to hydroxyurea, which actually causes genome-wide replication stress. And then in that context, basically using the same reporter, we scored for the GFP positive cells. So you can clearly see that compared to the control cells, RTL1 depleted cell shows 4 to 5-fold increase in the homologous recombination, which can be rescued upon co-depletions of the RAF51 paralogs. This clearly shows that in the absence of the RTL1 helicase, there is high degree of homologous recombination events basically, which can be rescued upon co-depletions of the HR factors like the RAF51 paralogs. So in the context of increasing the homologous recombination, we also see increasing the RAF51 foci formation compared to the control cells. Basically, in the RTL1 depleted cells, you see increasing the RAF51 foci formation. That again we can try to rescue upon co-depletions of the RAF51 paralogs. Now this clearly tells us basically there is hyper recombination events are happening in the whole genome basically when there is no RTL1 helicase basically. So if that is the case, can we rescue the replication defect in the RTL1 depleted cells by co-depletions of the RAF51 paralogs? That's exactly what we tested basically. So we perform DNA fiber studies. Again, what we do is that we label the replicating sites with the two halogenated thymidine analogs, that is the CLDU as well as IDU, and then so replication can be studied at the single molecule level. And when we score for about DNA, about 200 DNA fibers basically, here is the raw data. You can clearly see that compared to the control cell, the global DNA replication is actually reduced upon depletion of the RTL1. Now, this replication defect can be rescued upon co-depletion of the RAF51 paralogs, including RAF51 co-depletion. That clearly suggests that basically there is hyper-recombination events are happening, which is actually now interfering with the DNA replication in the absence of the RTL1 helicase. In fact, the the sister fork ratio basically, which is again increased upon depletion of the RTL1 helicase, that sister fork ratio also can be rescued upon co-depletions of the HR factors like the RAF51 paralogs as well as even RAF51. What is the actual, so the direct evidence basically in the absence of the RTL1 helicase, there is hyperrecombination events are happening basically. Whether these paralogs or even the RAF51 protein is actually persistent at the sites of the replicating sites. So to address this basically, we performed I-pond experiments. This is isolation of proteins at the replicating sites. What we do is that again, we label the replicating sites with halogenated thymidine analogs. This is just a DNTP analog. And then, so we do the pull down with the IDU-specific antibody, and then, so we plotted for various proteins basically, so like the RAF51 and the RAF51 paralogs. Compared to the control cells, if you notice in the RTL1 depleted cells, you see that compared to the control, so RAF51, the levels are actually increased in the absence of the RTL1. Similar to RAF51, we also see the enrichment of the RAF51 paralogs like the XRCC2, XRCC3 as well as even RAF52 protein. So this is the direct evidence that in the absence of the RTL1 helicase, the HR factors actually persistent at the replicating sites, and which is actually interfering with the DNA replication. As a result, now when we co-deplete the HR factors basically, now you can rescue the DNA replication defect in the RTL1 depleted background cells. So RTL1 is a PCNA interacting helicase basically. So we wanted to test whether its interaction is required for suppressing the homologous recombination during DNA replication. So we made a isoleucine to alanine. So in the PCNA interacting motif basically. So we studied this. So the HR events using the reporter system. Here is the GFP positive cells. Compared to the control cell, there is 4 to 5-fold increase in the GFP positive cells. That means HR events are high basically, which can be rescued upon wild type expression of the RTL1, but not the PCNA interacting motif basically. So we also wanted to understand the helicase function of the RTL1 in suppressing the homologous recombination during DNA replication. For that, we made 2 mutants basically, that is lysine to alanine as well as lysine to arginine. In the Walker A motif basically, which is actually required for the ATP binding as well as ATP hydrolysis. Again, these Walker motif mutants are actually defective in suppressing the hyper-HR, so the cells basically. So now, using these mutants, we wanted to understand, so the DNA replication basically, yes, replication defect is there upon depletion of the RTL1, which can be rescued upon expression of the wild type RTL1, but not this PCN interacting motif mutant or even the Walker motif mutants. That suggests that RTL1 interaction with PCNA as well as its helicase activity is actually important in suppressing the homologous recombination during DNA replication and taking care of genome duplication. So RTL1 genetic mutations leads to to 2 disorders called the Harrell-Hydrison syndrome. So as well as dyskeratosis congenita, these are developmental disorders with various clinical features including premature aging, so and then bone marrow failure, and patients are also susceptible to developing glioblastoma. So far, about 25 mutations have been identified in these patients basically, and which is actually spread across whole entire helicases basically. So these are all point mutations, missense mutations basically. Now we are trying to understand the functional defect associated with these mutants in trying to understand how they function in the context of suppressing the HR as well as in taking care of the replication problem. So just to conclude that RTL1 helicase is required for the normal DNA replication in our cells, and then hyperhomologous recombination as well as replication defect in the RTL1 depleted cells can be rescued with a co-depletion of the RAF51 as well as RAF51 paralogs, and then RTL1 PCNA interaction and then helicase activity is required for suppressing the homologous recombination during DNA replication. So this work we have communicated recently. And this is my current team, and Kumar started the work, and then Suruchi later, carried out majority of the work what I presented. In addition to Suruchi, Satyaranjan as well as Tarun also contributed to this work, and then I greatly applaud the many scientists outside India as well as within India for providing reagents for our studies basically, and I greatly applaud the funding support from various sources, and thank you all for your attention, and then happy to take questions.
Thank you. So I very much with time after that.
It's one phenomenon called homoplexus. So that's identification. The cost of use of the what you have any comment on?
It's a interesting thing. Absolutely. So complete shattering of the chromosomes. So we do see increase in the chromosomal aberrations, but specifically, so
You have said that the initiated structure what is it? What is the mechanism? What does it actually do?
that's a very good question actually. So one study by Simon Bolton group, they show that it actually disrupts the D loop. There are other helicase mechanisms wherein what it does is that it actually strips the rhabdicon from the single stranded DNA. But that kind of an activity is so for the RTL1 collapses. So that is what is some kind of implication that what we have is. But it is known to disrupt the D loop basically. Whether it has a role in disrupting the RAF1 pillar from the single stranded DNA is something. Yes. Because when we make a helicase mutants, it is destroyed. No, it is actually doesn't do this. Right.
So that's it.
So not in the prokaryote, but in the higher order organisms. No, this is RTL. Basically, the name goes with regulator of telomere elongation. So it is generally present in the membranes. RTL1 not for our knowledge. It's only the HR context. No, it is actually not involved in the expression of the HR factors, but it regulates the HR factors by possibly, as he was asking, what is the mechanism? Maybe it actually unwinds the D loop, but in addition to that, it might have a role in stripping of the RAP1 and then RAP2 parallels from the single strand DNA at the replicating sites. If that is not controlled, basically, then see persistent HR factors. So doing its hyper recombination events, which is actually possibly the reason reduction in the global DNA.
Professor Majumdar.
A big question if on suppress and you also suppress all the suppression of all the decompensation, the consequence of reduction uh,
because of the hyper HR replication is actually compromised basically. That is because of the hyper recombination events, because there is a persistent HR practice at the replicating sites. That is in the absence of the RTL1. So now that replication defect you can rescue by coating the HR factors. That's how we are able to see. Yeah.
of
Suppression of homologous recombination. Okay, so So we see increase in the HR. So in the absence of RL, which you can rescue with HR factors basically. So that because of the increase in the HR, there is a replication defect. That replication defect you can rescue basically. But what happens is that what some was also asking basically, the chromosome instability basically increases when you don't have both the proteins. With singly also, yes, you see increase in the genome instability, and if you have a co-depletion specifically, chromosomal aberrations actually further increases. But with replication is rescued, but with an accumulated, so instability and then mutations, which possibly can lead to cancer.
Are they redundant?
That's an excellent question. Any single knockout is basically embryonically lethal. Although many functions will show sort of similar function, but one cannot compromise with the another. So in the larger context, out of 5 parallels, there is a BC subcomplex, BX2 subcomplex, CX3 subcomplex. If you don't have one, the other partner actually undergoes destabilization. The protein levels actually reduces. So it functions in a complex, and then that is possibly some redundancy is there, but so cannot compensate for one mutation another time.
Good discussion. One last question.
Piece of work. A very technical. Hydroxyurea.
Yeah.
How serious that? When I ask,
Yeah, you are correct. So with sickle cell anemia, mechanistically how it will actually it helps it for the treatment. Yeah. So, but as such, it's needed for the ribonucleotide reductase, which is the enzyme which is required. When the DNTP synthesis is actually reduced, then naturally you will have the replication problem. That's how we use as a replication poisoning agent.
Thank you, Professor Ganesh.
Thank you.
Sciences at AICS Kolkata. He specialized in supramolecular assemblies of functional bisystems and macromolecules. He is recipient of unhumbled postdoctoral fellowship and also PR Scientist Award. Acted as associate. Associate of INSA. Running
At the very outset, I express my heartfelt gratitude to the Indian Academy of Sciences for giving me this wonderful platform and this honor, for giving me this platform to showcase some of our research activities. So I'm a polymer chemist, and we are interested in chemistry of polymers. And in our group, we work at the interface of polymer science from molecular chemistry, which are distinct. Our interest is to bridge the gap between these two areas and create new opportunities in making structurally precise self-assembled functional systems by combining the advantages of these two different areas of molecular assembly and polymer science. And in this endeavor, we use to explore non-covalent interactions such as halogen bonding, dipole-dipole interactions, hydrogen bonding as well, and also play around with crystallization of the polymer for achieving discrete structures of liquid specific function. So in today's talk, I will introduce to you a distinct problem in this area and what is our approach in addressing this problem. So before I go to the problem, let me give you a brief overview of this. For example, protein structure of viruses, cell membrane. So taking inspiration from Mother Nature, chemists have always tried to reduce the cell facility, but in a good way. And like many other groups, we are particularly interested in of pi systems, which are aromatic dyes, primarily because in their aggregated state, they show distinct physical properties, enhanced luminescent properties, enhanced biological properties, charge transport properties. So they provide very interesting avenues for making organic materials. So, but the problem in this field is that if we kind of this is a representative example of a dye. If we introduce a hydrogen bonding amide group, it can self-assemble and can form one-dimensional structures or spherical micelles or two-dimensional structures. But creating such structures today is not a problem. The problem is how to control the shape and morphology of the structures. Like, can we have more control over the dispersity? Because if we look into the biomolecules, they are all monodispersed. So can we control the dispersity? Can we predict the structure in advance? It's a big challenge in the field of cell facility. See, the internal order is beautiful, but when we look into the macroscopic properties, like the bundling and clustering effect, effective surface functional group display is not effectively approached. Even the complexity increases many folds when we have multi-component system. For example, here I'm showing you a dye or a monomer. There can be different ways of organization of this group chromophores in a chain. It can be random, they can phase separate, or it can be block structure. Dynamics of the molecule. So we also take the structure quite different techniques. So this is also our objective that how to kind of achieve this kind of sequence control structure. Because we know from proteins and peptides the sequence matters. So the property of the outcome of the structures are not only dependent on the chemical structure, constituent of the chemical structure, but also on how this molecules are organized in large microscopic. Another important problem is that so far there has been no syndromic strategy by which one can achieve controlled two-dimensional organization of this molecules. We have heard a lot about inorganic materials, but by syndromic approach, how can we achieve dimensional assemblies of purely organic molecules that with the precision of single crystals? So that is also another problem that we want to address. So as I am a polymer chemist, so we look in what is known in the field of polymers. So these amphiphilic glycopolymers is an extensive area of research. We have block of polymers that we have hydrophobic block and the hydrophobic block. So these kind of structures can self-assemble in water, but in a fluid way because the solvophobic block collapse in water or in a polar solvent. And as a result of the block, the hydrophobic block can take different type of structure. But because this is due to solvophobic collapse, so there is again a compromisation in the effective surface functional group display and precision in the internal order. Of late, people have also tried amphiphilic block of polymers that the hydrophobic block is a crystallizable block instead of an amorphous block. The solvent, it might crystallize block by change can be crystallized, then it can be highly ordered back. The other the role of the pulsar segment is to impart colloidal stability to the structure or the bonding structure that is generated through crystallization of the polymer chain. So this is an emerging field of research, and people have worked on different type of polymers which have crystallizing properties. And among the type of polymers, we are particularly interested in polylactides because polylactide is a biodegradable and a biocompatible polymer. It is also generated from renewable sources, and it's a kind of one can also play around with the stability of the polymer chain to use the polymer properties. So, polylactide chain, poly N lactide, attached to a large block, polymeric block, represented by the blue segment, then this can generate a polar solid with diamond-shaped 2D structures. So the single polymeric crystals as characterized in both, and these are newly layered structures. So the crystallization happens like the chain pole, represented by this red thing, and the corona block, that is the blue one. It is as a result of the chain, it explains the corona block to the surface, which imparts colloidal stability to the. So one can see that this is a aesthetically beautiful structure, but it doesn't impart any functional utility to the structure because the corona block is a large body block and its role is to merely impart colloidal stability. So we wanted to explore this molecular design and that can generate similar kind of 2D structures where we do not have this solvophilic giant polymeric segment. Instead, we introduce the chain element. And if we can still achieve the crystallization of this polylactide chain, so the dyes or any functional motif by different color codes can organize on the 2D surface. As a result of this organization, they can come in close proximity and can pass new properties from the 2D surface. And if this crystallization is not hampered, then we can use this as a generally applicable supramolecular tool for controlled two-dimensional organization of different functional motifs. So with this, and we can have multiple dyes or we can have different type of functional motifs on the surface with layered pigment. So with this objective, we first introduced the polar dye. This was a methionine dye. The reason we choose the polar dye because if we have to impart colloidal stability to this structure, there is a similar diamond structure and these are single layered. Now with the advantage that we have this 2D plate which are highly emissive and we do not compromise with the colloidal stability. We see that even for several days the structures are. Next we wanted to see the impact of as I said that polylactide is tidal. You can take this commercially available D and the L lactide and mix it and the recipe mixture in the amorphous one. The contrast between these structures, there are polymer generates ill-defined polymer structures because of the amorphous. But the crystalline one gives display to the structure. Next, we wanted to see whether the dyes that we have decorated now on the surface are the common feature. Yes, we have done extensive spectroscopy study and we find that as a result of the crystallization of the polymer chain, not only the dyes are on the 2D surface, but they come in pi stacking distance and they take part in pi stacking dynamics with high temperature. We were so happy to see that because homopolymer making is very easy. We thought that let us now try another dye and can be the same structure, but now with the other properties coming from a different dye. So, in this case, we have taken a red green emitting dye and the structure now with the ability to emit green emission from the same 2D structure. So we are going to use the predictability and the probability approach. So the crystallization is not hampered. So we can use the same crystallization technique from the polylactide scaffold and now introduce different polar dyes on the surface and get same 2D structure but with different from the texture. So if we increase the complexity, we have now taken dyes and assemble them together on the surface. We choose this blue and green dyes, pyridine and elamine, because they are thread paired and thread is highly distance dependent phenomenon. So if they come in close proximity on the surface, which can lead to energy transfer. And we do see energy transfer and also see that both the dyes are localized on the 2D surface and we have simultaneous blue and green emission of the structure. Now we can also we have done extensive characterization for the energy transfer and if the crystallization can be disturbed by adding a good solvent like chloroform where the polymer chain cannot crystallize, then it will disturb the structure that fall apart and the energy transfer will not happen. Demonstrated by disrupting the structure presence of the good we have now so far we have been able to achieve co-assembly of the dyes randomly on the surface. Now we want to have sequence controlled organization of the two dyes on the surface. So for that, what is important is that we restore the dynamics between the polymer chains. Because if the polymer chains are too fast exchanging, then we will not have a long sequential organization of the dye. So we independently emitting platinate and the blue emitting platinate. And then we mix them together and we see that for several days there is no fray. That means that the chains are not communicating and they are frozen. So that is very important. And knowing that added the polymers in the sequence control manner. We can know these kinds of shell structures diamond know the green emitting platinate then we use the blue emitting polymer and the blue emitting polymer now starts to move from the edges of the green emitting platinate as one can see from the enlargement in shell structure, so which can also be proven. So far we have been using only one homochiral poly L lactate. Now for playing with this system, we saw that what will be the implications if we take two polymers. One is the L lactide polymer and the other one is the D lactide polymer. And we have the local change. We choose this is that known in the literature of the lactide that the stereo complex T and the L polymer they form a co-crystal and the co-crystal is more stable than the homochiral polymer. So we can now play around with the co-crystallization tailor the property of the surface. So that was the objective. See that when we have the green dye attached to the L one and the blue dye attached to the and we mix them together independently also with that. But when we mix them, we also keep it in a structure. And they are also simultaneously able to emit green and blue emissions from the same structure. They also communicate and we can see energy transfer. And so basically now we have another system. The previous system was only both the polymer chain in lactate. But in this case, it is a co-assembly by the donor and acceptor stereo. Apparently, they are giving similar structures, but extensively studied their crystalline packing and we find that they have different crystalline. They have different crystalline packing. They should have different thermal stability because I mentioned that the stereo complex is more stable than the homocrystal. So we find that in case of the homocrystal, it melts at around 5 degrees, but even the stereo complex at high temperature does not melt. That because of the enhanced stability of the crystalline property of this polylactide, the dyes are also disassembling in case of the stereo complex. So we can have a temperature independent thread from the stereo complex while so having prepared that, now that can we increase the complexity in the system further. So in what we did, we have introduced another third dye, externally to this stereo complex to be collected. So basically we grow the structure, then we use that as a host. We are absorbing the red dye. So now on my surface of this 2D structure, we have blue emitting dye and the green emitting dye which are covalently attached to the polymer chain. The red emitting dye which is externally doped on the surface. So the reason we choose these three dyes is that they are so the it will transfer its energy to the green emitting dye. The green emitting dye will transfer its energy to the so in that we can have a cascade energy transfer to the green surface. So we can keep the structure fixed and we can generate more new emerging properties from the green surface. So that is the objective. And we find that yes, these three dyes are perfectly overlapping the absorption and the emission of these dyes are supposed to be overlapping for this cascade energy transfer process. And this is exactly what is happening. So, what is the advantage of doing that? So, if we take the three dyes in the solution, we do not see the energy transfer. Emission to emission, which is coming from the first donor molecule, that is the 5. When we are bringing the three dyes together on the 2D surface, we are able to generate white light emission from the same 2D surface because of the cascade energy transfer. But is now that we have not disturbed the structure. We have been able to achieve the same 2D structure, but now it has the simultaneously emit more kind of
question is pressing last question, and with that we will close discussion.
On the beautifully monodispersed diamonds.
Thank you.
I was just wondering whether you can reduce that to nano size, nanometer size.
Yes, what we have seen that if we sonicate these crystals, so it can retain the shape, but it fragments into small diamond particles. And that is what we are now using to make it even more monodispersed, because this is called living crystallization driven assembly. So what you have is, you have initially when you make the crystals, there are still some dispersity. I would not say, I mean, it's close to monodispersed, but there are still some dispersity. But if you make it into small fragments by sonicating and then allow it to anneal, or you add new batch of polymers from this pre-existing seed, when it starts to grow, it gives more control and precision in the shape of these 2D structures.
Thank you, Dr. Anindita, for the wonderful talk and wonderful discussion. And let's give a big hand. We now have the last speaker for this session, Senior Professor Vishwarup Mukhopadhyay at IISER Kolkata, and he is specialized in physics of high energy colliders, and he is very popular as a popular science article writer both in English and Bengali, a recipient of Santeshwaru Bhatnagar Award and also Distinguished Faculty Award from DAE, Government of India. He is an elected fellow of the Indian Academy of Science and also NASI. Professor Vishwarup.
Thanks for the introduction and for inviting me. The subject of my discussion is dark matter. This is a topic which came into forefront in the context of some astrophysical issues as well as issues dealing with the universe at large. However, this very fact that there is dark matter poses a rather stiff challenge to those who delve in the physics of elementary particles, that is to say, the fundamental constituents of everything in the universe. And it is on this aspect that I'll concentrate, although in a brief, sketchy, and non-technical manner. Now, it is an undeniable fact now that the energy, as far as the contribution to the energy density of the universe is concerned, more than 5 times of contributions come from invisible massive gravitating objects rather than the visible particles consisting of all the elementary particles we know about. However, there is no unanimity as yet as to what dark matter could be made of. If it is, so to say, constituted of some unknown particles, that it implies immediately physics beyond the existing theory of elementary particles based on a certain symmetry group. Let's not talk about that. But it means some extension of the standard model, as it's called, in terms of particle content and perhaps in terms of interactions as well, fundamental interactions. But then, what kind of physics beyond the standard model is behind dark matter remains anybody's guess now. There are suggestions galore, but prediction or interpretation of observations in this respect often depend on certain assumptions and bias on our part, assumptions in the terms of particle physics as well as on astrophysical issues or parameters. In practice, therefore, possibilities can be more divergent than we naively think sometimes. Now, the existence of dark matter in the universe was indicated or suggested by a number of things. The rate of rotation of galaxies, detailed observations of the cosmic microwave background radiation, which is ubiquitous, then gravitational lensing in bullet clusters, formation of structures in the universe, and appearing and disappearing claims on observation of gamma ray signals, excess positrons, or antiprotons in cosmic rays, and so on and so forth. Just to give pictorial description, this is the galactic rotation thing, something which triggered the first speculations on dark matter, where we know from this high school level equation that the rate of rotation of a galaxy at a certain distance from its center is connected with the total mass contained within that distance. And here, for most galaxies, the observation tells us that the rotation rates as observed testify to much higher mass contained than what is provided by luminous matter within the galaxy. Similarly, these bullet clusters is a very strong hint in favor of dark matter being made of particles. Bullet clusters are basically the remnant of two galaxies which have collided, leading to a bullet-like extension. However, the interesting thing is that even if you go beyond the part of such bullet cluster structures, which is visible, even beyond that, you see gravitational lensing effects, that is to say, starlight bending under gravity, telling us that there is some actually localized, concentrated mass, which is obviously made of some particles hitherto unknown. Anyway, now a dark matter particle cannot participate in either electromagnetic or strong interactions, because then it would have led to the formation of supermassive atoms or super heavy nuclei in some corners of space, and the spectra of these would have been very characteristically seen, and spectroscopists have done such a wonderful job of such observations that there is no possibility of that kind. So the main possibilities among several others are those where dark matters are weakly interacting massive particle or feebly interacting massive particle, WIMP or FIMP, and the rest of the discussion will be around WIMPs only. Now, in all theoretical analysis or theoretical formulation of scenarios accommodating dark matter, and also in terms of explaining or rather interpreting data or predicting them, there are some bias and uncertainties involved. First, direct dark matter search has led to the setting up of many, many experiments across the globe, but there the expected event rates depend on the flux of dark matter around the Earth, and there we expect or rather assume that in most studies that the distribution of energies answer to the distribution of so-called Maxwellian distribution and no local fluctuation or clumping. Then when it is weakly interacting massive particles, it has been involuntarily assumed sometimes that the masses cannot be very different from a few hundred GeVs. When we turned on particle accelerators or colliders, like for example the Large Hadron Collider, we assume that the signals of dark matter will consist in missing momentum or energy because dark matter particles are invisible. Then it is assumed, not always, but involuntarily, as I said, is that dark matter necessarily is a stable particle. And last but not the least, when dark matter annihilation and decay is concerned, because they lead to what is called the relic density of the universe, where there are clear limits, that the annihilation of dark matter and decay when it takes place is into two particle final states. Now, departure from any of these standard assumptions, or more than one of the standard assumptions, will alter dark matter search paradigms in big ways, and also the standard conclusions of the analysis of data as well as prediction of scenarios. Now, our judgment, therefore, may be modified in some cases. I'll just talk about a few illustrations of this. First, a scenario of trans-TeV dark matter. Tera electron volt is one order of magnitude above the scale at which weak interaction prevails, or rather the so-called phenomenon of electroweak symmetry breaking takes place. And due to some semi-speculative coincidence called the WIMP miracle, which I am not elaborating here, people thought the dark matter mass, WIMP dark matter mass cannot exceed this mass scale. However, on a closer scrutiny, this doesn't kind of cut any ice. And in fact, there can be dark matter particles weakly interacting, which can be in multi-tera electron volt regions, all the way till close to 100 TeVs, where there are some theoretical problems of unitarity of scattering matrices which can kind of jeopardize, which can be jeopardized. However, for masses exceeding a TeV, any collider experiment like the Large Hadron Collider, which is the most energetic particle accelerator produced so far, is unlikely to see, unlikely, almost it's impossible to see that. Also, the direct searches become ineffective because the rate of cross section of the ambient dark matter cloud with direct search experiments or detectors, this is a kind of elastic scattering process that we think of, there the rates fall rather miserably. Therefore, in such cases, for trans-TeV dark matters, one has to depend on indirect signals. Indirect signals consists of, as I said, gamma rays, cosmic ray studies, and so on. These have put certain limits. However, the regions of a dark matter parameter space that can be probed leaves certain regions unprobed as far as gamma ray signals, cosmic ray analysis, etc. are concerned. One possibility that has been widely studied recently is that of radio signals. Now, in certain galaxies or clusters where dark matter concentration is high, their pair annihilation may lead to directly or through cascade decays electron-positron pairs. These electrons or positrons undergo spiraling motion under galactic magnetic fields, in the process having several kinds of electromagnetic energy loss, but at the end leading to synchrotron radiation, and the most pertinent synchrotron radiations that we'll talk about are in the radio range. So radio synchrotron signals turn out to be interesting possibilities if there is some stellar structure consisting of large dark matter concentration. A potential sources of this kind are dwarf spheroidal galaxies, examples being things like SEG-1, Draco, Reticulum, Ursa Minor, and many others. These are in 10 to several tens of seconds, megaparsecs away from our Milky Way galaxy. Now, these dwarf spheroidal galaxies have large dark matter density. The star formation rates there are low, which makes backgrounds for spurious processes less disruptive. And what can be a problem are foregrounds or radio signals from some spurious source between us and the dwarf spheroidal galaxy, but these can be eliminated with the help of very high resolution radio telescopes. And one such telescope is, which is in the coming, is the Square Kilometer Array telescope, which is likely to start operation towards the end of the current decades, where you have dishes in South Africa as well as Australia. Now, some recent studies by us have shown that for dark matter masses in the 5 to 8 TeV range or even some 10 TeV ranges, radio telescope for many scenarios can lead enough and distinguishable signals rising well above the threshold of observability of the SKA in the range 100 megahertz to 4 gigahertz mostly in something like 100 hours of observation. And this can make the parameter space of a dark matter scenario spanned by the mass of the dark matter and rate of its annihilation amenable to detection. This is a typical example of one particular scenario. I'm not going into details, where this is the mass of the dark matter. Against this, we plot the annihilation rate. For any what's happening? For any dark matter mass, this gives the maximum annihilation rate possible consistently with gamma ray data. Actually, these have been slightly improved of late, and these are the maximum annihilation rate consistent with observation, or rather non-observation of additional positrons or antiprotons in cosmic rays. We can clearly see that there is a big region which can rise above the threshold of detectability through SK, and once you are above this band, this shows the minimum. Why band? Because of uncertainties in astrophysical parameters. Two quantities that very significantly affect the radio synchrotron emission are the diffusion coefficient of the galaxy within the galaxy and the magnetic field of the galaxy, which is the all important quantity driving synchrotron radiation. Now, very qualitatively, I would like to rattle off the factors governing the radio flux for large dark matter mass, trans TV mass. Of course, large annihilation that helps, but then on top of that, the spectrum, the rate of interaction, and the etc. matters. Basically, a large mass on one side suppresses rates, but provides more phase space to the electrons that are produced in interactions, and therefore you have a larger supply of electrons which through certain electromagnetic processes are in the right ballpark of energy to emit large fluxes of radio signals. And of course, large diffusion coefficient will cause loss of electrons faster, and larger magnetic field will keep bigger, rather larger number of spirals within the galaxy, which will sustain radiation. So these are the two factors which are important. Okay, the two other topics that I quickly covered, and if the third one is not covered, it's not a problem, is mass limits on a WIM dark matter. Now, some studies based recently on direct searches, indirect signals, various kinds, radio, neutrino, etc., and relic density observation from cosmic microwave background radiation indicate that WIMPs up to something like 100 GeV is exceeding some, I mean, up to something like 100 GeV is very, very strongly restricted unless you have some very, very contrived models. However, there are also suspect. For example, direct search limits as obtained through the sheer quantum mechanical calculation also make certain intrinsic assumptions which have sometimes been questioned. Then the indirect signals consisting of various kinds of things that I have already mentioned, they depend on the annihilation branching ratios. When two dark matter particles are annihilating, what they go into, there some of our theoretical bias based largely on the standard model of particle interactions guide us, and therefore we are somewhat restricted in the listing of possibilities. If such bias is not there, it was shown by these people just before COVID broke out that a WIM dark matter going down all the way to 10 GeV is not at all ruled out or disfavored if you can allow, or rather allow the possibilities of it decaying in many other ways. And when we were recovering from COVID, we showed that actually it is more unrestricted than that. There, in all studies, it has been sort of ignored that the dark matter particles can annihilate and annihilate dominantly into a pair of neutrinos. If that is allowed, and that is allowed to be large, then anything above something like 10 MeV can be consistent with all observations. And you cannot go down below 10 MeV in the present setup because that will affect the heavy element abundance rate, which is rather accurately measured and tampering with which will bring in a lot of new physics possibilities into which I am not going. So given all this, the branching, there is a region of the parameter space where you rule out the rate of annihilation of dark matter in certain ranges. And these are all to lie above this shaded dashed lines because that corresponds to the maximum relic density that you can have. However, if there are rates above that, they are constrained by various kinds of gamma ray, X-ray, etc. observations. And therefore, when neutrinos come in, They make some of the observed possibilities invisible, and therefore you have larger amounts of phase space opening. And if the neutrino branching ratio tends to be something like more than 99%, then any mass 10 MeV onward becomes allowed. Well, I can just briefly mention, I have just 1 minute left, that there is no fundamental principle ensuring the absolute stability of dark matter particles so long as its lifetime is not exceeding 10 to the 25 to 27 seconds. Now, a possibility consistent with current observations and highly studied in terms of constraints is that a WIMP dark matter freezes out, becomes decoupled from the cosmic soup through weak interactions, but decays slowly in various channels very, very slowly. And in these cases, in a duospheroidal galaxy, they can produce electron-positron pairs, and as we have seen, lead to tangible radio signals at the SKA. And this is the region of parameter space where this red part cannot be at all approached, whatever be the situation. This part is ruled out and probed in 100 hours of SK in certain scenarios, and this is ruled out always. So I think I have just come to the end of my talk, and I end with this observation. A really good scientist is one who knows how to draw correct conclusions from incorrect assumptions. Thank you.
Thank you, Vishwarupji. And any questions? Yes.
Yes.
There it is.
Well, it's a very interesting presentation.
Thank you.
As a layman, in some rough sense, I am curious to know the following. In the earlier part of your slide presentation, I found that the size of the dark matter is greater than 5 times the size of whatever is known.
Size of the, not size of the dark matter, the contribution of dark matter to the energy density of the universe is something like 5 times or more of the contributions from visible matter. Yes.
The energy contributed is 5
Yes.
times.
Yes.
When to me somehow it is a paradox. When infinities are involved, I want to know how you compare two infinities, 5 times something or something like that.
The energy density of the universe doesn't have to take a so-called infinite universe into account. You take galaxy after galaxy and you study the content of matter in the galaxy as evinced from the luminous part of observations. And you look at the, for example, times
So
the dark
that
matter.
means we are not dealing with total energies, some sort of
No.
density or
That is
something.
because of another assumptions. Going at a scale where galactic distributions can be assumed as continuous, that's called a cosmological scale. That is where the universe can be treated as of our present understanding as homogeneous and isotropic. So if that is so, then if something in this direction, if you go 3000 megaparsecs away, it will be very, very different.
So that means you are dealing with finite numbers there.
Yes, from finite we extrapolate through our faith in the idea of homogeneity of the
I got
universe.
a different idea because
No. It's like that.
Thank you, sir. Thank you. One last one, because we have already.
If you go sensitive enough.
Sorry, can you repeat?
Consider that everything will emit radio waves if you go sensitive enough.
Yes.
Now, is there any particular signature which your signal will show compared to other things in the sky?
In fact, somewhat technical.
Maybe we can have that discussion during.
Frequency integrated class. In some frequency, the entire range, 100 megahertz, 4 gigahertz. If there are at least one or more bins of frequency, where we can see the signal strength rising at least 3 times above the conduction threshold there, we can see. So we can see that there is something. Now, what you are saying means actually the analysis of bit by bit in terms of frequency reveals more information of the diversity.
Okay, thank you.
ecosystem in the Western Ghats. And there are also talks that deal with how satellite data can be used to actually know more about our Western Ghat ecosystem. And finally, have a talk about the potential hazards of our economic zone in India, which is given by an expert from Goa itself. So without much delay, let me get this started. I would like to welcome Dr. C.P. Singh from ISRO Ahmedabad. He joined the Space Applications Center in 2002, and he's currently working as a senior scientist in the Earth and Planetary Sciences and Applications area. And he has got the prestigious PR Pishardi Memorial Award from the Indian Society of Remote Sensing and the President's Appreciation Medal from the Indian Society of Geomatics. So on behalf of the Academy, I extend a warm welcome to Dr. C.P. Singh and invite him to deliver his talk on phenological monitoring, sensing the pulse of the forest ecosystem.
Thank you very much. Good morning to everyone. First of all, let me thank Indian Academy of Science for inviting me. It's a great honor for me to be here among this August gathering. So without much delay, let us talk about this particular subject. You may be aware, I think you must be observing around you the phenology quite often, watching your trees around you. But it's a different perspective that I'm going to bring, how we can see these phenomena and through remote sensing, through satellite remote sensing. So let us see what are the things that we can deduce from remote sensing perspective, which generally our eyes cannot catch, on regular basis. So what I'm going to talk about is phenology. All of you may be aware. This is nothing but the nature's calendar, nature's own calendar. In a way, it is all amalgamated by various factors, how they respond, the things, biological things respond to various factors. It takes care of all the plants, animals, behavior on an annual basis. So it's basically a horizontal science which cut above all the other sciences, and it has various implications, whether it is related to health, medicine, or shifting of timing of flowers to whatnot. So we'll try to see what all we can gather from this and how we can sense climate change impact using this particular science. So as we have discussed that phenology is basically governed by weather, climate, apart from the other abiotic factors. It's basically, it triggers various chain of events in the ecosystem. Then only we start having the leaf flushing or leaf falling. So the senescence and leaf greening is all dependent on chain of events which is triggered generally by the weather parameters. So very sensitive parameters. If we are looking into the climate change and global warming, which is happening, there are very sensitive ecosystems which are responding very differently And the chain of events happens in the ecosystem itself, where even the animals and other plants get affected very easily. Like this is one example where if leaf fall and flowering is completely deferred in a particular year, and the migratory birds are completely unaware of that particular thing happening there, they may be completely lost because of whatever heavy investment they are doing in terms of laying the eggs and trying to bring their offsprings. They may not be getting enough caterpillars to have for their feed. So such things are happening world over. They are getting reported, and these are very climate change impact assessments are being done based on that. So we have to be sensitive about those things. Forest types, when we see India is bestowed with many and varied forest types, around 16 forest types that we have as far as Champion and State classification is concerned. And all are manifestation of the weather that we have apart from other biotic and abiotic factors. So weather plays a crucial role, and based on that classification is done. So any change on the weather patterns related to temperature or rainfall which is happening will change the functioning of these ecosystems, and the entire atmospheric balance of the gases exchange, or you can say the water and energy balance itself will be compromised, or it will be different completely. We want to predict that what will happen to the phenology of this particular forest, how the gases exchange will happen, how the programs. So coming to the phenology, how they can be observed. There are various ways of observing the phenology, whether, routinely people in the Europe have been, reconstructing our past, alpine tree line records, niche modeling, snow line connection, lichenometry, and various other things, even trying to understand whether invasive species are growing because of the global warming in those regions. And apart tree line, the derivations using the LiDAR data, because the definition of tree line is any tree which is growing there with a certain density having height of 2 meters. Now that height thing was not getting captured in earlier remote sensing domains. So of the impact that we are having. We are even trying to understand how the lichens are, behaving, whether they are growing more in terms of in response to the global warming, and we are trying to understand the spectra, trying to match filtering with the satellite spectra and map it properly. Not only for the Indian regions where you don't have higher plants, only cryptogams are there, even for the Antarctica. Recently, this study was published where we have mapped the abundance of lichens in the Lasman Hill area. So coming back to the phenological variability, the tree lines are very sensitive, as I said, they are shifting. What is happening to their phenology? So that also we have tried to understand whether they are shifting or not. And we have found in Sikkim, Himalaya, there is a substantial shift in the phenological trend. Even in Arunachal Pradesh, the similar thing are happening. So based on these observations, we got encouraged and we have made a big protocol of having, similar to Himadri, can we have a phenomet network? So where we can observe the phenology, meteorological parameters, and we can ultimately have in the country, we can have a phenomet model developed so that we can gain a predictive ability for all the biomes, and we can, really forecast in future that what will happen to what will be the fate of the forest in India. So these are the places where we have actively gone and, already installed the phenocams through various programs. So there are some glimpses of those places where phenocams are installed, and even meteorological data are getting generated. Data processing is being done. Interestingly, we have seen in Tungnath, Himalaya, that there was 1 year where rhododendron has missed its flowering. And because of that, there are a lot of issues which are happening to the birds. And this flowering was missed because of the heavy snowfall in 2020, which because of that, there was a warming in the soil and the vernalization process could not take place. So this is the reason, and we have published it. There are some recent observations from the Dalma site, and we can use even the phenology observations for generating the net primary productivity, because it's observing the greenness over the year, and that greenness is a function of CO2 sequestration. So that can be modeled very well. So net primary productivity can be generated. Apart from that, there are various other offshoots which can be used in terms of phenology, because phenology gives the forest floor a loading of fuel. So forest fire starts from that itself. So we can predict that also. Thank you very much.
Thank you, Dr. Singh, for an interesting talk. We'll have some questions.
Dr. Singh, thank you for a very exciting presentation on phenomet networks. That's a very in thing now. My curiosity was, as I understand, phenology is a cumulative attribute of an ecosystem per se. So it's more towards a synecology approach. With this technology, are you able to also track nuances of individual species, and particularly the faunal groups?
Yes, very interesting question, sir. Yeah, actually, I missed to even inform this, that basically at landscape level, satellite can see, because generally we have 24-meter kind of resolution or 15-meter, 30-meter resolution. So landscape level. So one pixel is having amalgamation of all the trees, Now deciphering the information for each individual, you need something very high resolution. When you take very high resolution satellite, then you lose the repeativity, which is very crucial for phenological observations. So what happens, we are installing here the phenocams. Phenocam is giving you the landscape view at a very surface level. So you can, the various groups of trees can be very well identified. So you can say that teak is having its own senescence right now. Some trees have blossomed, what kind of trees. So you can, in that landscape, you can very well do that. Further to answer, if you can have drone-based applications where you can have automated drones which can fly every day and then take the observation, that kind of applications can be thought of. Otherwise, from satellite, but now in satellite also there are a lot of train of satellites are getting built. So you have like 3,000 satellites in the space, and they are all observing. So they are all commercial players which are doing that. They can give you daily data at very high resolution where even species level observation you can do. So they are giving 30 centimeter kind of data. So species level. Yes. Right now, we have built the entire phenomet network. So there are places, basically all these places are biosphere reserves or national parks. So where anthropogenic activities are close to nil. Hotspots.
90% has already gone. It's all because of the human activity. And directly because of the human activity, invasive species have come. Not climate change, not because of the climate change. Anthropogenic events have induced invasive species to enter. So this parameter, I think, is very important.
We have found that technology can be okay. The classification of itself is possible, one domain of application that I showed. We are able to do that. All these species are getting classified for vesicular students because they are evergreen. You can always the code of law that this area has been served from yes, because the technology is completely changed. This was applied time, and now the physics has down, and because of that technology is.
And one more small question. I would need, I think he can hear. The year-to-year basis, you are monitoring the flora from cryptogams to flowering plants. How can you do this? Year-to-year basis, it is impossible. 50 years, 100 years, we have not been able to do.
Cryptogram example that I showed you is not possible if you are observing in a wooded scope. It's only possible in area which is there is no other botanical element than the lichens or cryptogams. Like I'm talking about Antarctica, I'm talking about the Transimarga. That places there you can observe. But in Australia,
Selected species, yes, we can do. Selected 1 or 2, 10 species, we can do. Yeah.
Can you move on to the others?
very well. In fact, so big program is the IT cluster for mining where middle position the dust loading on the tree is deep. So we have tried to quantify where. From remote sensing, hyperspectral data, we can quantify that what how many of dust loading is there on the tree. This is that helps assessment processing to be good application that hyperspectral processing. In fact, in coming year, we are going to see the first phase of sensing light from Asia, taking it to various forested area against where 400 bands, creating spectroscopy is a concept which we are trying to the aerial flights.
I was also curious to know, I mean, are the migratory patterns of birds also a phenological indicator of climate change? Because they talk about tree lines here. I would imagine that it should also have migratory patterns. Have they been studied?
But from communication technology and navigation technology, we can use it. You need to color them and then try to see that when and where they are landing, but not from remote sensing.
Just one quick question, yeah.
So thank you so much for the talk. I wanted to ask, so you mostly focused on observation and tracking of this phenological shifts. So if you focus on any particular attribute, let's say this alpine tree line, does time series data analysis and does detection of structural change points play a role in this, in the analysis of how this change is happening? Some sort of time series data analysis, does it play a role in this?
Yes, yes. In fact, it's completely based on time series analysis only. The biggest time series that we have currently is NOAA AVHRR. That is from NASA. So from 1960s to till now, every 8 day. But only problem is that it gives you very coarse resolution data. Okay, so 8 kilometer data. So at 8 kilometer pixel resolution, whatever you want to analyze, so global study has been done. We have also done in alpine tree line also using NOAA AVHRR data, and we have tried to make sense out of it. But the best part would be if you can have a very high resolution data, high repetitive resolution, temporal resolution, then it will be much more meaningful. So, but we are waiting for that kind of archival data to happen, because the studies when it started in remote sensing, they were more into trying to understand the meteorology and the general mapping purposes.
Thank you, Dr. Singh, for a very interesting talk. We'll move on to the next speaker. It's Rajiv Kumar Chaturvedi is from BITS Pilani Goa campus. So he's an assistant professor at BITS Pilani. He's an alumnus of IISc Bangalore, where he did his PhD in ecology. And he's currently involved with India's National Greenhouse Gas Inventory Program. And on behalf of the Academy, I invite him to give his talk on forest monitoring studies in the Western Ghats, his work on connecting satellite and ground-based observations.
Thank you, sir, for this kind information. I hope I'm audible. Yeah, so, more volume? Okay. So thank you very much for giving me this opportunity. I thank Indian Academy of Sciences, also BITS, for providing me this very valuable opportunity. And I will take forward where Dr. C.P. Singh has stopped, because he has been the funder, like he has funded this research, and also we collaborate. But what we are doing, we will be focusing only on Goa. He has given you a broad overview of the entire country and beyond, but we are just focusing in Goa in one site at one plot. So that's what we will be focusing on. And but before that, I want to give you some idea about the broad context of our research. So you can see that the temperatures, especially in the Western Ghats, are increasing. This map shows the differential temperature increase. So India has significantly warmed in response to climate change. You can see southern India has warmed higher, especially the Western Ghats. And also in terms of rainfall, you can see the southern Western Ghats has experienced a reduction in rainfall significantly, whereas the northern Western Ghats has increased rainfall. So we are asking this question. So the fundamental question that we are asking, and also going forward, we are expecting a lot of, so whatever climate change has happened so far, that is just the tip of the iceberg. What we are expecting it to go further, much beyond that. This work of ours provides an idea about what kind of climate change we are expecting in terms of temperature rise and rainfall changes in the country. So the question, the critical question that in my lab we ask, this is the broad question, very, very broad question, how, and we are interested in the Western Ghats, basically, how is climate change currently affecting Western Ghats in the last 100 years, last 40 years? But see, we can't talk about 100 years, so because we don't have, see, all we have is the satellite data. We don't have the ground observations. In the developed countries, USA, Europe, etc., we have 100 years of, like thousands of ground inventory plots are there, which are maintained for very long period of time. In India, we have dearth of such measurements. ISC Bangalore has one such plot, which is being maintained for 40 years, but that is a Herculean task. It has its own structural limitations to carry on this kind of work. Secondly, we also want to understand how, because whatever climate change has happened, whatever even climate change along with the anthropogenic disturbances, all these things will continue to happen. So how forest ecosystems will change in going forward in future? This is the critical question that we are interested in asking. These are the questions that we are asking. So what we have, so this is a work that I contributed. This is a global work. So again, this is based on a new satellite product that is that became newly available. This is leaf area index. Based on this product, and the general understanding is that the Earth is sort of greening. Possibly because of the carbon fertilization effect, possibly, for example, in countries like China and also in India, agriculture is the dominant driver of greening, not the forest. But in China, a lot of new areas have also been planted, etc. So the dominant understanding in the field among the experts has been that the Earth is largely greening, but we wanted to go deeper into the surface of it. And recently we have done this study with IIT Bombay, and so here what we have done, we have segregated the entire thing, so we have especially looked into the forested ecosystems. So while you can see this leaf area index, the first column, the leaf area index is increasing all the way. It is increasing all over India, it is increasing the forested grids, it is increasing the croplands. So the earlier map is correct, there's nothing wrong in that. But if you look at other matrices, see, so if we look at, say, for example, net primary productivity, which is a very important indicator of the ecosystem's health, because that is what is actually being generated, that's what actually goes down the trophic level. So you can see that in terms of net primary productivity, the forest ecosystems are the ones where the net primary productivity is declining. And the decline in the net primary productivity is not happening all over the country. The decline is limited in certain areas, which again, areas like Western Ghats, Peninsular India, and Northeast India. These all Indias are like, so we all know that Northeastern India as well as Western Ghats, these are some of the biodiversity hotspots in the country, very, very important. And these also happen to be the climate change hotspots that we have shown the earlier figures. So probably we are not putting any linkage at the moment. It may be due to anthropogenic drivers, especially in the northeastern India. A lot of deforestation is happening. It is certainly the anthropogenic driver here, but in Western Ghats, we are not yet sure what is the thing. So we wanted to investigate further, and this opportunity that Dr. C.P. Singh has presented before us in terms of the phenocam and the phenological modeling, we also understand that phenology is one of the most important indicators of the shifting climatic regimes, etc. So, but let me tell you about the state of the affair. I think Dr. C.P. Singh has also briefly touched upon it. We have only 37 ground stations in India. He said less than 50. I'm giving you exact number. 37 studies are like 37 plots are there where the data will be consistently recorded. Out of these 37, only 6 are long term. When I say long term, the long term is only, the maximum is 40. others are 15 to 20 or something. So we are really data deficient when it comes to the ground observation. So ultimately, for making any large scale conclusion or something, we have to rely necessarily on a satellite data, and satellite data has a lot of limitations. And so that's why we need to discover something in between near surface observation, which is phenocam. And also, we have been fixated so much with the phenocam because it is really cheap. It can be deployed at a scale. I will come to all those things, but despite the fact that it is extremely cheap, it is a very easy to operate, affordable, efficient, very nice tool. In India, you can see that this is a paper that my student Karun has published recently. It has been accepted, not published yet. It shows that especially in the like in the global south, India is part of global south. So especially in the South Asian region, there's hardly any observations using the phenocam. This is, and whereas in countries like America, USA, Europe, and Japan, and even China, we have a lot of studies which are going on using this. So there's an opportunity that India takes a lead. India shows the global south at least how these kind of very cheap, efficient. So let me take you to, so this is the phenocam, how it works. So you can see, this is a very important slide. This is the reason why we are fixated with the phenocam. So you can see phenological camera, what it does, or phenocam, what it does basically, it takes, like it records the meteorological variables. Also, it takes pictures. It will take pictures in RGB band and IR band. So this is what the phenological camera will do. So, but by doing this, it can do all the operations. It can create all the indices that satellites can possibly create. Also, it can create things on the ground. Like it can also create things like observations of bird, buzz, leaf fall, etc., with precise, like with very good accuracy. You can see the R value is pretty high. So it is like an intermediate linking tool. So it can also create indices like NDVI, normalized difference vegetation index. It can do things which is done by tools like eddy covariance towers, etc. It can compute gross primary productivity, etc., which is and with pretty high accuracy. So that's why we have really decided to invest into this technology, and we are part of that IGBP network. And as part of the IGBP network and the funding that has been provided by, kindly provided by ISRO, we have established this tower in Netravali forest in Goa. So you can see that this forest here is at the tri-junction. It is right on the border of Kerala and Karnataka. It is, to go to this place is really very difficult. There's a dirt road, goes for 7 kilometers, and the road is like this. In the rainy season, it's very difficult to even reach there, but still we are doing there, and it is situated here. And we are like measuring a lot of like meteorological parameters, as well as we are taking the images. So these are the meteorological parameters which are written here, and what we are getting, so the kind of, we are getting a lot of meteorological parameters. Something what unusual we have found in the last 1 or 2 years of observation is that we have found very high levels of like the daily temperature rising, especially this year, March. It has been really very extreme. You can see even in that height, I expect the lapse rate to do some correction there, but the temperature was 38 point something. So the maximum temperature. The humidity was close to 20%. It was like, so it is quite an interesting observation that we are getting. We are also monitoring the rainfall, wind speed, and various other parameters here. And what we are doing, again, we are also getting the pictures in RGB. And then using these pictures, as I said, we can create indices such as GCC, such as NDVI, etc., which you normally get from satellite images. So we have created this end of season, start of season, as Dr. CP Singh has been saying. Now, our ambition is that by using this kind of 1 year, 2 year, or 3 year of observations, we will be able to trace back the satellite data, and we may be able to bias correct the past historical data sets and get some idea about how the phenology has changed in the last certain years. Also, what we are doing, we are trying to forecast this by establishing relationships between. So in our plot, we have found that T min, temperature, minimum temperature, is the variable which is having the largest effect on the phenological cycle over here. And then we are also going forward. So this is, we are trying to understand how we are trying to understand the current impact of climate change on the forest ecosystems based on just one plot. We are also like collaborating with ESA, and we are trying to develop a model that is amenable to the ecologists. I have been all my life like working with the dynamic global vegetation models, but these models were written by climatologists. These models are like grid-based models. These models were written primarily for the climatological parameters. For them, they wanted to take certain things from forest, but I want more. I want it to be ecologically amenable. I want a plot-based model. So we are like working with a very rich network of people for developing this model, and we are providing the data from our site in Western Ghats, and hopefully like after some time this model will be ready and we will be able to use it for understanding how to manage our forests better, how to try to adapt to the climate change. And this model requires a lot of data, like a lot of data needs are there. Plot level demographics, trait-based parameters, meteorological parameters, tree architecture, you name it. It's n number of parameters that are required. So what we have done to meet these requirements, we have gone beyond the mandate of the IGBP project. The mandate was to just install a phenocam tower, but we've gone beyond that mandate. So you can see that this is our phenological camera where we are looking at. So if you see what is happening near to the camera, you are just looking at 2-3 trees. As you go farther, you have 10 to 50 trees. As you go very further, you have 10,000s of trees that's there. So the field view of the camera is increasing. And what we did, we got a very convenient place here to lay a plot. So we have laid a plot here, and that plot, what we have done actually, we have done so we have like laid a plot here, and you can see the plot is here. The plot in terms of geographical location is coming somewhere here on the top of the hill. And we have been able to link, and for linking satellite data, what we have done, for laying the plot, we have done total station survey, we have done DGPS survey. DGPS survey is very highly accurate. It gives you accuracy of 1 to 3 centimeter. And by doing this, we have been able to locate each and every point, each and every tree, literally we know, and then we are able to connect it with the satellite data. So that's what we have done. And we have followed all the international protocols in setting up this plot, and so we have a very good idea about this. And based on this, what we have done, all the trees which are more than 1 centimeter have been tagged in this plot, and we have measured the DBH, which is the girth of the tree. We have measured the height. We have measured a lot of parameters in the tree, and we have these markings are there. So based on these, we have created the carbon stock values. What we find, since I don't have time, won't go in details of that, what we have find that we have approximately 1,200 tons of carbon dioxide that is there stored in this just 1 hectare. This is like, compared to that, if you see, this is the value that we are getting in Netravali is pretty high compared to other values that are published in literature. So our forests are pretty rich forests. We have a lot of carbon fixation that has happened there. At the same time, we have also looked at the diversity aspect. So if you look at the tree diversity aspect, we have found 5,375 individuals in our plot. Out of them, it is like a semi-evergreen forest. So we have 61% of species that are evergreen. And then, but what I want to highlight here is that we have done everything. So so the species richness, we have found 84 different species in our plot. This is again quite high compared to what is reported in the literature elsewhere. But you can see that if I was checking the IUCN list, 2 were endangered species, but 47 were not even evaluated. So what we are trying to argue that the IUCN red list, maybe of a species, maybe we should create a red list of ecosystems, because species are innumerable. Probably it is not possible. And even the most dominant species, that is the Olea, even that one is not even evaluated. So I think the red list of species is having serious deficiencies, and we need to propose about a red listing of the ecosystems rather. And then we have also gone ahead and we have measured a lot of trade-based parameters. So this is LICON instrument. Again, courtesy ISRO, we have received this instrument, and we have been able to create some of the photosynthesis rate sort of, and which these kind of things will go into the model development, and like especially so for key species, we have done this kind of analysis where we have varied the CO2 concentration in the atmosphere, and have seen the effect of that on the tree. Our experiments are going on. We'll do more experiment in order to support. And then we have also collected leaf area index information simultaneous to this other information. And I will not take much time. We have also taken, so one of the requirements of the model development you may have seen is the tree architecture. We have taken the terrestrial LiDAR to this site. And how can I play this? It's playing, yeah. It's playing. So it gives you an idea about the forest. And as sir has suggested, we have selected certain species, individuals. From LiDAR, it is quite difficult to get this. This is enormous amount of point clouds. But we have selected only few individuals. We are working on them, trying to create their tree. For example, we have exactly complete information about all the individuals, their tag numbers, their species, etc. So we are creating the architecture, tree architecture information from this kind of data set. And this video also shows you by the kind of terrain we are negotiating, how to stand there. So this is just to illustrate, and yeah, this is also part of our experiment. We are thankful to the team from IT Roorkee who has come and supported us in this kind of endeavor. I think my time is running out, so I will now conclude this. Some of the concluding remarks that I would like to make is that in India we have lack of long-term observations. It is important not only for a sector. We have n number of, even in Goa, we have enormous, like lot of diversity, lot of ecosystems are there. We need to monitor them. If we monitor them, then only we'll know what to do with them. So this monitoring is lacking, and this camera is a very good idea. It may be very cost effective. We are working on that. Maybe a startup will be built, which will build these phenocams and will be given. So we have all those ideas. And that's why in this context, satellite data emerges as a very useful tool. However, satellite data has uncertainties, so we needed this cost-effective tool. And then we are also, so conventionally, the dynamic vegetation models were developed as an accessory to global climate models. With grid structure, tree diversity represented as PFTs, we present an alternative approach of dynamic vegetation modeling here, that is eco-evolutionary plot-based model that better represents biodiversity and provides insights into solutions. How does what a tree does when a drought is there, how it copes. That is because like those kind of learnings we need to get. I'm hoping that our model will be able to do that, but probably it's a tall order, but I think time will tell us. Thank you very much for your kind attention. I would like to thank all my collaborators, all of them. It's a picture taken in the forest with all their equipments, and the funding agencies, everybody is listed here. I especially mention like Mr. Karun and Nasla. Here, these are the backbone of our work. They are here also. So they have done all the work actually, and I'm just taking all the credit here. Thank you very much.
Thank you, Dr. Rajesh Chaturvedi. Can have some quick questions, please.
I'll be very quick. Dr. Chaturvedi, thank you so much. I've known you for quite some time. You're a brilliant geographer. But what is a little alarming to me as a student of biology is your proposition that we should now look at ecosystems for conservation assessment. That will defeat the idea that every individual species is a unique genomic microcosm and needs evaluation at that level. So I'm not very comfortable with the idea that we should now, red listing is a conservation assessment tool that needs to be species specific. That's my submission. Thank you so much.
Sir, agree, sir. Can I, if I answer? So, red listing of species remains. At the same time, we also do red listing of ecosystems. Complementary. I'm not saying that that is to be scrapped. Yes, yes. It is already there. It can be scrapped.
Yeah, I have some two, three observations. Number one,
Request you to be
50 years
brief.
ago, this is a, it's not a perception, it's a fact. 50 years ago, when I joined my first service in Shillong, the people had not seen the fan in Shillong. Today, not only the fan, there are air conditioners in all the houses today. That is the situation. Climate has changed. That I agree. Your study mainly pertains to the climate change vis-a-vis the vegetation, impact on the vegetation that you have done, particularly Western Ghats and the other gentlemen studies in the Himalayan region. Yes, I have. See, it is, we all accept that it is no more a perception, but it is a fact. Climate change is impacting. The question now is not that one. How if there is a shift now, desert areas are getting more.
Yes, sir. Yes, sir. It's taxonomy. Yes. It's taxonomy. Yes. Fine. And third, that critical third, I want to inform you that the PSI will be arriving to blocks and like we are collaborating with them. Yes. That's it.
Thank you.
Yes.
Question for you, Rajiv. So, we talked I missed the first half because I missed it. But you're also building a can you measure, you know, can, can you like this? When you talk about climate change, you both of this, you know, we talk what you have to worry about that. Do you actually try to measure it? You can do it from that.
So we are doing that. In fact, we will be going to the plot again next year also. Every year we go. And since we know exactly the location of each tree, we have tagged. So we'll know who has died. So we have complete. So that's why we say census. So recruitment and mortality. Every individual will be recorded. Who has died? Who has been the new
The census,
having all that idea information about this.
the census is not a measurement. It doesn't tell you how much methane it is putting out. But it comment what it's rotting into.
Methane aspect I do not understand.
We can talk. We can talk. We can talk. We can talk. Quick comment. I want I think I want to respond to Raja. There is a revolution going on in terms of satellite-based technology for making studies of the various components of the Earth system. Revolution. Recently, one of our former colleagues and director of the State has opened a private company to develop drone-based synthetic aperture radar. Once that is done, all the questions about farmer senior scientists will be added. You can pinpoint the species in the forest. So we have to wait for some time. And not only that, when our own satellite having the synthetic aperture radar NISAR, it is developed in collaboration with the NASA and ISRO. Basically, marvelous, world class. There is no comparison. So in future, when we have the satellite working in P band, L band, C band, and S band, there will be huge amount of data. And like that, it was thinkable years back. So very bright future and complement being in space. He is trying to stay station and monitor very difficult task. I know that I have been go to the Antarctica to pass. I know difficulty, but I consider young boy.
So thank you very much, Dr. Rajiv, for the nice presentation. In the interest of time, we can move on. Our next speaker is M.A. Atmanan, visiting professor at IIT Madras, and several years of experience in the field of technology. He was formerly director of the National Institute of Ocean Technology, Chennai, the recipient of prestigious awards, the Ministry of Earth Sciences National Award, Excellence in Ocean Technology, and International Society for Ocean and Polar Engineers Award. On behalf of the Academy, I welcome Dr. Atmanan to give his talk on Indian Ocean observations.
IAS.
Develop technologies. So right now, as I am at class, let me thank all my colleagues and which I'm going to say is effort of so many hundreds of technologies and I just want to just tell you in minutes. Some of the platforms in the ocean, voice, computers, control. So I don't have to explain songs of days that it's easy, 1.5 degrees centigrade. All these things are about the light. But just we see that golden is the sink of the carbon dioxide, and even it dominates in the Earth's carbon cycle, and the productivity also takes place in the surface of the ocean. So really, ocean is the important thing. What I find is that unfortunately we know more about the Mars and the moon rather than the ocean, which is quite vast. We know unfortunate. It was really unfortunate because okay, it's not that it's not but first about the ocean, which is really giving us life. So you see that there is where are we? I mean, the atmosphere takes only 0.3%, but the ocean is taking such a large value of 94%. So the analysis has been done, and you can see the data here. And it will be we are the ocean, sorry, the earth is suffering. Like when we have the issue with the human body, the doctor it says test analysis, etc. We are aware of the concepts of there. But what you know about it? But the see, I'm about what is exactly. The point is that what exactly is happening. What exactly is happening? Doctor observations as well as the We have many, but still it is not adequate, as we can see. So what we need various types of observation. One is mood, which is stationary in the ocean, and measure parameters, both subsurface as well as surface. But there are other ones which can move, like for example, they are called Lagrange intervals, which move along with the current and the waves, etc., and measure the parameters. So I will just not quickly go through one of them, but 20 minutes is quite difficult for me to every. I try to see what is best possible. Now, if you see the Indian context, perhaps all of you realize that the weather prediction has improved from what it was about some 20 years back. One of the important factors in this is that we have got a lot of observations right now, which is getting into the models of IMD and decoys and other schemes where the prediction has improved. Cyclone prediction, you see that quite accurate in space and time is possible, and the life which is not is quite reading. That basically because of and what are the observations is they could have lot of bits of instruments in that which really measure the parameters, which are basically like where what wave, data, gas, all these things are measured on real time. Now, data points are basically something like this. You can see that they are all located at various locations in the Indian waters. It is all for Arabian Sea and the Bay of Bengal. It is something like this. They are floating in the water, and you see that. Okay, I will go here. You can see these boys are somewhere like this, and it's floating, and you can satellite. It comes to the Earth station, it comes to IoT, and you can see that these are cyclone paths, which has been traced out by the various data points in the ocean, and the ocean observation system was established in 97 at IoT, and from there onwards we get a lot of data online, and you can see that there are about observatories globally, but we have much more. I mean, we have got all this data available in this format for countries, so the same, because it's a shared data, it's a common mankind. So this is how the system will be put together. A tall one, which is directly moved on the seabed, or a slack one, which is a earthen catenary, it's called, it's a polypropylene rope. Signal loss. This really have various applications. For example, the top one has got a problem that it may just move along. In case of a cyclone, it's got to move with walking happens. But instead of that, we do it in India with a stack mooring. You can see that it's something like this. You can see the various sensors which are there are put on the mooring line. You can see that's conductivity, temperature, CPD, all are from here. And there is something called a induction mooring. Data is transmitted through induction into this moorings. It goes to the server. There is a data system that is transmitted to the Earth station at the IoT, from where it is disseminated to these guys who are the custodian of all the ocean data. And of course, this is how the surface of the we also have it not only in the deep ocean, but we also have in the coastal region. Coastal region mooring is quite challenging because you have got a lot of corals, etc. This is a picture of the one which is in Andamans, set up in Andamans. Now, if you see, this is how the cyclone, one of the cyclones within, I think, has been tracked, and you can see that the wind speed and air pressure, the wind speed has suddenly measured, and there's also a system where we have higher data acquisition that's happening exactly when the cyclone passes over the point. So all these things have helped in predicting the cyclone track accurately. Now, there are issues. When you have all these things in the ocean, you know that either human or natural, because of which these are being damaged. So this may be the fishermen, they come and conduct, they come and conduct these things which are in the deep sea. And in case of maybe the net will get attached, they get lot of issues and they damage the walls. So this has been going on. Let me also tell you that one of the hotspots of that is Goa. So perhaps some of you, when you are landing in an aircraft, you can see that there is an island there. You can see this picture also there. So this is the region where always there is consistently the same location this happens. So we have again done some indigenized or ingenious method wherein you have put the cameras there and we call it as others, where we just monitor it because it's close by, so we can even do the data transfer to our normal 3G or 5G. So this is set up in this location to identify some of these. Now we also have, you know, that tsunami is another important issue which we have to work on. So what basically happens, we all know that technology, etc. is well known. So what is done is pressure is measured, the change in pressure that's happening at that particular incident is measured using the potentiometer recorder, and that is acoustically transmitted to the center reservoir, and it goes to an IoT and voice where them say that whether it is cause a tsunami or not. And this is has to take place at a depth of say some 4,000 meters, that is 100 bar pressure. We have a variation. So the only sensor that's possible to take and you can find out the change in the frequency and measure the particular variation. So this is also compensated by temperature. So this is the one which is actually put by all countries, including Japan, which is really sitting on a display. So this is set up at various locations as I showed you. So the data will come to, of course, it twice, and they give the warning. Perhaps if you put the website of the choice, you can find out that any moment in time whether a tsunami will occur or not is given by them. Now, this is a particular situation which occurred some time back in 2013, Pakistan, and you can see and depth, etc. And picking up the the pressure measurement, pressure variation. So you can see that when the such a pressure variation occurs, the data acquisition is done at a faster rate, and we can see that this is the data buoy, sorry, the tsunami buoy, which picked up the data, and these are the some of the data that is being this is sure. Now, there is another aspect that we know that there or not in Africa and India. So to study this, we simply are Goa here and developed a buoy which is deployed in a particular location. Consider on your is, and there we have got the moved observatory with many of the sensors there, and you can see that this is how it is. Why is completely underwater because you cannot transmit online. Most of the time it's covered with ice. So the transmitter there will get damaged. So there's completely underwater. So the only problem is that and here not as go there. That is the issue there. But that's being done by the people now. Now there are so many other platforms. There is something called Data Boy Cooperation Panel, which all the data is transmitted all the countries as I told. So these are the these actually taken from the various countries which are the data points available, points, points, etc. Shows the latest one as last month. And if you see that lot of data is there from various devices. As you can see that I will just go through these systems just quickly so that they get an idea about that. One is Argo Pro. Argo Pro is run by the Day2 satellite, but again, unfortunately, ISRO has still not given a satellite for these purposes. So we have to spend on satellite. And you see that it has got a variable modulus engine. Just a simple arm distance is up and down. There is a bellow here which oil is filled in and taken out. It goes up and down and the data is collected and it is transmitted when it comes to the surface to the Argo satellite mediums. Now you see the Indian Argo program started quite long back. The idea is to have a 3x3 and to constitute the area, have one of them, one Argo Pro. So a lot of data is collected from the various locations in this. And you see that next one is a glider, which really does not require any power for its locomotion. It goes by the drift, the drag and the drift principle. And this is also developed, this is also being by India to collect data at various locations. And you see, this is a list of all of them. This is really quite large. I may not be able to go into detail about each of them. You can see the operators, etc. The drifter can just drift along with the current. Current meters connected to the buoy again to measure current. Then we have got CTD, that is connected to temperature depth by from the fittings. Then we have HF radar. I just explained that minute. Then of course all these things are quite at various locations available. I want to explain about little bit about this HF radar. Yeah, what is sitting here? HF radar data. Here we have transmitter and receiver. See here, locations are shown here in this map, and you get the data in a complete area, so to say. That is, see here. You can see that in an area about 200 kilometers, you get the current, not like a point one, which as a vector, get the so this is done in various locations, but still it is not complete India. Definitely, due to the prohibitive cost, we have still not done it in everywhere in India. Of course, we have the various other programs as part of the deep ocean mission, which is coming up. We have got deep Argo floats to go up to more than 2,000 meters. Then we have various campaigns there, various Arabian Sea field campaign, many campaigns with institutes as well as other Indian institutes. So this has been already explained in detail, so I'm not going to explain about this space-based ocean observations. So many are there. OceanSat, Megatropics, all these things are Indian satellites from which you can measure the wave characteristics, ocean color, all these things are possible. I will just like to conclude that it is necessary to expand this. Earth is really not adequately monitored enough, so we need to India has to follow suit, and any other coastal erosion, beach restoration, all these things need data on a large scale. So this is really essential. The predictions, it requires more data. The main last thing is more important because we need a large number of trained manpower employed for this. People are not fully aware of this. Everybody knows about space, but very, very little knowledge about the ocean is there, unfortunately. Thank you very much once again for giving.
That was very nice. I had a question about international waters. So, if India wants to put buoys outside its territorial waters, how does any country go around doing that? Or do we have to cooperate because sometimes waters do not belong to that particular country? How that situation?
If you want to put a boy outside the EEZ, that is normally called as common heritage of mankind. It doesn't belong to anybody but belongs to everybody. The only issue is that the data collected there should be shared in the GTS format as part of this DBCP, data point formulation panel. So data should be available for all countries. You should not say that I will put a boy and I only will use the data. That is not allowed. See, this is the they really have a restriction of that. Otherwise, international waters, it's fine. How empowering this Bhubaneswar is from here. So I think Bhubaneswar has been doing
Sent undergraduates. Absolutely, I agree.
Let me also just tell you on the other side, these angular manpower takers in India for this are less. They can take, like, I know when I was there, the government provides enough money, but not regular manpower. You take manpower on
Absolutely.
I completely echo his sentiments. The biggest challenge for all of you at the Indian Academy of Science is to do handholding with great urgency with the academia.
Can I make one comment, quick comment on this particular on this particular aspect? So involving university. You know, in Monsoon Mission, I am from IIT. So there is a recent resolution, right? It's categorically told that intramural fund will be given to university. Okay, it also will be given from IIT. Computer also will be given to IIT, and universities will be given this graduate students and faculty to do analysis for their respective region. So one university from Assam, Tripura, and everywhere, and faculties are joined couple of universities, and data will be shared. Computer also will be shared, HPC, and intramural funding, JRF salary will be shared, and to take them on board.
I just get with one thing. The only point is get with JRF, SRF, the project.
You have
Thank you. Thank you. The scientific.
Correct.
What happens is okay, so boys what happens not like they say really when they are doing it, this boy gets entangled somehow. And you know the cost relax, so it gets damaged. So when it gets annoyed with that, this is what happens as a campus.
Thank you, Doctor.
Of this session. Final speaker of the session.
Important indicator.
Deep sea survey and mapping. He's a marine geophysicist with a PhD in geophysics from the Arctic University of Norway. And he has over 13 years of experience in collecting and analyzing marine geophysical data. His research focuses on geohazards in the exclusive economic zones in India. And he also is interested in hydrothermal systems in Europe. Today he'll be talking to us about the hidden beaches and the potential hazards economic zone. Welcome.
Hello. Yeah. I hope I'm audible now. Thank you for the introduction and thank you very much to the Academy for inviting me. It's a great honor. I'm a marine geophysicist. What I basically do is help of sonars study how the seabed looks like. As the previous speaker, Rosa Adnan said, we don't know much about the ocean or how the ocean bottom looks like. So it's actually a excellent follow-up of talks about how we monitor the waters, ocean waters. Where now I'm deeper down to the seabed and show you what it looks like and what are the benefits out of it and what are the potential, some hazards, potential hazards. Can be hazard, but it's kind of probably in like a futuristic way. What could be any hazards like tsunamis or landslides or certain things like that. So first of all, to just introduce everybody to what is an exposure extent. So here you're looking at the tide line where you are at the beach, and from there, the low tide line, you take 200 nautical miles out into the sea, and that is the exclusive economic zone of any country. And within that borders, any country has its right to explore and exploit any resources within the water, that is fish, or any other like wind energy or wave energy. Or you can also go to the seabed and actually see if there are oil or gas, minerals. So any country has its own right to exploit that. So we classify this seabed in terminology. We don't usually say economics. We usually say like, okay, you have a continental shelf, that is the shallowest part out of any deep, and then the depth suddenly start to increase much more rapidly, which you call a continental slope. And then you have a continental rise, which is where it flattens out, and then you have the deep sea. So India has the 18th largest economic zone in the world. You can see here in the map. That is one. So here you can see this is basically 200 nautical miles out of the coast of India. But where it shares, when there are other countries nearby, then usually you kind of agree to a central line, almost nearly equidistant between each country. Sometimes it can be controversial, but more or less they agree to a particular line. So you can see India shares its economic zone with many countries: Pakistan, Maldives, Sri Lanka, Myanmar, Thailand, Bangladesh. So it's quite large, nearly over 2 million square kilometers. Why do we need to map the EZ? Mapping the EZ here, I particularly mean how the EZ looks like, what the seabed looks like, what is there on the seabed. Because it's important to know if there are any potential resources and how to manage them. And also just navigate these waters. And if you want to construct something like, for example, platforms, oil platforms, and also if there are any geosas, if there are potential for earthquakes, or if there are potential for landslides, things like that. Applications to the common public involves the fishing operations. They can benefit from such mapping and mineral resources, oil and gas, and also laying of pipelines and communication cables, and also construction of near port or near port constructions. Now, scientifically, when we look at it, you have different focus. You can look at the seafloor morphology and how it looks like, what are they. And you can also understand that sedimentation is how the rivers have controlled the sediment output into the ocean over the past. You can study how the life looks like under the ocean. You can also look at how the Indian margin has developed over the years and how it's going to develop in the future. Also look at how climate has changed. You can sample the sediments on the seafloor. Then actually reconstruct how the climate looked like in the past and how it will, based on that, you can try to predict how it will look like in the future. And then another key aspect is a geohazard, where these how the processes occurring in the ocean can have an impact on the human, for humans and also human-made objects. So with these objectives in mind, the Ministry of Earth Science Government of India launched this program known as the Geoscientific Studies of India. So its main objective is to create a seabed topographic map of the economic zone of India, and also put them together in a database which can be accessed, which can be accessible to everybody. And additional scientific project is involved evaluating the geothermal potential and also to understand the sedimentation and paleoclimate in the of the Indian Peninsula. So this is a long-running program, it's still ongoing, it's not complete, and it's started way back in 2003-4. That is when it was initiated. So it's been running for nearly 20 years, and it's an ongoing program. And NCPO, the National Center for Polar and Ocean Research, is the nodal agency for mapping the Indian Peninsula, along with these three other institutes: National Institute of Oceanography in Goa, and the National Institute of Ocean Technology in Chennai, and Geological Survey of India in So we are conducting these surveys in two phases, where the phase one survey takes a deep water surveys, where it's below or deeper than 500 meter water depth. That is NCPR is handling. And phase two, where the shallow water surveys, that is near coast areas below 500 meter, has taken care of mostly by NIOT and NNA. How do we go about mapping this research? So you essentially need a platform, which is usually a research vessel. So we use multiple research vessels. These are Indian research vessels. So we Sagar Kenya and Sagar Navy. These are operated by the Ministry of Earth Sciences. And sometimes it's very hard to get time on this, dedicated time on this, because these are always so busy with a lot of different objectives and a lot of different institutions vying for the time. So we sometimes have to chart foreign vessels, such as these vessels, Boris Petrov and MGS Sagar. So we usually have to take these out into sea for months on end and survey the waters. And what we say by mapping or surveying, here it's mainly acoustic surveys, basically a multi-beam echo sounder. That is a sonar, basically. It's very simple. It's attached at the hull of the ship, which sends the acoustic pulse, and then it measures how long it takes to come back. And then if you know the velocity of sound in water, which is somewhere around 1470 to 1500 meters per second, then you can calculate how deep that location is. So you do this, that's sometimes you call it a single beam, you send one signal. But usually how we do this is in multi-beam echo, you have multiple beams. Usually you have 800, 900 beams going perpendicularly from the direction of the ship motion. And then it's basically sending back information over a large area of the seabed. Usually it's 3 to 4 times the. So, and then we basically create maps like this kind of topographic maps, where here the red is shallower and the blue is deeper. And usually this frequency range is between 10 to 500 kilohertz. That's the acoustic frequency. We also do other types of acoustic surveys known as side scan sonar, where we have a fish, which is basically an instrument we tow much closer to the seafloor at a higher frequency range. It kind of gives you some information of what type of sediment it is based on the reflectivity of the reflection strength of the signals. We also sometimes use a subatom profiler, which is much lower frequency range, which also gives you some information about a few hundred meters below the sediment, how it looks like, what is there. And we also collect some samples at certain locations. It's not something we conduct all over, but at certain locations, and if the facilities are available, we also collect sediment samples using gravity cores, and also biological samples from the water column, like plankton, using a bomber. So what is the status of this program? As of now, over 90% of the deep water area, that is below 500 meter, has been covered. So the green area that's been covered by NCPR, you can see the western margin, the deep water area is more or less covered. Where we have still some gaps, these red areas, those are the big gaps, and also, sorry, not the red areas, the black areas you see here, for example, of Paradeep here, and also in the Andaman coast, those are the big gaps, and also we have a bigger gap on the southern part of India. But I will focus here, I'll show you some glimpse of some of the results from these studies. And also, only I am focusing here now on the west coast, where we have a more complete understanding of how the seabed looks like. So it's not that we not completely know about we don't know completely about the seabed. This is a publicly available map from the General Barometric Chart of the Ocean, JEPCO, this was released in 2020. So here you can see there's a western coast. You see the cities Mumbai and Cochin here, and then you see the coastline here. You have the Cornell Shelf here, and then it suddenly gets deeper. So this is a shallower part, and then it gets deeper and deeper. The blue here represents nearly 4,000 meters. where the shallower part is usually is less than 500 meters. So this is the publicly available data. So resolution is the 500 meters. And I will now overlay on what we have collected, and then you can see the difference. How different it is, like putting on a glass. We can see particularly in these areas, you look at this area, there was not much information, we don't know what it was. And then suddenly you have a very nice submarine channel complex. You see a lot of new mounds and features popping up all over in this area. So, example, some of the features what we found. There's a bathymetric heights. These are basically underwater mountains or hills. They don't breach the surface water, so these are not islands. They are below the seabed. And I will come into more detail in the later slides on those. And then you have submarine landslides, which are basically like landslides, which are basically similar to what landslides are on land, but these are on much larger scales. And you have submarine canyons, which are basically these very narrow, steep-sided valleys. And these are basically important for the supply of sediments into the deep sea and also very important biodiversity hotspots. And you also have submarine channels, where these are basically underwater rivers, you can say. And these are very important in terms of oil and gas reserves and sediment supply into the deep sea. So here I will focus on two particular features, that is bathymetric highs and then the landslides, where I will try to share a bit more information. So why bathymetric highs are important? Because these are basically like underwater mountains, and in water it affects ocean circulation, as it affects air circulation, wind circulation on land. In the ocean, it affects circulation, and also it creates these local upwelling zones where the water is coming up and creates a very nutrient-rich water, where you have very high productivity, and actually you have a lot of animals there. So this is a very interesting area for potential fishing, and also very interesting habitats, some unique habitats as well. And also it's very crucial for safe navigation. This is the image of a submarine USS San Francisco. It hit in 2005 on an uncharted bathymetric high. This is actually a mountain, and it damaged like this. So it's very important to know what is on the seabed before you go with the submarines or anything, and then to navigate these waters. And these are also potential mineral organisms. This is an example of cobalt-rich minerals which are deposited on seamounts. So there are multiple ways in which these are important. So in the western potential margin, we identified about 33 distinct bathymetric heights, which were not identified before based on this data. And these bathymetric heights are classified in different ways. Some are called the seamounts. Seamounts are usually, it is 1,000 meters above the seabed. So, but it shouldn't reach the water. So it's if it's taller than 1,000 meters, then it's called a seamount. And if it has a flat top, then you call it a guyot. If it is less than 1,000 meters, you call it a hill or a knoll. So it's kind of like how we say something hill and you have a mountain. And then if it is not a seamount, so it's below 1,000 meters and it has a plateau, you call it a plateau. It's the classification by the International Hydrographic Organization. So particularly, I would like to focus on how these bathymetric heights are formed. Because the undersea volcanism is the main driver. Because you can have a mantle plume sitting, and then because of the plate motion, plate tectonics, the plates are always moving. So you have a volcano forming in one area, and then after some time it moves. And this is process happening over millions of years. And then you have these extinct volcanic islands going, and erosion and all the other activities, and it becomes sea. So we believe that the seamounts in the western margin is basically derived from the Bayunian and the Marian volcanism. So the Indian plate movement over those hotspots that created these seamounts. An interesting example of such seamount is the Sagar-Kenya bathymetric complex. This we are known for some time about this particular feature, where you see three distinct bathymetric highs here, and it kind of resembles volcanic caldera, kind of collapsed. So I believe it was probably a past volcano, which is in this shape now. Maybe it was much bigger in the past. The next feature I want to focus is submarine landslides. So basically downslope movement of sediments in response to the gravitational force. So it usually occurs between the continental shelf and continental slope, where your slope is slightly higher. And then you have loose sediments, sometimes very water rich, and you have a lot of sediment being supplied from the land. Then it can create these sort of slides. And the major difference from the on-land landslides is that usually this runoff distance is very high. It's hundreds of kilometers in usual case. And the landslide scars are much bigger compared to what you see on land. These are usually caused by earthquakes and also loading of slopes, that is heavy sedimentation. You have suddenly a lot of sediments coming in, and sometimes weak layers where they are very water rich, and sometimes also gas added. Dissociation gas addeds are materials which are basically ice-like materials which composed of water and methane. When it dissociates, it releases the methane, and a lot of water is released, which kind of destabilizes the sediment. And also volcanic activities are known by such landslides. This is a particularly large submarine landslide we identified off Cochin area. It's 46 kilometer wide, this slide scar, and the total length of this is about 100 and nearly 140 kilometers. That's basically distance between Mumbai and Pune, for example. And it covers a very large area. This slide deposit occupies nearly 5,700 square kilometers. And this we think is likely caused by high sedimentation rate in this area. Another interesting one is of Kilon. It's also on the very southern part of the here, if you can see the location in this map here. And here is very interesting because you see some slide deposit here if you look here, but you also see these cracks, these fault cracks. Here there's a profile across it which is about a 55 meter deep. These cracks, you can see a subbottom profile across it, and you see some planes where it's sliding off. So we think that it's about 60 kilometer long, this crack. We think that this could be a precursor to future landslides. So we have actually provided this information, for example, in buoys, where they can actually potentially deploy their tsunami warning buoys, for example, to if something happens. We don't know this data has been collected already nearly 10 years back. So there is no continuous monitoring because we don't have time and money to go to the same place over and over again. So we don't know how this is developing, but it's an interesting area to focus on. So to summarize, basically it's a very essential, although we have been, as I saw, it's been going on for a quite long time, over 70 expeditions have been conducted, but still there is quite a lot of area to cover, and many areas need to be resurveyed for to know what is happening, to monitor these geohazard locations. I would like to thank the Deep Sea Survey and Mapping Group, where I'm from, and also particularly Vijesh and Sushant, who has been working on these data mostly. Thank you.
Curious to know which among the bathymetric files contribute most significantly to bentopelagic coupling in terms of what drives the energy budgets in the deep sea. I understand that the hydrothermal belts will exceptionally rich bilevels.
Yeah.
How do you explain that in absence of sunlight?
In the hydrothermal event case, yeah, that is the basis of an ecosystem. There is not photosynthesis, chemosynthesis. So then, so you have sulfur-reducing bacteria and all that, and then you have higher level organisms based on that. That is hydrothermal event. Here you don't have such an energy source. This is basically all the biodiversity is due to the upwelling of this nutrient-rich water coming up into the closer to the. Which among means yeah. So usually it is not which type of this is actually contributing. It's mostly how the structure of it, like how its slopes are, and also how much surface area is there, and how close it is to the surface. So it can be in either one of these types, but it depends on how close they are to the where you have more photosynthetic activities.
Is there any international treaty is also? solution. So, scientific studies, it is allowed go and study everywhere, or is there exclusive zone?
You cannot go to another country, see EZ and collect data without their permission. So that you always need to. And in case of fishermen, because it's a very common happening in, for example, between Sri Lanka and India, a lot of fishermen going back and forth. And this is a common problem. There is a Coast Guard always monitoring, putting them back. And as far as I know, I probably don't know whether there are such treaties available between the countries, but this is a very common problem. And these Coast Guard usually catch these people and then send them back. It's because overfishing and yeah, the overfishing is a big issue. He's NCPO's first founding director. So
Let's cover both the Arabian Sea and India. After the collaborations of the National Institute of Oceanography, National Geophysical Research, Hyderabad, under my leadership, let me thank yes, it was me who took up this challenge of mapping the entire Indian Ocean region for submitting to the United Nations under a mega flagship program of the Department of Ocean Research. It was 2 crore, more than 100 crore. But it was done meticulously, and that data is the backbone of all the deep ocean missions, all the explorations and everything. So now what is why I want to make up about what the Mundati, I will not have somebody else to get that. But that is that. Now you have to share all this information with the artificial data, like since the person has been asking about the mountains and hills and the, they all can be tagged with satellite data. Can be good the information of the system. So even the United States and USA, they are putting upon our Department of Topography. And we are talking about the very big program. I had people answer 20 years from now, what people are thinking. So now the quality of the architecture side, millimeter and variations you can from the space right now. Technology is changing, so we have to use those data to get the complete Indian ocean topography, and then validate this thing with the data which is the rich resource, which is NIO and NIO. So that is the comment.
Thank you.
Fascinating. With regard to the slides, the potential subterranean oceanic, is there has to be a way to connect them with what's happening on the land. Because if you've got, let's say, in Kerala and other places, you've flagged potential huge slides. And we know there's a lot of deforestation and and if you have changes in the monsoon patterns, increased rainfall, for example, seasonal rain, you will get a lot of flooding and a lot of ocean. So are these data being integrated, the oceanic data, to the land data?
There has been, for example, these particular areas where we think these landslides are mostly caused by high sedimentation. But in the past, so currently we don't have a monitoring system to see what is ongoing. So if there is a high rainfall season, how much is deposited coming into the ocean, we don't have such monitoring capability. What we know is, okay, we see these past examples of slides, and we know that high sedimentation rate is the reason. So we can actually use these collect these sediment samples and actually predict how monsoon changed, whether it was stronger during certain period or less during certain periods. And these periods are not yearly; these are centuries or even thousands of years periods. Then you can actually say something about, okay, whether this can contribute to some landslides or hazards. So, but currently, as of now, we don't have such monitoring system in place. And these sedimentation rates, what I'm talking about, is not very high. It's not like dumping a huge load. It's like a millimeters. The thickness increases in millimeters per 1,000 years. So sometimes even if you say like a 20 millimeter per 100 year, then that's very huge. So it's a very long, yeah, it's very long-term process. And the monitoring such systems, we only recently developed such monitoring systems, so we are probably wait another 6 months to see some.
I just want to comment, a short comment on this question first. So there are separate programs which is called the DSTP, Deep Scheduling Programs. India also has a separate program called IODP. Now, to answer your question and these queries, what we need is the archive where such informations are collected and stored. What we do is that the sediments have the best archive for collecting and retrieving such information. We go down, in the deep sea ocean, we collect these ocean cores, and then we analyze them for different parameters, including sedimentology, chemistry, biology, and everything else. Them together to ultimately decipher what has happened in the geological past. We can go back to several hundreds and thousands and millions of years. We can also go back to shorter time scales of hundreds of years or a few thousands of years as well. And CPR has been coordinating a very major program called IODP. I've been part of that, and we have been supporting expeditions around India, and also we have been participating in other missions in different parts of the ocean to collect such data and analyze that. So that's the information.
Thank you, Rajiv, for the comment. I think we don't have any more questions. We had a very interesting session which covered both ops. So we learned a lot about how remote sensing can be used to map our forest ecosystem, and how the Indian Ocean can be surveyed. And we had a lot of interesting technological tools that were available, which one would hope would lead to the development of early warning systems, especially in the context of tsunamis. And more recently, and we also learned about how to monitor the exclusive economic zone of India, which has enormous commercial interest apart from its scientific value. All the 4 speakers for their very interesting talks. And I also thank the Academy for giving me the opportunity to chair this session. Thank you very much.
Let's thank Professor Vinod Nivasan for sharing this very interesting session. And now we break for lunch. And lunch is arranged behind the auditorium, the same
how the civilization expanded, matured, early, mature, and late civilizations, and so on. We have a reasonably good time scale, time constraints in terms of the evolution of the civilization based on a very sound and a very solid archaeological record. Now, this information was a little bit complex, became a little bit complex when becomes a little bit complex because we called this civilization, or we call all civilization basically, as a river-based civilization, riverine civilization. And when we look at the modern landscape of the Northwest India, and particularly where most of the civilization sites are located, there are no large rivers today, right? You have Satluj Valley here, and then you have Yamuna Valley here, and this entire region between the Satluj and the Yamuna, where most of the Harappan sites are located, I'll show you a map in a minute, there are no large rivers today, right? So therefore, when you see, we will of course talk about all of these windows which I have shown here. So when you look at the distribution of the Harappan civilization sites, particularly the mature sites, which is 4.6 thousand years to 3.9 kilo years, which are highly concentrated in that region where no present-day system actually, goes through. This is actually an interflow region between the large Satluj Valley and the Yamuna Valley, the modern-day Yamuna Valley. The obvious question arises: why are these numerous settlements scattered around these lines, around these areas? And that led, and of course the hypothesis and the thinking started to grow when we discovered this very famous paleo channel, which is the so-called Saraswati River, which is flowing through that same region, which is presently the desert, and presently there are no modern day systems here. This was discovered because we had access to some really good thermal infrared images. You don't see them in the normal satellite images anyway. So then the hypothesis started to grow that yes, there is a river culture hypothesis, and the debate started. And the hypothesis was immediately made that most of these urban settlements, because they are clustered around the paleo channels, and they must have been related to rivers. And the so-called mythical Saraswati River, which has been referred in the Vedic text, that was linked to that, and that it was suggested that the civilization disappeared because the river disappeared. So very obvious and a very logical conclusion to do. There's nothing wrong about that. But the problem with this hypothesis was that, first, that yes, we discovered a paleo channel, very broad thermal or infrared image and all of that, but we had no information about the subsurface existence of that paleo channel, how big the channel was, how thick the deposits were, absolutely no information on that. We had no information about when and how the river disappeared, no timing constant, although there was very strong timing constraints on the archaeological data set, but no information about the migration or the disappearance of the river. And of course, no information on the causal factors of such large-scale shifting or disappearance of the river as well. The consequence of tectonics, climate, nothing was known until about a decade ago. But we kept on arguing about the river culture hypothesis, etc., which has been true in many other parts of the world as well. So we started this work about a decade ago. We used multiple tools, resistivity surveys. So our job was first to detect the subsurface continuity of the paleo channel. Our job was to detect or map the channel in the subsurface. Our job was to date how old these channels are and how and when they were deactivated. And we use, of course, multiple techniques of resistivity, drilling, coring, OSL technology, and then of course, and also of course to find out where these sediments came from to form these large sand bodies. So this is the processed image. When you see these paleo channels, these are some very important archaeological sites which are located very close to this river, very large urban settlements like Kalibangan, Rakhigarhi, and Banavali, and all of that. And then you can also see a slight trace of a paleo channel here, which connects to the modern-day Satluj. Okay, so all of this was, this also was a very specially processed image because you don't see anything on the modern day image today because all of these are very highly agricultural areas. So you don't see any trace of these channels today on the modern satellite images. So we had to process these images a little bit. And then, of course, you have the modern Jamuna somewhere here. So this is how we went about. To answer the first question, we ran a series of transects for the resistivity survey. It's a very blunt technique. It doesn't give you a lot of information, but it has the power to distinguish the sand and mud, and of course the hard rock very nicely. So we could very nicely map that, yes, there is a subsurface extension of these channels, and these channels are about 20 to 30 meters deep, where deep, 20 to 30 meters deep, as inferred from the thickness of these sand bodies. So we ran transects here, we ran transects here, so that transect, this transect, corresponds to this, and you can see again, almost 20 to 25 meters thick channel bodies, and here again, you see the thick sand bodies up here. So we actually, what idea was, of course, we also had several other transects, which I'm not showing, but we could very easily establish that, yes, there is a very nice surface extension of these channels. So therefore, and these thick bodies of sand cannot be deposited by a small, there is, by the way, along this channel, what we run today is a small seasonal Ghaggar river, which actually originates somewhere here and then drains for like 15 days a year, for example. So obviously, it's sitting in the same old valley, but this kind of a river is not capable of, of generating such large 25 to 30 meters thick, deposits. So therefore, the first answer was done, that yes, there is a large, large river, signature for large river, which exists along these, inferred paleo channels. Then we went about drilling several holes. We drilled about more than 11 holes, 11 holes here, and then also some other holes in the Jamuna Valley later on. And we were trying to map the characteristics of these sand bodies. We also tried to date the individual sand bodies in terms of the OSL dates. We also tried to do the compositional analysis. We also did the provenance analysis. There's a series of analyses which actually went on these cores. These were one of the first cores, first detailed cores which we obtained from this kind of a region. Then what we got was very startling. This is a transect, somewhere here, very close to a place called Kalibangan. And what we saw was a very nice amalgamated, multi-storied sand bodies, which were basically lying on a base of what we called as, what are the Aeolian sands. All of these were sand, but these were very distinct. The Aeolian sands are very distinct, as most of the sedimentologists would know. So they were fluvial sands, multiple stories of sand, which basically indicates that there were multiple channel activities which were recorded in this entire transect. And you can see the dates were also done. So there are shifting of the channel belts from here to here, here to here, and so on. What you should also mark, that there is a very nice demarcation of where these sand bodies are ending. The arrows are marking there. And note the dates are ranging from about 25,000 years to something like 23 and 13,000 years and so on. These are the markers which basically tell you when this large river activity ended. And please remember the dates, 25 to 12, 13,000 years back. And then there are two sets of deposits. We can easily see there is a older deposit and there's a younger deposit here as well, and so on. Now, what this tells us that any large activity of a river ended, around 12,000 years or so, or close to that, basically. Of course, there were minor sand bodies, which you can see here, and also in other locations, which continued up to about 8,000 years or so 7 to 8,000 years, which means that there was no activity of any major river, after about 12, 10, 8,000 years or so, okay? And then of course we had another transect somewhere here, along the Satluj, the paleo channel of the Satluj. We got similar information from there as well. We again had the last fluvial activity reported at about 10, 11, and 8,000 years as well, which means that again, in the entire region of the Paleo Satluj Valley or Paleo Ghaggar Valley, whatever you want to call that, the large river activity ended by about 8,000 years. The Harappan civilization continued up to about 4,000 years.
okay, which means there is a gap of at least 3 to 4,000 years between the demise of a large river and the demise of the civilization. Therefore, they cannot be contemporaneous. There is no way they can be contemporaneous. That's one. Then, so that's what I just said. Then we also looked at what were the reasons for their disappearance. We looked at detailed depositional history, and we could relate to several depositional events in a time scale of something like 80 to 90,000 years until about 4,000 years, and we recorded several activities of fluvial aggradational activities, and which were strongly linked to the climate change which happened over the time period, over this kind of a time period. So that was one. After that, we again had another problem to solve, and that was the Jamuna link. How much was the contribution the Jamuna was made? Because again, the hypothesis has been that this branch of the, or rather this branch of the Satluj and this branch of the Jamuna, they both actually made this so-called large Saraswati river, which was supposed to be at least 4 times larger than the modern day Ganga system, for example. So therefore, this connection was also equally important to examine as to what was the role this was playing. So we actually had a very similar strategy. We again did a very detailed transects of the resistivity surveys. The paleochannels were existing, but the subsurface existence was not known, and we could establish again that they were 15 to 20 meters thick sand bodies which are located, at several points. And there are many paleochannels, around there. So we took up a transect here. All these black lines which you see are actually paleochannel traces which people had been mapping before. And along each one of them, we could discover that there are paleochannel thick sand bodies which are located there. And then these are the cores which we obtained. We analyzed them for centimeter by centimeter, looked at detailed lithologies, facies, and interpreted them in terms of stratigraphic development and all of that. And then we again had the dating of the cores done. We generated almost 45 OSL dates, and we could discover that again, this part of the valley, the Jamuna Valley, also went through very similar stages of aggradation, but at different times. There were 4 different phases which we discovered. The only other interesting point which we discovered that this valley was abandoned almost 18,000 years ago, much, much before the Satluj was abandoned. And therefore, this also may not have contributed significantly or at all, in fact, to the demise of the Harappan civilization again. And then if you start looking at how this whole drainage systems actually developed, this is the model which we developed. And so there was basically 2 series of avulsions. One was one along that, and then which happened around 18,000 years ago, and then this happened around 8,000 years ago. And this is the reconstruction which we did for the last 80, 90,000 years, and different phases. So it all happened. This all was reconstructed through the sedimentary record which we had, the dating data we had, and so on. So what is very well established now that the Jamuna migrated 18,000 years ago, the Satluj migrated 8,000 years ago, and the Harappan civilization continued until 3.9 or about 4,000 years ago. And therefore, they were certainly not, the demise of the civilization was certainly not contemporaneous to the large rivers. And of course, the modern day landscape which you see today has always been reworked by the smaller rivers, et cetera, and so on. Now we had one more problem to solve that, okay, we have large sand bodies sitting there. There is a subsurface existence of that, but where did the sediments come from? Are they actually coming from the foothills, or are they coming from the large, catchments of the Himalaya? Any large river which was draining in that region must be originating in the large part of the Himalaya. And we have a wonderful technique, in geology to sort of a fingerprint the sediments. We use what we call as a uranium lead dating of the zircons, which we extract from the sediments. And interestingly, each part of the Himalaya is actually known by a very well-established range of the UPV dates, for example. So the higher Himalaya, for example, are 500 to 1,000 million years. These are Trans-Himalayan and Karakoram in the 50 million years and 1,800 million years and so on. So this has been established by several workers, and of course the idea of this work was very different. They wanted to understand the exhumation history of the Himalaya, et cetera, and so on, but we used that for testing the provenance. And then we analyzed several samples from our cores, all the cores which we had, some samples from all the cores,
And what we found, very interestingly, that almost all the cores are centered around 500 million years, which is actually very similar to what the Satluj actually shows, okay? Very, very close to the Satluj age, which means that most of the sediment were derived from a Paleo-Satluj river, which was draining a large part of the Himalaya, was draining in a different direction at that time, and then it was obviously avulsed. Then we also use another technique which is called argon-argon ages, which is basically the cooling ages of the mica. Again, let's not get into the details of that. But again, this is also used as a source, and again, it suggested that almost all the core sediments have an age which is very similar to the modern day Jamuna, okay? So therefore, modern day Jamuna and Satluj as well. So there are overlaps of Satluj and Jamuna dates here, and these are the two large rivers which might have contributed to that. This is basically what I just showed you from a geomorphic reconstruction using sediment cores. So the sedimentological data, the geochronological data, and this compositional data, they all support this, this kind of a hypothesis here. And then there is a modern analog for that. If all of you remember the 18th August event of Kosi, where one river shifted by more than 100 kilometers in just one shot. So such events, such mega migration of rivers are known, even today they are happening. Of course, the reasons may be different. There are anthropogenic factors also coming into picture, but such large scale events have happened, in the history as well, historical scale as well as in the geological time scale. So what we therefore believe that the settlements around the Indus or the Ghaggar and Satluj and the Jamuna rivers, they actually developed when these valleys were abandoned and the floodplains were stabilized, and then people started to settle in there rather than staying in a river which is 4 times bigger than the Ganga flooding every time and so on and so forth, right? So therefore, when these rivers reduced their mobility, they got confined in the incised valleys, then was the time for the civilization to actually mature, for example, in that region. So therefore, our conclusions are that yes, there is a subsurface extension of these paleochannels which we see on the satellite images, but then the timing of these rivers for which they flowed in that region is much earlier than the demise of the civilization, and therefore it is highly, highly improbable that the Indus settlements actually flourish because of the perennial source of the rivers and so on. What we believe that the confinement of these rivers and the stabilization of the floodplains, plus the development of agriculture, water resource, the storage structures, all of that actually played a very important role, to stabilize these settlements. So what we believe that it is actually the departure of the rivers rather than the arrival which triggered the growth of the urbanism and not the other way around. And I would like to sincerely acknowledge the help of all of these people. This entire work is actually an outcome of 2 PhD students, Ajit Singh and Imran Khan, and then of course several collaborators across the world and funding from many sources, including Ministry of Earth Sciences, SCRB, and also Royal Society and UKRI. Thank you so much for your attention. I'll be more than happy to take a few questions.
Thank you very much. Very interesting. And obviously there would be many questions. First.
Question, if the rivers receded or died 8,000 years ago and the Harappan civilization lived from between, let's say, 6,000 to 4,000 years, where did they get their water from?
Well, see, what I'm trying to argue is that there was no large river, but I'm not saying that there were no rivers at all. There were small rivers there, and the population was not very large at that time, so that was enough to sustain them. That's number one. Second, remember that by that time, the agriculture was very well developed. The Kalibangan is the oldest agricultural sites in the whole world, where we discovered agricultural signatures about 9 to 10,000 years ago, for example. And then, if you also look at the archaeological sites, they had very nice water storage structures. So they knew how to store and water as well there. So I think there was enough water around to sustain such kind of a small population of civilization, by the way. Then, after 3.5, 4,000 years ago, there are other hypotheses which are mostly true, that there were climatic transitions happened and all of that. So that, most of us know that.
I have a rather uninformed question.
Yes, please.
I have come across a suggestion put forward not by earth scientists, but by some archaeologists, and I can't testify for their authenticity, that actually there was up the stream of the Saraswati at some point in the past, some very strong earthquake, as a result of which what is called river piracy took place, and the water of Saraswati was largely diverted to some eastward flowing stream. And there has been attempt to relate this to the fact that the Yamuna, which meets the Ganga at Allahabad, Sangam, is also kind of mythically called as containing the river Saraswati as a result of this river piracy.
Okay,
Do you have any comment on that?
I got your question. See, I have not commented at all on how and why these rivers emerged. Okay, that's a very separate question to answer. There are strong models which are not very authentic as of now, but there are many possibilities: earthquakes, tectonic events, large floods, and all of that. So there are very separate questions. I think that the fundamental point I'm trying to answer here is that there was no river when the civilization was existing. That's number one. So, but then, the shifting of the rivers could have happened by multiple factors, and we are yet to understand that. That needs a very separate set of investigations to do that. Earthquake is just one possibility, but then there are no, documented records of that. These are again speculations, by different people, but there are possibilities. So that's a very separate question altogether. No, there are people who work on what is called as a paleo-earthquake analysis, but as of now, we have no data to suggest that there was an earthquake at that time which might have triggered. That's one. The second point I wanted to raise here that is again a misnomer that the civilization disappeared or died. Actually, it did not happen. The civilization do not die. In particular, this civilization did not die. They only migrated. So what happens that they migrated to the north, towards the Satluj exit and all of that, and they also migrated to the east, to the Ganga Valley, for example. So the civilization did not actually die. It was just because the resources became limited, the monsoon, declined, the precipitation became lower, the resources became, scarce, and they migrated to the east, for example. So these are, all sorts of misnomers which are there. So they migrated to the Ganga Valley and then they survived there again. Yeah. So this is what we are today is just a continuation of the same civilization in a different way altogether. Yeah, there are. Yes, please.
Yeah, I'm just wondering, the entire very interesting scenario of meandering of the rivers. How does this corroborate with the marine records? Because we have some, similar evidence coming from the strengthening of the rainfall and strengthening of the monsoon.
Actually, I showed you this data.
Yeah. So how we can try to corroborate this evidence from the marine data?
Yes, it's a good question. See, what I showed you here was, this correlation with the summer monsoon factor. And as I mentioned in both the records, from the Ghaggar as well as in Jamna, we have very strong climatic, correlation between the major phases of aggregation and the climatic events. And most of this data is coming from ocean records, by the way. So the compilation of the climatic records has come from the ocean. There may be minor lags, which are part of the system, but broadly, we do recognize that most of these climatic factors are correlating with the events. Yes. Yes. Yeah, that record is here only. This is literally the marine record actually. Thank you.
If there are,
oh yeah.
Road made by the tectonic thrust towards the northeast.
Yeah, I was trying to answer that same thing here. That I mean, see, you to move a river like Satluj or even Jamuna, you need some external forcing to happen, right? It could have happened through tectonics, but it is not essential, and that is the reason why I gave you the example of the Kosi River. The Kosi River migrated by 100 kilometers in one shot like that, And there was a different reason. There was a sedimentological readjustment. The river bed was so high that it just could not be contained within the river, within the channel, and it evolved. So there are alternative hypotheses available. It is not necessary that tectonics would have played a role only. There are other reasons as well. But I think we are yet to discover that. We have no documentation on that as of now.
So will any of your
Yeah.
core samples
Yeah. No. No. No, there is, these are all riverine, stuff, and we did not discover, no, no. The sea never reached here. It is too far from the sea. If you go further down in the Indus, there people have discovered some, interaction between the fluvial and the marine, but not, this is too far. Yeah.
Sediments from the Himalayas.
Yeah.
The Himalayas also do contain marine sediments.
Well, they will be reworked, and the fossils are really unlikely to survive. So yes, the sediments are coming from the Himalaya only, but these are all reworked sediments, right? They have undergone several cycles of deposition, so it's very unlikely that these tiny forms will actually survive. We haven't discovered anything. In fact, the best sediments, best type of sediment where these are preserved are muddy sediments. We are looking at sand material, which is very hard to find anything. Yeah, exactly. All the recycling has happened. There's one more there.
There is no time, maybe you could.
Yeah, you said the Saraswati River died by about 12,000 years back. And the literature, you said this is mentioned in the literature. So can we relate the time of those literature to at least 12,000 years old?
No, the Vedic literature doesn't really mention about the time. Vedic literature mentions about the civilization part of it, but it does not really mention about any specific time of the existence and the demise of these rivers. I don't think there is any data on that. These are very vague data sets. These are scientific data sets which are coming from very specialized analysis.
Correct, but if the river didn't exist after 12,000 years old, then how somebody could mention about it unless they see it? That's my question.
Yeah, so river existed until about 8,000 years. It was smaller versions of the river which existed around that time. So the Vedic text is not limited to just 4,000 or 5,000 years. It's going back in time, basically, right? So the rivers, there were signs of, there were enough signs, enough signatures of a large valley, large river valley which was existing at that time, but not until the demise of the younger, the mature Harappan civilization, basically. So all I'm arguing is that the river existed, no doubt about that, but most of the large part of the river was died. That died, by about 8,000 years or so. Jamuna died or moved, for example. I would say it did not die. What happened was that this river moved to that position. That's all. It did not help you die. So there was, large rivers always existed, but it was not existed around this area where the civilizations were developing. That's the point.
I'm sorry, I have to stop this.
Thank you. Thank you so much for your attention.
Thank you. So the next speaker is a young associate, Bibin Anand. Anand is a research assistant professor from the Department of Biotechnology at SRM Institute of Science and Technology at Kattan-Kulatur. He did his PhD from IIT Jodhpur, then he continued further to work at University of Alberta in Canada, and then moved to Boston, and slowly he moved towards neurodegenerative diseases, and he's going to talk about that to us.
Thank you, ma'am. So warm good afternoon to each and everyone. Thank you so much for giving me an opportunity to talk about my research work. It's a great pleasure for me to talk about my research amidst of the esteemed members in the country. So I'll be talking about a title known as Understanding the Molecular Mechanism Underlying the Protein and Metabolite Cross-Catalysis and Coalescence Using Biophysical Approaches. So it's nothing but protein co-aggregation and cross-seeding reaction. So we all know about protein. What are proteins? Protein is present everywhere, especially in most of the plants, animals, and we humans have a lot of proteins. And these proteins are, synthesized from our genetic code as a form of, polypeptide chain of amino acid, then further folds into a regular, beautiful three-dimensional structure. And the native structure of protein is very important for its function. Either it can be, controlling the brain and, wound healing and other structural functional activities though. So, there are several factors, that are responsible for the structural plasticity and stability of these proteins. So, under unfavorable conditions, these proteins, tend to aggregate or, they misfold due to various factors and form, some toxic conformers. For example, you can say about, A beta 1 to 40 or 42 peptide that is, most commonly present in Alzheimer's disease patients. So if this protein misfolds into a random conformers, they lead to a disease known as Alzheimer's disease. And another classical example is about prion disease. It's the prion protein is rich in alpha helical conformers. So due to any structural mutations or any unfavorable events, these protein lose its alpha helical conformers and then end up in a very rich cross beta architecture, and these are known as amyloids, and they then self-assemble and form plague-like entities in the brain that leads to prion disease. So as I mentioned earlier, so the conversion of any soluble protein from its native structure to well-organized higher order morphologies are known as amyloids, and these amyloids are named as named by a scientist known as Rudolph for its beautiful cross beta architecture. So the accumulation of amyloid fibers due to protein aggregation is a hallmark for several diseases, and recently single metabolites have been reported to form amyloid-like structures that have similar, that exhibit similar biophysical, biochemical, and cytotoxic properties like that of amyloids. So a mechanistic understanding of protein aggregation is very, very important. As we know, the protein has to be synthesized from the ribosome, and after that it undergoes a folding process to attain its native conformers. And due to unfavorable events like mutation or unfolding or misfolding, it forms a toxic conformers known as amyloids that are rich in cross beta architecture. So due to this amyloid formation, there are two things that is happening in our system. One is loss of native function of protein, and another is toxic gain of function. So it's very important for us to understand the mechanistic process that is happening in our system. So here is a graphical representation that shows the list of amyloid-linked diseases like Alzheimer's, Parkinson's, ALS, and Huntington. As well as there are several non-neurodegenerative diseases are linked with protein aggregations like type 2 diabetes, cataract, and tumorigenic cancers. So this is a beautiful movie by Aldrin and Elcock that shows the coexistence of several several proteins inside our system. So these proteins try to bump up and try to coexist in our system as a combined unit. So due to some unfavorable events, if some protein aggregates, these try to bump up with each other, leading to the various disease severities. So anybody can study the protein aggregation. So here is a graphical representation that shows the process of amyloid fibril formation. As you see here, the soluble protein misfolds and forms another conformers known as oligomers. These oligomers further act as a nucleating agent, then form protofibrillar entities, and then further self-assemble into and form as amyloid fibrillar entities, and these fibrils further self-assemble to form amyloid fibers, and these fibers are rich in cross-beta structures. And several biophysical tools can be used to understand the protein aggregation. So as we all know, several protein coexist in our system, and they are we believe, like for example, if there is a small structural variation or if there is an unfavorable event that triggers the aggregation of one protein, what will be the fate of other protein? This is a kind of fundamental question, and there are several reports that suggest the coexistence of two different amyloid linked diseases in a single patient. For example, an Alzheimer's disease patient will have, coexistence of another protein aggregation disease. So we questioned what might be the reason for, such coexistence of two amyloid-linked diseases. So we try to understand the co-aggregation of protein system. So we did protein aggregation studies under temperature-induced protein aggregation system. Aggregation studies. That is, we tend to aggregate the protein close to its melting temperature. As you see here, this is the thioflavin T aggregation kinetics graph that shows the aggregation of BSA and insulin molecules. So these BSA and insulin protein aggregated with by showing its respective thioflavin T signal with respect to time. So once we mix two proteins together, aggregation was faster. And once we mix three proteins together, the aggregation was even faster without any lag phase. So we believe that there is a coexistence of two diseases, two amyloid-linked diseases, and there could be a monomer interaction that favors this lethal aggregation process, as well as the formation of amyloid-prone intermediate species may trigger the aggregation of other globular proteins or proteins present in its vicinity. And apart from that, one interesting information is that we believe that there is no sequence similarity does not play any key role or major role in triggering protein aggregation process. As you see here, these are the TA micrographs of protein aggregates that we got, and that shows the regular fibular architecture that is known for amyloids. So from here, it is evident that protein can co-aggregate and form heteromeric amyloid fibrils. That can be one of the reason behind this kind of protein aggregation linked diseases or the coexistence of two different protein aggregates. So now we understood why there is a coexistence of two different amyloid-linked diseases, and we questioned that is there any other unfavorable event that happens in our system? For example, if a protein misfolds and form amyloid-like structures, so this protein amyloids can trigger the aggregation of other protein present in its vicinity or not. That is kind of understanding the protein cross-seeding reaction. And we observed that there is a clinical report that shows the existence of 483 different biomolecules in an A-beta plaque. It means of postmortem patient samples. So this question that why there is different number of biomolecular entities in the system. So which is the key factor that is triggering the aggregation of these biomolecules present in the vicinity of this protein. So we try to understand. We want to understand this phenomena. So we thought of studying this. With one of the very interesting peptide that is A-beta 40. We know that A-beta 40 is linked with a disease known as Alzheimer's. The aggregation of A-beta 40 is linked with a disease known as Alzheimer's disease. So Alzheimer's disease is a progressive neurodegenerative diseases. So due to this disease, leads to the formation of neural dysfunction or leads to the leads to neuronal loss, and this is caused due to the two key peptide that is 40 and 42, and so we believe that the coexistence of these biomolecules in the plaque, if we identify why there is a coexistence of these biomolecules in the plaque, will help us to unravel vital information for devising therapeutic strategies against these diseases. So first, as I mentioned earlier, so we don't know what is which is the process or which is the important event that initiate the cross-seeding process. So we want to understand the cross-seeding process. We started with A-beta 1 to 40. So we try to aggregate A-beta 1 to 40 at mimic physiological condition. Here is a thioflavin T signal that shows the aggregation of A-beta 1 to 40. As you see here, once we mix these preformed A-beta fibrils to monomeric A-beta, it induces the rapid seeding of A-beta 1 to 40 without showing any distinct lag phase. And this is the TA micrographs and AFM scans and thioflavin T stained images of these obtained aggregates. So as I have said earlier, we want to understand why these A-beta plaques are multi-component entities or multi-component system. So we try to study the effect of preformed A-beta 1 to 40 fibrils with monomeric protein molecules. So we've selected few proteins like globular proteins like BSA, insulin, and lysozyme and cytochrome C. So we try to seed the reactions of these proteins. So what we took, we took these preformed A-beta fibrils and seeded with different protein monomers. And as you see here, protein monomers did not aggregate as you see here, there is no thioflavin T signal. But once you add the preformed A beta 40 fibrils, it was able to seed or trigger the rapid aggregation of globular proteins present in its vicinity. And this is another example that shows, this is a CD cyclodichronism spectroscopy of these proteins before and after aggregation. As you see here, there is a clear conformational switch towards beta sheet structure from these proteins. So from this, we believe that A beta 40 fibrils can do one interesting phenomena, that is cross-seeding. These preformed A beta 40 fibrils can trigger or rapidly cross-seed the globular proteins present in its vicinity. Not only proteins, it was able to trap the aromatic amino acids present in its vicinity too. So here is a thioplavin T stained images of these formed aggregates. As you see here, all these aggregates are having regular fibular architectures, and these are the AFM images that shows the fibular architectures of these amyloid structures. So from this study, it is evident that A beta 40 or A beta fibrils aggregates, and after aggregation, it has another potency to trigger or trap the protein monomers present in its vicinity, as well as not only protein monomers, it was able to trap the aromatic metabolites present or aromatic amino acid present in its vicinity and form a multi-component system, and that may be one of the reasons for the multi-component nature of A beta plaques. So from here, we believe that there is a strong interaction between monomeric as well as fibrous species, as well as the amyloid-prone intermediate species can interact rapidly with the protein monomers, and they can able to trap the protein monomers towards an aggregation reaction. And more specifically, this protein aggregation is independent of sequence similarity. So so this is just, I want to highlight the relevance of amyloid co-aggregation and cross-seeding that leads to a cascade of events in our system. So we believe that cross-seeding it can be one of the reasons that induces the neurodegenerative diseases, as well as there is a rapid co-aggregation and cross-seeding between these protein molecules. So that's why the patients have the risk of developing other protein misfolding diseases in the system. So after studying, understanding this protein aggregation system, we want to understand, like, if protein is aggregating, there are several reports that says that even single metabolites can aggregate. So there are certain disease conditions where the level of tyrosine has there is a homeostasis in the tyrosine metabolism can induce any disease or not, or it is linked with any neurodegeneration or not. This was a kind of constant question with us. So we want to understand the self-assembly mechanism of these metabolites, because these metabolites have been reported to self-assemble and form higher order structures that has amyloid-like biophysical, biochemical, and cytotoxic properties as well in case of tryptophan, phenylalanine, and dopamine. So we try to understand the aggregation of tyrosine at physiological conditions. So we try to aggregate these tyrosine molecules under mimic physiological condition. As you see here, this is the thioplavin T kinetics graph. Those shows the rapid aggregation of thioplavin T at mimic physiological conditions. And here is the same image of these formed tyrosine aggregates, as well as this is an AFM image that confirms the presence of higher structures. Apart from that, we could also observe the presence of spheroidal entities too. So from this data, it is evident that tyrosine molecules under mimic physiological condition can self-assemble and form higher order structures that have similar biophysical properties like that of amyloids. So next we questioned, would tyrosine fibrils drive the aggregation of globular proteins or not? And does the resultant fibrils have any effect on biomolecular system? So we try to understand the aggregation or seeding potential of these tyrosine aggregates with globular proteins. As you see here, this data suggests that tyrosine aggregates were able to induce rapid cross-seeding reaction with the globular proteins. That is, it is able to trap the protein molecules in its vicinity and make it to aggregate in a higher order conformers or toxic conformers. And again, we also checked the biological relevance of these preformed tyrosine aggregates. So we did a dose-dependent seeding effect of these tyrosine aggregates, and we found that these tyrosine aggregates were able to trigger the aggregation of protein in a dose-dependent manner. So from this data, we could confirm that there is also a cross-seeding reaction that is happening due to this tyrosine aggregates, as well as our results, even unpublished data, some suggest that these tyrosine fibrils or tyrosine generated, amyloid structures were able to cause intense damage to the neuronal cells. So from this study, we could understand that tyrosine molecule aggregates under physiological conditions, and tyrosine molecule can induce cross-seeding of globular proteins under physiological conditions. And apart from that, hydrophobic interaction seems to be crucial for this lethal aggregation process. So we believe that these results are highly relevant to devise or strategically design therapeutic targets against amyloid-linked pathologies. So another one interesting observation is that after studying with tyrosine, we want to understand, the mechanism of phenylalanine aggregation, because there are reports that suggest the, rapid heating and cooling of phenylalanine molecules can, induce them to form higher order structures. So we want to understand whether phenylalanine molecules can self-assemble and form higher order structures. So we did observe that phenylalanine molecules under mimic physiological conditions were able to self-assemble and form higher order amyloid-like structures, and these phenylalanine molecules were able to induce a lethal aggregation lethal aggregation to the globular proteins. So you might be wondering why we want to study about the phenylalanine aggregation, because excess accumulation of phenylalanine is linked with one of the important disease known as phenylketonuria. So we want to find out whether these phenylalanine fibrils were able to trigger rapid hemolysis of these red blood cells, and we've observed that these phenylalanine preformed phenylalanine fibrils were able to induce rapid hemolysis to the red blood cells, as well as the phenylalanine generated amyloid fibrils were able to cause an intense hemolysis, and we believe that excess accumulation leads to aggregation of phenylalanine molecules, and then further this accumulation of these sweeter-ionic phenylalanine molecules were able to trap the globular proteins at its vicinity, and then induce adverse cross-seeding and co-aggregation reactions. So another important observation after doing this, phenylalanine study, after working with phenylalanine, we all know about aspartame. It is one of the ubiquitously used, artificial sweetener, and more than 6,000 food products contain, aspartame. So aspartame is a dipeptide made up of phenylalanine and aspartic acid. So we questioned actually if phenylalanine is aggregating, does aspartame aggregate under physiological condition? And we were like, strikingly, we found that these aspartame aggregate under physiological condition, leading to higher order amyloid-like structures. And these aspartame molecules were able to induce rapid co-aggregation, cross-seeding, as well as it was able to kill the bone marrow stem cells as well as neuronal cells. Not only under cells, these aspartame aggregates were able to induce rapid hemolysis to the red blood cells. So we believe that it is very important to understand or to redraw about the concept of using the artificial sweetness in all our foods. And from all this data, we could understand four important things. That is, protein aggregation is independent of sequence similarity. Apart from this, there is a mechanism known as templating mechanism, where preformed aggregates can able to trap the monomeric proteins into the aggregated conformers, as well as this protein aggregation is mainly driven by hydrophobic and electrostatic interactions. So throughout the research, we asked few questions. Does hydrophobicity play a pivotal role in initiating protein aggregation? Is cross-seeding influenced by templated dependency? As well as, what is the significance of metabolite aggregation, and can metabolite aggregates can be linked with other diseases or not? So these are the few questions we try to ask, and we understood a lot of important things, and we are still working on all these things. And I would like to thank my PhD mentor, Dr. Karnakar Kar, who and my junior, Kailash. So we all work together to understand these very important and complex phenomena that is happening in our system. And I do thank my collaborators and colleagues. Apart from this, I would sincerely thank my IIT Jodhpur and JNU, where most of this work was carried out, as well as Academy for giving me a wonderful opportunity. And I do thank the funding agency who funded this research work. And currently, I'm a research assistant professor at SRM Institute, and I'm kind of establishing a biomolecular self-assembly lab, and I'm backed by 4 of these students, like they are BTechs and MTechs, and I do work with Dr. Anil, who is working with neuromodulation. Yeah, thank you.
Thank you very much. Let's give him a big hand. There's a question over there. Three questions only, because I've seen three hands. We don't have too much time. Yeah, show me. Show me.
Yeah. So I'm here. Good talk.
Thank you, sir.
This method, how it actually get aggregates, what actually makes them aggregate, and
So,
what initiate, just let me finish. That is quite not known, even in mouse model or human model.
yes.
So doing that in the table or in a test tube, do you think that it has any relevance with what is happening inside the brain?
Yeah, exactly. It's a wonderful question. So, this is what, we are also trying to understand. Uh, for example, yeah, in test tube, we can understand, like, we can mimic some concentration, and then we tend to, understand whether it's aggregating or not. But in case of A beta 40, it's like kind of, reported concentrations that tends to aggregate. And as well as in, animal models, it'll be very interesting to, study those important phenomena in animal models that actually I want to do. That's why, I came back in India. I want to establish these kind of studies because, about tyrosine forms very nice cephalosome structure, but no one has solved the structure yet. So, and even like lysozyme aggregates, its cross beta architecture is not yet resolved. So I want to work on all these, important things, like kind of, working on animal models, as well as I did find some collaborators who in Canada who has this kind of PKU mice model systems, where actually I want to, look into these things. I think it's a kind of future perspective.
Hello. Sir, a nice presentation.
Thank you.
I'm wondering, because most of the experiment you have performed, you have used concentration which is very far than the what is expected to be present in cell. And the second thing is, because you are saying that all protein is going to form aggregate in absence of even any structure similarity. So then it create chaos in the cell, all cell is getting aggregate. So it
Yeah.
should have some, trigger molecules, trigger sequence. Without that, all cell is going to be aggregate only.
Yeah,
So what is your comment on it?
yeah, exactly. So, for example, I was mentioning on hydrophobicity. In case of A beta 40, it's the sequence from 16 to 25 that is rich in phenylalanine molecules. Two phenylalanine residues are there in A beta 40 and 42. So, we believe that that region is a kind of aggregation prone region that induces the conformational switch of A beta 40 or 42. And apart from that, as I said earlier, like, aromaticity is one of the key factor. So maybe it can be tyrosine or tryptophan, phenylalanine. We believe like these aromatic residues are more crucial in inducing the aggregation. What you said was, I do agree that like some of the experiments, are not exactly matching the physiological condition, but few does. Yeah.
Okay, thank you.
You talked about the aggregation of aspartame,
Yeah.
and also cautioned that the use of such sweeteners are dangerous. But aspartame is not absorbed as dipeptide to the cells, actually. It is digested as phenylalanine and aspartic acid.
Yes.
So there is no issue in that case, right?
Exactly. This was the kind of, debate. I do understand the importance, but I believe that, this has to be again rechecked, because if I'm having an aspartame in my, maybe a kind of whatever. So we don't know what is the state actually. While consuming, I'm not aware, am I consuming the aspartame oligomers or aspartame aggregates? Once it goes to my system, it might be breaking down into phenylalanine and aspartic acid. But phenylalanine, though, phenylalanine even self-assembles. So there is another question, and it also leads to the formation of methanol, I think so. Yeah. So there is a kind of a debate. So this is kind of, we are still working with this in a different aspect.
Thank you. Thank you, Vishnu.
One question only. Actually, I want to ask, is there any particular concentration at which these aggregates are starting? In case of aspartame, you were saying.
Yeah, aspartame aggregates even at, I think, 0.7 millimolar. Yeah.
0.7
Yeah,
millimole.
yeah. It's aggregates even at the lower concentration. That is, that's far lower than it is present in most of the food products.
And it's only due to the hydrophobic interaction you are talking about. There is no hydrophilic interaction in that?
Yeah, because it's made up of phenylalanine and aspartic acid.
In case of proteins, we are having amino acids.
Yes.
And we are having positive and negative ions. Electrostatic interaction must be there.
Yeah, electrostatic interaction is there. So we believe like once it aggregates,
yeah.
So this can be a template, or this can be a conformer that will, because our protein is always dynamic. So our protein can switch itself to that kind of conformance. That's why I say it as a template dependent seeding activity might be observed over there.
Thank you.
Thank you.
Thank you very much.
Thank you.
And, so the next speaker, I'm really happy to chair a session where my friend Shangmita is speaking. She is a professor at the Indian Statistical Institute at Kolkata, and also the second-time director of all the ISIs. Her research interests include computational biology, soft and evolutionary computation, artificial intelligence, and machine learning, all very well-known terms these days. I'm not going to read the awards she gets because it's just too many, including the Padma Shri last year. And thank you very much.
Thank you. Thank
Keep to
you.
time.
Yeah, and first of all, I would like to thank the Academy for giving me this opportunity. Am I audible? The back?
Yeah, we
I
can
have
hear.
a very bad throat, so please excuse me. I'm sure I'll be coughing sometime, so I have my water here. I come from the Indian Statistical Institute, and I want to tell you a little bit about the work that we've been doing in machine learning and its applications in biomedical research. I am a computer scientist, so my area of interest, I am happy when I'm developing algorithms. And I was quite terrified of biology in my school days, but then it so happened that a student joined me who got me interested in this area. It was a two-way traffic. He taught me biology, I taught him computer science. So it was very convenient in that sense. So I learned a little bit of biology so that I understand what I am doing or the applications that we are doing. So artificial intelligence these days, everybody is hearing about artificial intelligence. It's going into domains never thought of before. What is artificial intelligence, how it stands vis-a-vis the machine learning that we also talk of, and that is in the title of my talk. And in these days, we cannot escape this term, deep learning. It's everywhere. So artificial intelligence is the superset. A part of it is machine learning. There are things which are in artificial intelligence which is not machine learning. And a subset of machine learning is deep learning. Machine learning essentially means a computer, the system, will learn from the data that is given to it. Artificial intelligence could be like expert systems, which is not machine learning. Rules are coded there, and then the system, it reasons it out through those rules which are already put there in the system. Those are expert systems. But machine learning is, I give it sufficient amount of data set, and it tries to learn things without being told explicitly. So as I said, AI, we try to program machines to mimic human behavior, and now we are in this era of deep learning and generative AI, where even when we are doing, let's say, neural networks, artificial neural networks, traditional ones, we have to tell the system that these are my inputs and this is the expected output, and then tell me. I have to extract the features. I'll come to it in one of the slides, it's there. Deep learning has removed that requirement also. You just give it raw data, and it tries to learn the input-output function. So I'll not spend time because time is very limited here. Turing test is something which was devised in 1950s to define when can we tell that a system is artificially intelligent. Turing test passed to a certain extent. CAPTCHA that we do every day is a reverse Turing test, actually. So AI, as I said, AI is these days everywhere. You think of city planning, you think of autonomous vehicles, healthcare, agriculture, industrial automation. It's IoT, it's Internet of Things. You have many, many devices, many, many sensors taking input all the time. And then you reason it out and you analyze that data through artificial intelligence methods and come up with something, some predictions or summarization, etc. So machine learning is when you are given, for example, pictures of cats and dogs, and you're given a new picture now. You did not see it before. You have to reason it out that this is a cat. Now to come to healthcare in particular, AI and ML in healthcare, uh, various aspects of healthcare, we can find applications of artificial intelligence and machine learning. For example, you have the patient management systems, the electronic health records. There, you can use artificial intelligence methods to make various types of predictions, looking at all the data which is there. You can improve the diagnosis, improve diagnostics, better patient care, personalization, survival prediction. Also, using AI and ML methods, you try to bring down the costs as well, improve population health in general. So various aspects are affected by AI and ML. And an MIT report, a part of it is, I've taken from an MIT report. It says that there have been different revolutions in healthcare. The first one, of course, it started with the discovery of the structure of the DNA. Then looking at entire genomes, looking at what makes up the genome, understanding the genetic basis of diseases, looking at data in multiple modes from different sources of data. We try to analyze and come up with our analytics, come up with predictions, looking at genomic, proteomic, expression, images, clinical data, a lot of it. And now we are in this convergence phase where all the disciplines are coming together to solve the problems. We have the life sciences, physical sciences, at the core, in engineering, technology, mathematics, statistics, everything has to come together now if we are to really solve the problems that is facing us, facing humanity. And not just in healthcare, I guess this is true for any problem, climate change, environment, any problem that you can think of, it's an era of convergence now. So to just put it in perspective, we have the cell, inside the cell, the nucleus, we have the chromosome, and if we look at it in detail, it is the double helix here. And if just one quick slide of the central dogma of molecular biology, where we know that parts of the DNA is the gene through the process of transcription, we have the messenger RNA because it is carrying the message for the corresponding protein. And then from the messenger RNA, we have the peptide, which is a sequence of amino acids. Without going into the details of all these, so we know that we've already heard from the previous speaker about the proteins and how it folds, and then we finally land up because of various interactions with the tertiary structure of the proteins. And proteins are sort of omnipresent, doing variety of jobs in almost anything that we can think of. So what I'm trying to now do is to give you a flavor of different things, four things that we've done in our laboratory from, let's say, about 10 years back to what we are currently doing, because it would be very difficult to explain each in detail in these few minutes that we have. But just to give you an idea of what's going on, that this is in my group. So we started off actually, I started off in computational biology with this particular problem of looking at small RNA molecules called the microRNA. And this was when microRNAs, not a lot of research had been done in microRNAs. So we were looking at that molecule, of course, computationally, all computational algorithms and the data that was already publicly available. We did a fairly comprehensive study of the involvement of microRNAs in various diseases, in particular in different types of cancers, and found that it was very heavily implicated in almost all types of cancers. So without going into the details of the biogenesis of microRNAs, what happens is a normal protein coding gene would, inside the nucleus, form the mRNA, the mature, it would come out. The mRNA would normally have produced a protein, but at some point, this microRNA gene, which is also in that same genome, gets active and produces a small RNA molecule called the microRNA. It's just about 20 to 22 nucleotides long. It's the mature microRNA. What it does is it goes and sits with this mRNA in the region preceding the actual in the UTR, it is called, of the messenger RNA, of its target messenger RNA. It either goes and represses the translation of this messenger RNA into its protein, or it simply degrades this messenger RNA. The effect is the same, that the protein which was originally getting produced does not get produced anymore. So this is how microRNAs typically work. And we were studying the direct effects of microRNAs. We were also studying the timed effects, meaning secondary effects of microRNAs. So we had a fair amount of publications in that area. But what I will just tell you about is a machine learning problem that we tried to solve there, which was a classification problem. So we wanted to know that if a new microRNA comes up, what is it doing there in the system? And in order to understand that, we have to figure out what messenger RNA it is targeting. If we are able to figure that out, then we would know that this messenger RNA was producing this protein. So that would mean that this particular microRNA has some effect on that protein. So it gives us some handle on that new microRNA that we have discovered possibly. And it was posed as a computational problem of classification, very well known. There are many well-known classification techniques. So the problem essentially was, I have a set of a pair, I have many pairs, microRNA and mRNA pairs, which are known to be targets or known to be non-targets. So people, for example, for this pair, biologists have actually experimented, put them together and found that this microRNA has no effect on the messenger RNA. The expression of the messenger RNA does not change. So that means it is a non-target, very likely to be a non-target. So this was the problem. We had a set of targets and a set of non-targets. These are microRNA and mRNA pairs. Now given a new pair about which we do not know whether it will target or it will not target, we have to infer, we have to predict whether this is a possible target of this microRNA. So this is basically a classification problem in computer science terms. The problem here, there were some prediction methods which are already available, but when we checked out all these algorithms which are there at that time, the results were widely varying. Okay? And we figured out the machine learning methods, typically they need training data. And training data means there should be enough positive data, there should be enough negative data in order to train a classifier. The problem is, in this scenario, there was a lot of biologically validated positive data, positive meaning targets. A lot of it was available, but very few non-targets were available because biologists, when the experiment failed, a particular microRNA did not target a particular messenger RNA, they did not report it. So there was a lack of negative data, and therefore the machine learning people, what they were doing is they were artificially generating the negative data. And in that process, that artificially generated negative data became very much different from the true negative data. It was also quite different from the true positive data. So what happens is, if you have a positive class here and a negative class here, which is very distinct, it is very easy to classify, put a boundary in between. But then the real data is not that distinct. It is actually very much overlapping. The negative data has a lot of has a lot of characteristics like the positive data. The positive data has a lot of characteristics like the negative data. So real data is not that simple. So since people were actually working with artificially generated negative data, which made it quite different from the real negative, so there was a wide variation when it actually was applied to real data sets. So what we did was, we came up with a sequence of steps of generating the negative data. We actually started from a set of predicted positives, predicted targets. And from there, we filtered off, using very, very stringent methods, anything which had a possibility of being a true target. Anything which had a possibility of being a true target was eliminated from that set of predicted positives. And now what we were left with after various steps of filtering was a set of negative data which was which had positive characteristics, because these have come from predicted positives. But it had very, very less chance of being a true positive. And indeed, the classification was standard. The results are here. You see, this has a very high, it has a high true positive rate and a low false positive rate. So this is the quadrant where we would like our algorithm to be in. This, and another very popularly used method, target scan, it appeared there. The others were either too conservative or too, what should I say, too optimistic. So they were performing at different levels. And we use this method. In addition to that, we actually, we made a regulatory network inside the cell. So gene and transcription factor networks are well known. Transcription factors switch on or switch off the different genes, and transcription factors themselves, being proteins, would be generated from some gene. So there's a gene regulatory network in action inside the cell. We at that time put in also the microRNAs. That was sort of the first work where microRNA induced transcription factor gene regulatory network was studied. And we studied it for breast cancer and colon cancer, did some graph theoretic analysis, and came up with some interesting results where certain microRNAs, which was not known to be involved in those cancer types, they those got predicted. So that was interesting because this was, these two were new microRNA predictions for breast cancer, and this microRNA was a new prediction for colorectal cancer. Our work had to stop thereafter because we could not take it further, but we published, hoping that somebody else somewhere would pick it up. And people did pick it up, validated for the one microRNA got validated for breast cancer. I have not checked out later what happened to these others. Quickly again, because I don't have time, we also, with one of my students who did her PhD, we looked at the drug design problem, posed it as a problem of multi-objective optimization. So what we know, for example, this is a flu drug. This flu virus. So this virus has the RNA inside. It has several proteins, which helps it bind to the surface proteins of the human cell, helps it multiply, helps it, again, escape from the human cell and go and infect other human cells. So the point is, can we target any of these viral proteins which is necessary for the survival of the virus through small molecules which could be drug-like, which are drug-like molecules. So this is, again, I don't have time to go into this, but a team from Australia, led by Professor Peter Coleman, actually designed a small drug molecule using such methods, which is rational drug design, methods of rational drug design. And this small drug molecule called Relenza, it was actually discovered through these principles. We also tackled this problem, but we were looking at the problem from a multi-objective optimization point of view, as I said, and we were looking at different objectives which we want to optimize. For example, the molecule themselves should have very high, should have very low energy component. It should have high oral bioavailability, et cetera, et cetera. So different objectives we were trying to optimize at the same time. And we were looking at multi-objective optimization techniques that we had developed in our own laboratory. So it is not that you have 5 different objectives, you just combine them W101, W202, and come to one objective and then you optimize. That is not how this will work. It is all the objectives are kept separate and optimized simultaneously. So what you get is a set of solutions at the end. So what we were doing is we were looking at these 3 objectives which we wanted to optimize, and we were targeting the REC-A protein of Mycobacterium tuberculosis. REC-A protein, as there was a discussion in the morning, it repairs the bacterial DNA, and therefore the antibiotics which harm the bacterial DNA, REC-A is responsible for that DNA repair. So REC-A is quite a well-used target for different viruses, including for bacteria. This we were using for bacteria, Mycobacterium tuberculosis. And what we got, interestingly, was several inhibitors. Our method gave several inhibitors which are already known for REC-A target, giving us confidence in the method that we developed. At the same time, several other inhibitors got proposed by the system that we developed, and we did some QSAR studies, quantitative structure activity studies for these molecules also, trying to show that these could be potentially good molecules. But thereafter, our expertise ended, and we were unable to proceed further with our limited expertise. Now we published, and then hoping again that the methods will be taken over by other researchers who can actually look at the biology part of it and test it out. We were also doing some single cell, so clustering methods we have where very small rare cluster types, rare cell types could be detected. And I don't have time at all. I can see the clock ticking. So we had, for example, this data type where very small clusters also could be detected. Ordinary clustering methods would be unable to detect such very small clusters. And in cancer in particular, with such a heterogeneity in the tumor cells, it is very important to detect rare cell types. This method was able to do it. And finally, we are now working in these areas. There's a current area. As I said, from raw data, you do machine learning like this. So you have to extract features, do feature engineering. Deep learning has done away with that feature engineering part, but with graphs, if you are working with graphs, then even this is machine learning, application of machine learning is also very difficult. So what we are doing now is working with something called graph neural networks, where given the graph, we are coming up with embedding of the nodes. Embedding in such a way, this embeddings would be learned in such a way so that nodes which are similar to each other, their embeddings would also be close to each other. So that is, so now for every node, you would have a set. So for every node, you have a vector of numbers. Now you have a data set of numbers now. Apply whatever machine learning technique that you are fond of. So this is the good thing. We are working with a network of drugs and proteins and genes and trying to solve problems of adverse drug event prediction, drug-to-drug interactions, and if there are adverse effects, we are trying to predict those. I'll stop with that. There are some references here, old references as well as the new ones here. And thank you very much for your patience, for listening to me. My acknowledgements to my institute and all other agencies, and thank you very much. And if you look deeper into this thank you, you will see the face of the founder of our Indian Statistical Institute, the face of Professor Prashanta Chandra Mahalanobis. Thank you.
Thank you very much, Shamita.
I did not cough.
Since we are already running a bit late, only 2 questions around, allowed rest of the questions during tea.
With some trepidation, trying to pose a few questions. I'm afraid we'll run out of time, I know that. And there's a lot of material, very interesting material to be examined. I take up the first few sets of slides when you draw a Venn diagram for that machine, artificial intelligence, machine learning, and deep learning. That's one. And there is a reference somewhere in between to the double helix and things of the sort. And finally, you have a graph network and all that. For the concerning these three, I have 3 questions. I'm sure they will get impatient with me. I apologize to them in advance. With reference to the first one, let me consider machine learning first. You have a set of photographs, chimpanzee, one face and all. Then the question is, you are going to train the network based upon these things. But then the problem is, what is the amount of data required to be able to generate meaningful parameters of your network? That is one. Thirdly, with reference to deep learning, what exactly will you achieve without tutoring the machine? Because deep learning is a self-learning, some sort of self-organization you can form. I mean, if you want to treat it that way. No, that's I know we can discuss it at length. So what type of mechanism is involved in deep learning which will facilitate discrimination automatically? So that is one set of questions. The second one is on
maybe one by one, otherwise I'll forget.
No, because
I'll forget the questions.
And the second question is with reference to the double helix and all that. Linus Pauling and others have been fascinated. They have almost hit upon that idea. They didn't succeed. Assume that we have all the data required until Linus Pauling time, and somebody else, Crick and Watson, they came out with the result, and then Nobel Prize and all that stuff. Now, suppose I want to come out with a machine which can declare, hey, there's a double helix based upon the data. What type of processing will you need to do that? That is AI. Suppose the AI, you say, is going to solve all our human problems with that type of data, with no other knowledge. I'm very strict. That means there's a boundary. Can AI declare, hey, I have got the double helix. I will get you a triple helix for some other data. The third one on the graph network. What you are trying to do, if I understand your graph explanation, you are trying to see whether there is a clustering mechanism embedded in the clustering in the data, which will facilitate your node choice. Thank you for your patience. I apologize for the
So first question was.
I would suggest, Shangita, there are several long questions, and
Yeah.
I think you will need long answers.
Yeah, I would need a little bit of explaining.
Unless you have a very short answer.
A very short answer to only one thing. It's not all self-organizing, self-training. For example, in deep networks, you would have to give it enough examples of, let's say 10 different things, 10 different types of images, meaning images of buildings, images of clouds, images of glasses, images of snow. You have to give it enough number of examples and tell it that this is the image of so-and-so, this is the image. So you have to have the tutoring there, but only the input-output. Then I think we have to take it up outside. Yeah. Yeah.
In early literature, there was a terrifying comment. What was that? You are over teaching. You are saturating. Even a minor departure will not be accepted. So over teaching is also very unsatisfactory.
Understood. Now, actually, the point is we have to learn millions and billions of parameters. So data sets have to be really large to train the deep learning models. With small data sets, this is not going to work. So yeah, and the other questions, I think we would need a little discussion. I've forgotten also many of the questions, but we will discuss, certainly.
This during the 90s of the last century, people talked about combinatorial chemistry as a panacea for drug discovery. They discovered, they made millions of molecules, but the whole concept miserably failed. Didn't come up with a single drug molecule. So this machine learning and AI and all that, I'm just worried. I mean, what is
No,
your?
you don't need to be worried. You see, it's at least at this point of time, we are not seeing machines replacing human beings.
No, I'm
So
not
that
worried
human
about that.
expertise
I
will
have
be there.
already retired, so I am not worried about my job. So,
But you said you were worried. So, but the point is, you see, you cannot do without these techniques anymore. You just cannot do at this point still. It is still, we are developing the algorithms, we are controlling it, and so on and so forth. But way things are going in the artificial intelligence research, the entire world is worried. That, in fact, there are calls for stopping AI research, putting regulations in place first, and then continue.
I'm not worried about that. You see, I'm from IIT, and I can see that all the engineers are doing all these calculations without taking into consideration the opinions of chemists and biologists.
That's wrong.
That's and they are talking about drug discovery at the same time.
Absolutely.
The problem.
What we have found, see, it has been easier to collaborate outside India than in India. I'm sorry, but this is a fact. And that level of respect for each other's expertise is sort of missing. A biologist will think, I know everything, and there are software which are out there. I'll just take out of the shelf, and I'll apply and do it. You see, if real problems are to be solved in a meaningful way, experts from all these different domains have to sit together and solve a problem. In India, that has been a real challenge. We've tried, at least thrice I've tried with my students, ended up with acknowledgements in the papers. So naturally, you can understand that PhD students are also getting demoralized. It's only a citation, acknowledgement.
Just one small comment. This molecule that you have showed, this molecule that you have showed, Australian scientists came up with a molecule. That is in the market since 90s. So it is not a rationally designed drug. No, that's a very different. Rational design of drug is a completely different concept, and there isn't a single molecule in the market till today which has been designed rational. Yeah. Thank you.
Last day is Professor Bijnan Bandyopadhyay from IIT Jodhpur. He is a distinguished professor at IIT Jodhpur, visiting professor at IIT Jodhpur. He specializes in multi-rate output feedback-based discrete time sliding mode control and nuclear reactor control. So he is very well awarded everywhere, including IEEE Distinguished Lecture Award, etc. Elected Fellow of IEEE. Thank you very much, sir. I mean, we are already.
Yeah, thank you. Thank you for the introduction. At the outset, I again thank Indian Academy of Science for inviting me to present this talk. Also, I thank BITS Pilani Goa campus for holding this meeting here. So I'll be talking today on modeling and control of large nuclear reactor. This nuclear reactor is first time in India. It has been commissioned at Tarapur and producing electrical power. So in a short time, I cannot discuss the entire work, of 5-6 years, half a dozen PhD students' work. So I will just touch some of the important points, what we have done in those work. And this is implemented, this reactor is at Tarapur, it is producing two of them, 540 megawatt. So basically, being an electrical engineer, control system engineer, I will be discussing the modeling of this reactor, how it is modeled. And this modeling is different from all other reactors which are there in India. There are 10, 15, 11. So that part I will touch. And then I will touch one important algorithm, the sliding mode control, the robust control technique, how it is implemented here. That this control technique is a very novel technique, is one of the most accepted robust control design techniques. It is invented in Soviet Union, in the then Soviet Union in 1950s, and then it was confined there until a paper in control transactions came in 1977, and it sparked research interest all over the world. And this has become a well-accepted technique for the robust control system design. This is so popular in IEEE Control System Society. There is a technical committee on this topic, and they are responsible to hold a biennial international workshop on this topic. So that I will also touch, and then certain simulation results on this reactor, and then certain references. So modeling and control, as I told, the control problem. What is the control problem here? The nonlinear model, how it is obtained, linearization, then a state space model of 70th order, 71st order differential equation, which is model. This is not, small reactor model, that is not sufficient, that kind of modeling. How this big model, the 70th order model, has been developed over several years by myself and my PhD students, that I'll be discussing. And then, as I told, the sliding mode control, how it is implemented in this reactor, and some simulation results and selected references. So this is the schematic diagram of the reactor, that the core part, then control rods, then the coolant, the heat is taken out to the secondary side, then the turbine and all these things, electricity is produced. So basically, it is the Otto Hahn, I think the Nobel awardee of 1939 or 8, I'm not very, around that time. It is due to him that it is the fission. It is, chemist, 1938. Okay, it is the fission reaction that was invented by him, and it is the fission, due to that the power reactor is possible. The concept of power reactor. Electricity can be obtained from the nuclear reaction. So that was first invented by him, and then the power reactor concept came. So what is the nuclear reactor? A nuclear reactor is a device in which controlled fission reaction takes place. The fission reaction, it has to be controlled. That is the control problem. That is done in the all nuclear power reactor. So typically, a nuclear reactor, neutrons are absorbed in the fuel. Uranium fuel is used for this reactor which I'll be talking, which undergo fission resulting into production of fission fragments, neutrons, other radiations, and release of approximately 200 mega electric volt energy per fission. One fission reaction, this much energy. So this is, as of June 2023 in India, 23 power reactors are operational, producing this 7.5 gigawatt approximately, besides 4 research reactors at VRC Trombay and IGCAR, and for isotope production and research. So most of the power reactors in India, pressurized heavy water reactor of capacities 220, 540, and 700 megawatt, using this as a fuel heavy water, D2O. D2O is a moderator and deflector, and also heavy water as a coolant in this pressurized heavy water reactor. So 2 such units are operational at Tarapur since 2005-2006. And 1 700 unit is operational at Kakrapara since 2023. Similar kind of concept, similar design, only it is 700, and it is little boiling is, the limited boiling of coolant is allowed in 700, which is not there in 540 megawatt, which is at Tarapur Commission. There's no boiling, only pressurized water. The heat is taken by the coolant to the secondary side. So the dimension is basically this kind of why the control system engineers are interested to design controller for this kind of reactor. And this the physical dimension of a reactor. If it is roughly 25 times more than the migration length, the slowed down length and the diffusion length, then the no longer that concept of the small reactor will work. Means, I will explain a little more. The category of this is called as a large reactor to be operated at flattened flux distribution, produce electricity. What is the large reactor? The neutron production and absorption phenomena in one part of the core are not felt in other parts of the core. That's why the control system in one part will not work. Although you can control the global power mathematically, but in reality, it will not control the total power of the reactor. That is the basic problem. That's why this spatial control and several control systems are needed for this kind of reactor. This is coupled with non-uniform rate of production and removal of xenon is a species which is a fission product, which is very important, which I'm coming. Why it is? Because it can absorb neutron. So xenon dynamics has to be included. So I'll come to that, how the xenon is produced. So that's why it is necessary, the modeling and control of such a reactor, and not only the nuclear power precursor, also there are 2 products, that is the xenon and iodine, their dynamics are to be considered. And also it is a large reactor, that's why the whole reactor is divided into several zones. In this case, this 540 megawatt reactor, it is divided into 14 zones. It has been divided in other zones also, and then it has been found the simulation results with the benchmark results, which is Sandia Research Lab, they published. So it is compared, and then it is found, but this reactor, the 14 division is all right. So reactor needs to divide several zones, each having own flux detector and reactivity control. So this is the reactor. The front hub and the back hub, it is 8-meter diameter and 6-meter long, this particular 540 megawatt reactor. So this is one of the such thing it is shown. And each zone is to be described by a set of equation, the rate of change of power, which is essentially the proportional to the neutron density, concentration of the delayed neutron precursor. When fission takes place, it splits into 2, and they again decay neutron. So that is also controlling the dynamics of the reactor. That is known as the delayed neutron precursor. Then, among the various fission products, the xenon enjoys a special importance due to its large thermal macroscopic cross-section and long half-life. Large macroscopic cross-section, it can absorb neutron, and it can change the reactivity. So xenon has to be modeled. And the xenon is produced from the iodine immediately after the fission reaction. So iodine has to be modeled. And then the major portion of xenon is formed from the iodine, and only a small fraction results directly from the fission. But the major is obtained from the iodine. This is that reaction, the uranium-235, the tellurium, in a very quick time, less than a second, and then less than a minute, iodine, and then 6.7 hours, iodine is converted into xenon, iodine. Next, and then the cesium, and then the barium, very stable isotopes. They are not of interest for modeling. So this is the formation of xenon 135 in a reactor and its decay. Now I'll come, what is happening in this reactor, why the control problem is very challenging, that what happens in this kind of large reactor, that increase, which is not happening in a small reactor. That's why small reactor, you can model by second order differential equation, one power equation and one precursor equation, which is not true here. Why? This is the reason for that. I will quickly go through those equations, how, but not very detailed discussion of those equations. An increase in neutron flux in one part of the reactor increases the rate of consumption of the xenon. If it increases due to certain fueling, refueling, or disturbance or anything. And then what happens, the rate of consumption that increases and its production through the decay of iodine will happen several hours later. So that the flux of the neutron gets another chance to increase further. And then it increases further. That's the net immediate effect is that increase of the rate of consumption of xenon will further increase rate of consumption of xenon will further increase the neutron flux due to reduction in absorption of thermal neutrons. This process leads to continued decrease in xenon concentration and a steady increase in neutron flux. This may continue for several hours till the production of xenon from iodine becomes appreciable. So it takes several hours, 7 to 8 hours. And then again, the xenon, then what happens? The iodine produced, and xenon is produced from the iodine, and reverse process takes place, and xenon concentration start increasing, and flux is decreasing. So it is about 15 hours in one cycle. So total is 30 hours. What is known as the spatial oscillation. It was not known earlier. And if this oscillation is not controlled, if it is not stabilized, the whole reactor can blow up. So this was not considered. This is not to be considered in small reactor, but this is very important in the large reactor. This kind of oscillation, a lot of research have been done. What kind of oscillations, what are the different frequencies, magnitude, and all these things. So this is known as the spatial oscillation. I think it is also discussed here very briefly that you see the whole reactor, two parts, if one considers, and due to some reason, if the flux increases, then what happens, the xenon gets a chance. Initially, they're all in equilibrium. So xenon gets a chance to absorb those neutrons, and xenon decreases. And then xenon, not much xenon are available from the fission reaction, then the flux further increases. And this goes for, as I told, for some time, and then the xenon produced from the iodine, and after a few minutes, the flux tilting takes place. What is the meaning is that some part of the reactor will be demanded to produce large power, some part is a less power. So flux tilting takes place. And that tilting, it can exceed the design of the reactor, thermal capacity, a lot of other kind of design problems it can cause. That's why this kind of oscillations are to be controlled. So this is dealing with the spatial control of those, power. If we control every zone, then this kind of tilt will not be there. That is the idea. So what is this? This is the control problem of a large reactor. Maintain the reactor power close to the set point called the power demand. Maintain the power distribution closer to the desired power. Maintain the fractional zonal power close to their equilibrium values. This problem has been attempted by various approaches. This output feedback, there are two of them, multi-rate technique in control, that if you create a rate difference, that is a very important problem. Those who are in control system engineers, they know that control system has so much advanced, but there is an open problem. The static output feedback control, the stability of the system is not guaranteed. There is a paper in Automatica, 100 papers are referred by Peter Dorato, who was the chief editor of Automatica. And conclusion is that by using a static kind of output feedback, static output, you cannot stabilize. You cannot guarantee, although it is the simplistic structure, simple structure. So there is a technique called multi-rate technique. In discrete case, in continuous, it is not possible. So if you create a rate difference between the input and the output, then it has been shown that a arbitrary pole placement or eigenvalue placement of a dynamical system is possible using this kind of feedback. So that is called, these Tiwari, Sharma, Reddy, they are all PhD students. Tiwari was a BHC scientist, recently retired. In fact, the PhD first work started 1990 with his PhD work. He is now joined one of the IITs as a visiting professor. He retired from BHC, and then other also did work on this. Sliding mode control is a very, as I told, the important technique which has been implemented this thing. So there I will talk. This is that, the reactor is divided into several zones. And this particular reactor is, the control rod is not inserted. It is the JETCC, zonal control compartment, and there is a water level. So by controlling the water level, the reactivity is controlled, and the fission reaction is controlled, and the flux is controlled, and the power is controlled. So this is the mechanism. This is this kind of reactor. So this is shown that there are this cylinder kind of 8-meter diameter, 6-meter long. So two tubes, two JCC are inserted one side. At the middle, there are two, and other side, there are two. So this 2 is divided into 2, 2, so 4, and middle is 3, 3, 6, and other side is 2, 2. So there are total 14 zones. Water level is controlled independently, and that's why it's the multivariable control problem. There are 14 input and 14 output. Each zone is divided into 14, and each flux is detected by a flux detector. And so that they are now known as the output, and they are known as the input. So it's a large multivariable system, 14 input, 14 output, 70th order system. So powered their iodine concentration. So this is, in fact, to model this equation, it has taken 5 years. So I cannot speak in 5 minutes, but what is done here, the reactor is considered in two flux energy, phi 1 and phi 2, and their partial differential equation. So nodalization is needed, and then it is converted into ordinary differential equation. And then each zone, the reactor is divided different zones. So every zone is represented by their average values. There is no time, space dependent, it is time dependent. So that way, that is two group, and then further it is, I'll quickly run down, and the two group equation, and by it is converted into 1.5 group equation by some technique developed in Stuttgart Research Lab. My student was visiting, and then an effective one group flux for every zone. So there are 14 zones, so 14 flux, and 14 iodine, 14 xenon, 14 precursor, and 14 that reactivity, that the actuator dynamics. So there are, that's why 5 in every zone, 5 of them. So there are 70 differential equation, nonlinear equation, which is representing this reactor model. Then linearization is done. All these things is standard very, and the linear model is obtained. They are the state variable, output variable, and input variable, and it is put in a state space model. Then this control, these are the different things. It is an unstable system, high order, possesses 2 time scale property. It is a stiff system. Some are operating very slow dynamics, some are very fast dynamics. So it is called the singularly perturbed system, stiff system. So control is more challenging. Then this, few seconds that there are fast and slow modes. System response to be observed for hour to conclude the performance of a controller due to the xenon oscillation, and system is controllable and observable. Then there are various techniques. So this SMC, sliding mode control technique, as I told, it is very simple in control structure, and it is one of the most accepted robust control design techniques. So there are sliding mode discrete, because we have to implement through computer. So discrete sliding mode is proposed. Then discrete output feedback sliding mode control also is proposed. Then this concept of, these are the discrete output feedback control, various people have proposed. We have proposed the multi-rate technique of that discrete sliding mode control, and which is implemented. So this is the state space, the discrete model, and this is the discrete, multi-rate lifted system, the Ricci law, and then the control is obtained. So this control, 3 simulations are carried out. One is the power maneuvering transient. That means how it is settling to a new demand from 1800 megawatt. That has been shown. Then initial power mismatch, if it is disturbed the initial power, how it is settling to the new power, the actual, the rated power, the equilibrium power. That will be shown. And if there is a disturbance is created, external disturbance, and then how it is sustained, that external disturbance, and the controller can perform again correctly. So these are the 3 simulations. This is one is that 1800 megawatt is the rated, the full power, and then 10 megawatt per second for 18 seconds, the power is reduced. And then it goes to settle to new value in, actually in 1500 seconds, it goes to within 1.5, and 3000 seconds, it settles in within 1% error. The new demand from 1800, 10% megawatt for 18 seconds, it goes to 1640 megawatt. 1640 watt is electrical power. So 1/3 of, I'm sorry, nuclear power. It is 1/3 of that is the electrical power. And this is the zonal power variation. Then xenon concentration, it takes several hours, 30, 40 hours, because it's very slow. So when such a tracking, the new demand is put, how the xenon, again, it settles to its new demand, new equilibrium value. That simulation is shown, and this is the global power, how the global power is controlled by adding the local power, the zonal power, the 1800, how it is goes to 1620, and then the actuator dynamics, the actuator, the control, how the control is. The control is the valve. The valve can open, -2 volt, completely open, 2 volt, completely closed. So that water level, by closing the valve, it is controlled the water level at the tube, which is controlling the reactivity. So this is plotted. Then tilt, as I told. The 3 kinds of tilts are important here. That is the axial tilt, side-to-side tilt, and the front-to-bottom tilt. That means there, the justified how control is good if the tilts are controlled. So tilts are plotted here, and it has been found the tilt, the top to bottom tilt, all tilts are almost, it settles to 0, and top to bottom tilts, it settled within 2%, which is the actual tilt also when it is designed the reactor. So this is the tilt is plotted, and then the global power, how initially there is an increase in the global power because the initial, there is a mismatch in the initial power, so there is a global power increases, but it settles to again the new values. Then there is a mismatch in the, there is some kind of signal is added to the matched uncertainty, added to the control signal. Then how the different zonal power again settles to, it is increased to 0.01 voltage per second for 10 seconds, and then another 10 seconds it is decreased to 0. So such a disturbance, when it is created, a uncertainty, some signal, some electrical signal is sent through the control, actual control, then that's why there is a change in the power, transient power, and then it settles to, again, its actual value. So these are the 3 simulations are carried out, and other variables.
Mr. Mukhopadhyay, can we
yes,
go to the
done?
summary slide?
Yeah. That's all, and there are several references who have done PhD on this topic. And one of the article which is published in the world largest encyclopedia of online, the EOLS publisher, we were invited to contribute an article there. So this is, let me check, that is, yeah, this is there's the largest volume, 400 print volume equivalent. So we were invited to contribute an article in this encyclopedia and many other IEEE transactions on nuclear science. Thank you.
Thank you very much. And because there's no time, we have really overshot.
One or
We
two
will break
questions
for
I'll
tea and then
love to
further. No,
take
I'm
up.
sorry. No
Huh?
questions.
One or two questions.
I'm
Okay.
really sorry about it, but I've been asked not to. Thank you, all the speakers and the very receptive audience with so much interesting discussions. I thank Academy to give me the opportunity to chair and to such a interesting discussion where I had to stop people, which I should not do, but thank you all. It was a
Thank you very much, Professor Somtita Sinha, for chairing the session. Now we break for tea, but we have the business meeting of the fellows at 4 o'clock. So I would request all the fellows to assemble back here at 4. No, it's here. There's a change of venue. It's going to be here. And for the others, let me remind you that there is a public lecture at 6 o'clock in the auditorium. Okay. So the fellows, please get back here soon.
mandate to uphold the cause of science, the progress in science, and also while maintaining a strong connect with society. And as part of the annual meeting, we usually organize public lectures to communicate certain ideas of relevance to the general public, as well as introduce our fellowship to topics which are relevant to the science and technology, but bring in different dimension of thinking in our ideas. This lecture is being live streamed, so it will be also accessible to large number of fellows who cannot come here. It's a pleasure for me to introduce the speaker for the public lecture today, Professor Gurmeet Kaur from the Center of Advanced Study in Geology, Punjab University. Professor Kaur specializes in petrology, mineralogy, and geochemistry. She has been involved in the international projects such as the International Union of Geological Sciences, UNESCO-sponsored Heritage Stones for recognition, a step forward from 2020 to 2024, and IGCP 637 Heritage Stone designation from 2015 to 2019. She has been the chair of IUGS Subcommission on Heritage Stones from November 2020 onwards. IUGS Publications Committee member, visiting faculty at Laurentian University, Canada, and a visiting scholar at the Department of Geology, Lakehead University, Canada. She has mentored senior school students at the Department of Science and Technology Inspire Camp organized at Punjab University, and was elected a fellow of the Geological Society of India. It's my pleasure to welcome Professor Gurmeet Kaur to speak to us on the saga of laterite.
Good evening, all. Uh, First and foremost, I would like to thank Indian Academy of Sciences for this immense opportunity given to me to speak about my initiative uh, where we talk about stones which have been used in the monuments and why it is important to document these stones. Most of the time we visit monuments and we are very happy looking at the historical accounts or the archaeological accounts, but we don't talk about the stones. So this project is way forward that geology should come to fore, where we document the stones and the stories of the stones, because these stones have documented all the historical accounts, archaeological accounts, architectural accounts, and how the societies have shaped. So my first slide tells you about the structuring of the talk. I have divided the talk into three parts. The first part is stones in monuments or monuments in stones. We have to differentiate. There's a difference, stones in monuments and monuments in stones. Because if there were no stones, we would have never got monuments and we would have never got the historical accounts. Relevance and significance, why they are important and why we need to document them. The second important point, laterite, its formation and distribution, because I have been specifically invited to talk about laterites. The third part of my talk will be laterite, the subtle narrator, because laterite only knows what has happened in the past. We are just trying to interpret. So to break the monotony, I would like to give one example that we all identify this monument. All of us know this is Taj Mahal. And the rock on the left side is marble, which is derived from Makrana. Most of the people, they are aware that Taj Mahal was built using Makrana marble, which was transported from a far-off place, which was not in UP, it was from Rajasthan, a place which is almost northeast of Jaipur, and the stone came from that place. So how the stone was transported for miles to reach Agra. So there are very interesting stories about the transportation of this stone. But most of the stones, they are locally used. For example, the stone laterite. The monuments all along the Western Ghats where laterites are commonly forming, the monuments can give you a beautiful glimpse of the utilization of laterites. And recently, I was at Hyderabad, where I witnessed this sound and light show at Golconda Fort. And I have been to this fort many a times and have even seen this light and sound show 5, 6 times. But this was a different way of looking at the show. And I want to play a small clipping from the show where the narrator tells if the stones could speak. And as a geologist, I vouch stones do speak. We have to have the art to listen to them. So I want to play this.
जिसकी दुनिया में मशहूर थी एक गुलशन था जलवा इस जगह रंगों जिसकी दुनिया में मशहूर थी बेगमों की हंसी गूंजती थी यहाँ शाह की शान शौकत भरपूर थी आइए आइए इधर से आइए मैं सुनाता हूं आपको इस किले की कहानी ये पत्थर देख रहे हैं ना अगर इन पत्थरों की जबान होती तो गोलकुंडा के ये पत्थर हमें जाने कैसे कैसे किस्से सुनाते बहादुरी और हिम्मत की शान और शौकत और ठाट बाट की बीते हुए युगों के किस्से जो वक्त की बहती हुई धारा में विलीन हो गए क्या शान थी इस किले की इन बुर्जों पर कभी सिपाही पहरा देते थे इन मेहराबों के नीचे से होकर सुल्तान गुजरते थे इन गलियारों में बेगमों और रानियों की पायें खनकती थी और बाजारों में दूर दूर से मुसाफिर और व्यापारी आते थे आज सिर्फ बाला हिसार की ये सूनी इमारतें वीरान खंडहर और उजड़े हुए महल ही गोलकुंडा की बीती हुई शान की निशानियां हैं ये दीवारें और चट्टानें ही उस शान और शौकत की गवाह हैं वक्त और इतिहास को चुनौती देती हुई खामोश रहस्य भरी और अटल
So this small clipping tells us that whatever we interpret vis-a-vis this Golconda fort is through the rocks, through the monument built in the rock. So the essence of my talk is based on this message only, which was clear to the narrator 23 years back when this video was recorded, but it took us so long to understand that we need to document the stones. We read a lot of plaques where it is displayed that this ruler was part of the fort and then it was invaded by the next ruler, by the third ruler, and how they changed the fortification, they enlarged the boundaries of the fort, or for that matter, any other monument. But we don't talk about the stones. All right, so Makrana marble I already showed. The second stone is Jaisalmer limestone. I'm first trying to give you a little account of why stones are important, then we will talk about the laterites. The first one is an expression of love, memory, architecture, craftsmanship, and aesthetics. We all know it's a UNESCO World Heritage Site, Taj Mahal. But strangely, the criteria which has given this a UNESCO tag is architecture and aesthetics. So UNESCO is also planning to incorporate stone as a criteria for all the stone-built heritage sites. That is the project on which we are working. The second stone is about our expression of divinity because it is a Jain temple in Jaisalmer, the Ludarva Jain temples. about our religious beliefs, about craftsmanship, intricate carvings, and architecture again. So these are all based on stones. If we had no stones, we would have never had the religious beliefs documented, and we would have never known that how the practices were in the past, how the craftsmanship was in the past, what were the emotions of the people, why they made such tomb structures, or why they made such temples. Then the last one is an example of Tughlaqabad Fort, which is made up of Alwar quartzite. Now this fort, anywhere in the world, the forts are expression of power, defense, strategy, architecture, and craftsmanship. So these are all diverse monuments, but what is common to all this? All these monuments are the stones. Stones of diverse types, but stone is the common foundation of all these monuments. So currently, these are all tourism spots, but why can't we make them as geotourism spots, where we make public aware of the stone, the stories of the stones, how the Jaisalmer limestone formed, or how the Makrana marble formed, how the Alwar quartzite came into being. All right, now this very interesting rock, which is again part of Western Ghats, especially concentrated in Maharashtra, parts of Gujarat and Rajasthan, is basalt. Very nice basalt flows are visible in Mahabaleshwar. If you ever visited Mahabaleshwar, you'll see number of flows, and these basalts are known for Ajanta Ellora caves, Elephanta caves, Gateway of India. So these monuments, they utilize the local stone available, which was basalt. And one of the examples I would like to place is from Elephanta caves. So one of the caves have this triple-headed Shiva, which is known as Sada Shiva in basalt, and it is found in Elephanta caves, which is off Mumbai. You can see the location of the Is this working? It is not showing. Okay, anyways, we can see Mumbai is on this side, and if we go towards the east, there's this elliptical red enclosure, which is the location of Elephanta caves. Now, this was interpreted as that the central sculpture shows calmness. The left side, Shiva, is little angered. So if you look at the interpretations by archaeologists, the image of the center is calm and welcoming, while the face to the viewer's left shows Shiva's ferocious aspect. His fierceness is emphasized by a twisted mustache, frowning expression, skulls and snakes in the matted hair, and earrings in the form of snakes. The face on the viewer's right, meant to signify a gentler aspect of Shiva, this face is shown with soft curls, wearing a sweet-tempered smile, and carrying a lotus. So this is how we, okay. So this was towards the left, which shows an angered face, and towards the right, with the lotus in the hand, shows a pleasant face. And in the center, he is very composed. So the interpretations are extremely important, which are again sculptured in the stone. So what's the relevance and significance? The science of architecture came into being because of the earth's materials, because of the stones. The science of geology as a formal subject came into being because of the stones. The history, the historians, they make up so many stories based on what has been recorded in the past in these monuments. The archaeologists, the evolution, history of art like sculpturing, inlay. For example, marble is a perfect rock for inlay. When we talk about Taj Mahal, what comes to our mind is white marble, but there's beautiful inlay work throughout the marble inside the monument. So the semi-precious stones which enhance the beauty of the marble is through inlay work, which is a form of art. Then inspiration for poets, lyricists, writers, and directors. So we saw a glimpse in this Sound and Light show where the narrator talks about the importance of the stones. The tourism a huge tourism industry where people visit these monuments and enjoy them, and much more to it, because the societies and cultures are based on our historic past recorded in the rocks. So I work for a commission, as I said. This is part of that project where we are documenting many stones from India, not only the laterite, and these stones, they all are related to propagation of cultural values and the traditions. Now, we have three subcommissions. One is subcommission on geosites, the subcommission on heritage stones, and subcommission on geocollections. Subcommission on geosites talks about the natural sites, the sites where there is no human intervention, which are sculpted through natural agencies like wind, water, glaciers, and we have a lot of scope of documenting these natural geosites. For example, the Lonar Lake of Maharashtra, or the columnar basalts in Karnataka, St. Mary's Island. So these are naturally sculpted. But we, as Subcommission of Heritage Stone, talks about stones which have enhanced the heritage value and where humans have played a major part. Subcommission on Geocollections talks about the museum collections, because everyone cannot visit the field. So for them, we display a lot of artifacts or samples in the museum for them to enjoy and understand the relevance of those specimens. So how do we define heritage stone? It is a designation, just like a UNESCO World Heritage Site designation. IUGS heritage stone is an IUGS designated natural stone that has been used in significant architecture and monuments, recognized as an integral aspect of human culture. So, to date, 32 stones from around the world have been designated as heritage, IUGS heritage stones, and India is fortunate to have 4 stones designated. One is Makrana marble, the second Alwar quartzite, which tells you the historical accounts of Delhi. Delhi was ruined and rebuilt number of times. After 10th century, Delhi came into life 9 times, and what we are experiencing now is the 10th city. So the foundation of this city is based on the quartzite. You all must have heard about Delhi Ridge, which crosses Delhi. Then the third stone is Deccan basalt, which you all are aware of, the beautiful flows of Deccan basalt. And the fourth stone is Jaisalmer limestone, which is known for the Jaisalmer fort, the only living fort of India where the people lived inside the fort. Okay, so this is the kind of certification which is given by International Union of Geological Science. For example, the Deccan basalt India has been designated as a IUGS heritage stone. The Western world takes a lot of pride in documenting their heritage stones. So India is a bit slow, and we are trying to document because we have the most, I should say, enriched monuments in terms of their architecture, in terms of their artwork. So now we start with laterite. This is a plaque which is displayed in a place called Angadipuram in Malappuram district of Kerala to celebrate laterite, which was first time named, although the stone was into use or was into being for many years, but in 1807, a man named Francis Hamilton Buchanan, who was a surgeon, he named this rock as laterite based on a Latin word later. Now this is a very intriguing rock because when you cut it, it's very soft, but when you leave it and expose it to air conditions, it becomes hardened, and it has survived many years. Whatever monuments you see in and around Goa or all along the Western Ghats, they are made up of laterites. Now, laterite forms by weathering of rock in humid tropical parts of the world. It is not only confined to India. If you go to Cambodia, you will see Angkor Wat temples. They are also made up of laterites. Is usually reddish and mottled and porous in texture. So I was just having chit chat with one of the dignified ladies in the auditorium, and she spoke about her childhood memories when her grandmother built a house of laterite, and she said it was so porous that we could see through the pores, and it is still standing strong after so many years. So the beauty of this stone is that it withstands the atmospheric vagaries, but to preserve it or to increase its longevity, one needs to lime plaster it. And it is economically also very important rock because it is a ore of aluminium, ore of iron. When we weather this rock, the iron and alumina in this rock, it changes into hydroxides and minerals like limonite, goethite, bauxite. We all have heard about bauxite. It is the only ore of aluminium. So economically also it is a very important rock, and as a building stone also it has been extensively used. So just to give a brief introduction, I'm sure all of you know what is a laterite. So the bedrock, which is the rock which has not been affected by the external weathering conditions, the profile will form based on how deep the water percolates. So from the surface, there will be a ferricrete capping, which we call as gosan. That's why the soil appears so reddish. So me and ma'am were talking about it. She said the soil is so red. The reason for the reddening of the soil is high iron content. And then we have limonite, which is iron hydroxide, followed by saprolite, which is a partially weathered rock, and then the original bedrock. So laterite is composed of, this is the laterite profile, and you can see a real laterite profile. On the top you see the soil which supports the vegetation, followed by mottled area and the lateritic profile. So to celebrate laterites and its usage, Geological Society of India erected a monument, and this is the monument in the memory of, again, Francis Hamilton Buchanan, who named this rock as laterite from Angadipuram, Malappuram district in Kerala. So this is just as a mark of respect, they have displayed this as a national monument, geological monument in this area, from where the laterite came into, this name came into existence. So I already spoke about what are the minerals which are present in the basalts. They are minerals like pyroxenes and plagioclases. And these minerals, they are rich in iron, and plagioclases are rich in aluminium and alkalis. So alkalis being mobile, they will leave and the rock will get concentrated in iron and aluminium. So that's why we get minerals like, minerals like goethite, hematite. Then kaolin is a form of clay, which is alumina rich, and gibbsite, aluminium rich hydroxide. Megascopically, like what we see through the eye, the samples are reddish brown to dark red in color, so we can see the coloration. And the rocks mainly constitute ferruginous minerals, again, goethite, hematite, imparting reddish brown to dark red color to the rocks, and yellowish white colored because of the kaolin and gibbsite, which is alumina rich. So the best bauxites which are used for, the best laterites which are used for building purposes are iron rich, which are found along the Western Ghats. All right, so this is a textbook picture where we tell students that these are the Western Ghats, which runs for 1600 kilometers, and the average width is somewhere 50, 80 kilometers, and somewhere it reaches 100 kilometers. So this zone is capped by laterites. Everywhere we will get the color change because of the bedrock, difference in the bedrock. For example, in Maharashtra we will see basalts. In Goa region or Karnataka region, we will see some metasedimentaries, some metagravicates, and in the Kerala part, we will see a lot of granulites. So the rocks are different, but the rock formation is the same, and the laterite will take the nature depending on the bedrock which is getting weathered. So we see the location of laterite in the form of mesas and carapace and some laterite quarries from where we extract the stone. So the quarries are marked in yellow. This is a very generalized profile where you all know where is the Konkan coast, the narrow track from where the Western Ghats starts, and the Malabar coast in the Kerala part. So we can see again the laterites forming all along the Western Ghats, and it is because of the hot and humid climate where the rain is torrential, and then there are periods of humidity, and the weather fluctuation leads to intensive weathering of this rock. We come to Goa because the laterites which we will discuss, the monuments prominently which we are going to discuss are from Goa. Now, the laterites forming are along these 10 plateaus marked here. We also call them as flatlands, tablelands, and you can see the distribution of the quarries, laterite quarries in Goa, and the monuments in laterites. So the source of the laterite for the monuments is very proximal. The quarries and the monuments, they are somehow related. So when these monuments were constructed, there was no formal science of geology. So people had that sense how to use the stone. The architecture was not a formal science, but people knew how to stack the stones together, and I think they are much more stronger and stout than what we construct today. So the last part of the talk, laterite, the subtle narrator. Okay, so we start with the cave monuments in a place in northern Goa, in Bichulim, which is Arvelam Cave, and this is very near the Harvelam waterfalls. If you have not visited this place, all of you are in Goa, you must visit this cave, which is a 5-chambered cave, and it is believed that the 5 Pandavas once occupied these 5 chambers. But what we found in this cave is the placement of lingas, the Shiv lingas, and it is identical to what we found in Elephanta caves. And this is an in situ, like the stone was not transported from anywhere. That was the architecture at that time, that the cave was excavated, and you can see the profile. This is the profile, and I'm showing the profile here, the formation of laterite below the soil profile. So if you see, the central shrine is with a similar pitha enshrined by a linga with visible cylindrical top and square bottom, which is similar to the early usage of linga at Alora and Elephanta cave. So this is how the archaeologists interpret. Now, going a little ahead in Bichulam, we see the color change. There you saw the color was much darker in comparison to this. So that is because the iron content which has changed and the amount of weathering. Along the cave, it is not very much weathered. It is, the bedrock is still seen. But as you go far off, you will see the soil profile is thickened, the mottled part is thickened, and then we see the lateritic formation. The second cave, which is part of South Goa, and I'm not sure how many of you have visited this cave. This is Rivona cave. Another interesting cave where people believe that the Buddhist monks, they meditated. And why they thought so is because of discovery of a headless image of Buddha in the vicinity of this cave. You can see this is all lateritic. I'm sure you can all identify how the laterite looks. Then I come to some of the forts which were constructed not only during the Portuguese when they came to India, but during the invasions from the northwest. And this is one of the fort which overlooks the Vegator beach. I'm sure some of you must have visited this fort. And what we see is just the remnants of the ramparts which once were the defense walls of the fort. So it is it is all in a condition where we need to preserve and restore them. And the fort is fortified with laterite walls with several gun and cannon points. That's the bastions and the parapets. But as of today, if you look at the entire picture, you can see the ramparts, the fortification, but what we see is just the fortification wall and nothing else. The second fort, which I discuss, is Rais Magos Fort, which was a defense fort, which was used for defense purposes, but lately it became a prison for the people who rebelled to free themselves from the Portuguese rule, and after that converted to a hospital, and finally it is an art gallery. So how the things have evolved over a period of time. So you can see the solitary confinement cell and the restored fort, which has the craft center, the exhibition halls, where these displays are from those exhibition halls only. These displays are from the exhibition, which give us the historical accounts how this fort has been used since 1493. Okay, now very interesting petroglyphs which we get in South Goa in a place called Usgailimal. This is the place, and this was an accident. A torrential downpour led to the visibility of some of the petroglyphs, and the local people reported this to Archaeological Survey of India. The whole area was cleared, around 3,000 square meter, and around 120 to 125 petroglyphs were recorded. So you can see some engravings, that is of labyrinth, a pair of bulls, a headless bull, and a peacock. So this was the expression of the people during those times. Otherwise, we would have never known. And this labyrinth, which is also reported from different parts of the world. So for comparison, you can see a labyrinth at Cornwall, UK, which is in a shale. And this is from Goa. So it is a very interesting engraving, which is supposed to be used for clearing of the water or for the passage of the sacrificial blood. So this is how they interpret. Who knows why these were created? So a clear picture, and they even relate it that you don't see too much of meshing inside. It is from the center to outer part, and it is like two cobras sitting together, coiled together. Then we come to very famous UNESCO sites, the 7 convents and churches of Old Goa. So these are all documented here. The Chapel of St. Catherine, the Church and Convent of St. Francis of Assisi, which is in a dilapidated state, although it looks, when I went there, I really enjoyed the place, but I think we need to restore this place as well. So all these 7 group of convents and churches, they form UNESCO sites. And why were they designated as UNESCO site is because of the architecture, which was architecture which involved Manueline architecture, which is very Portuguese architecture, then Mannerist, which is Renaissance, New Renaissance from Italy, and the Baroque architecture. So it's a merger of 3 different types of architecture which led to the designation of these as UNESCO sites. So the front, or you can say the facade of the Basilica of Bom Jesus, where you can see the utilization of laterite along with basalt. This is what I've read, that this is basalt rock. I could not sample it because they don't allow you to take any sample. And you don't see any carvings on this rock because it's highly pitted, porous. You can't carve this rock. So the beauty of rocks, for example, the limestone can be carved, marble can be carved, but you cannot carve a laterite. And this is a closer view where you can see the pitting very clearly in the laterite. So I was talking about this ruins of Church of St. Augustine, where the bricks of laterite were used and lime plastering was done. So it could survive longer than the time anticipated, because this rock needs to be conserved through lime plastering. All right. Then the use of laterite in Chapel of St. Catherine, again you see very intense utilization of laterite and how it is getting deteriorated at places. Okay, there's very interesting Portuguese colony, or you can say a small part in Panjim, which is known as Fontinhas, and it is a UNESCO heritage zone. So the core structure, if you look at this, is made up of laterite again. In the core you can see the laterite bricks have been used, or masonry stone has been used. And these are again lime plastered and very bright houses. It's a beautiful setup in Panjim area. And UNESCO heritage zone since 1984. Now I move to Kerala, where you see these mushroom shaped rocks and the circular pattern, where these are known for urn burials. The remains have been buried beneath these structures. And these are from the past, like we don't really know when this culture of burying the remains started. But these features are very common in Kerala. Again, the utilization of laterite in Karnataka, in Mirjan Fort, and the bastions you can see on the fort sides. And then this is a fort in Kerala, the St. Thomas Fort. Again, utilization, and still the laterite quarrying is going on, and we can see the division of this 1.72 meters, for example, into 9 sets. So the blocks are cut like this, and they are left for 3 months to get hardened, and they are still being used. If you go to the countryside, you will see a lot of residences made up of laterites. So with this, I want to finish off this little account on the story of laterites. And I want to acknowledge my co-authors, because I came all the way from Chandigarh to cover this area, and these are my co-authors. One of my students is sitting here, Parminder, Jaspreet, Uday, he's also a student who's working with NCPOR, and Professor Raymond Durai Swami, who's from Savitribai Phule University, Pune, Bobby Matthew from St. Xavier's College, and C. Srijit from Ponani College, Kerala. And I want to thank you all for listening to the story of the stones, and I hope you will remember the saga of laterite and some unique monumental stones which I discussed. And I want to play this small clipping once again.
Are the relics of Golconda's past. These rocks and ramparts are the sole witnesses of that splendor. They alone have survived, unfazed by the march of events, defying the ravages of time, mute, inscrutable, and unmoving in their silent vigil. The legacy of India's glorious past.
जिसकी
So the poet says, what witnessed the grandeur of this fort is the star laden sky. But as a geologist, I would like to say the rocks witnessed it all. Thank you. Oh, of course.
Nice, very nice talk. Really interesting. I also believe in the old. When I am in Hyderabad, and I know whenever I hear
You
this
become
song,
very nostalgic.
yes,
That's
nostalgic,
true.
true, and I feel that I should have been there at that time. One thing I want to ask: you said some of the stones are heritage stones.
Some of
They're
the stones are?
heritage stones, declared as heritage stones like
Yes,
Makhana
yes,
marble,
yes.
which means that it cannot be used anymore or something like that. Stop
Yes,
in.
they are being used. The Makrana is being used. The Jaisalmer stone is being used. But one of the important issues which we raise is that we need to declare them as historical quarries. We don't want to replenish that resource so that we get it for restoration purpose. As you know, the Makrana is almost finished.
So you need a special permission to use that stone for any construction purpose or?
Yes, this is being used because the Indian government is not ready to stop quarrying. It gives you a lot of economic loss if you stop quarrying. Laterite was stopped by the Supreme. The quarrying was stopped by Supreme Court, but I've heard it is again reopening. So
I think we should, like Western countries, we should stop using it. Perhaps if you want to preserve them.
Yes,
I feel that's
some
my
of
view.
them, especially the ones which sourced the stone for the monuments.
Certainly.
Yes, and in the West they do this.
Here is one question.
Hi.
Yeah.
Thank you, Professor Kaur. Like it was marvelous talk, and means definitely change. It will change our perspective whenever we will go to such kind of monuments. So just to like you showed one world map where
Yes.
you showed like all the different
The designated stones.
Yes, yes.
There are stones like Carrara marble. Everyone
Right,
knows about which.
So ideally, means if you see like Africa or maybe some other different countries or even like continents, there is no information about different type of rocks here, right? Like I'm
Because
just wondering.
this concept is slowly picking up. If you look at the map, you will see most of the stones are from Europe.
Right, right.
It is a very unbalanced scenario.
Right, right.
And India is the only country from entire Asia which has 4 stones.
Okay.
And let me tell you, next year laterite will be a heritage stone.
Okay. So
we are already working on the proposal.
So you already talked about like in Karnataka
Correct.
several monuments, but like let's say Hampi. So is it also laterite or is it
No,
like
it is
some?
not laterite. It is a gneissic rock, granitic gneiss.
Okay. So have you done any kind of?
We will give chance to others, please. Yeah, yeah.
Thanks very much. I didn't know that Goa has got caves and all that.
Oh yes, sir.
But if you permit, I would like to slightly deviate and ask you what is Shaligram Shila and the Shivlinga from Narmada?
I could not get his question, please. Can you repeat it for me?
Shaligram Shila from Gandaki, from which the deity of Lord Ram is being constructed, and installed at Ayodhya. Huge rock piece. Normally, the Brahmins carry a very small piece of Shaligram Shila always, whenever they worship Vishnu. And all the Shiva lingas, they come from Narmada river. So, what kind of rocks they are? And
So-
why are they so much, worshipped?
What area is he talking about? Shaligram something? What place?
From Gandak.
Shaligrams are ammonites. So
yeah,
when you
we
break
get it in
them,
Nepal and all those places. When we break,
you
yes,
get an
ammonite.
ammonite.
Yeah,
Ammonite.
is he asking about that Shaligram?
Yeah.
Yes.
Yeah, that's just a belief. I don't think it has something to do with the real fact.
Okay. I think we will have a question from Renee
Can
now.
I?
Yes, please.
Can I
Excuse
ask
me.
a
Yeah, yeah. Excuse me. Thank you.
Can I ask? You had the sub commissions. You said geocit.
Subcommissions, geocollection,
Right.
site, natural
Right.
site,
Yeah.
stone.
Where are fossiliferous rocks? Where do they feature? Because as you know, India.
Stone is full of fossils, and Globigerina stone. If you see,
Right.
it is from Malta.
No. So I'm saying, do you have a special category for fossiliferous rocks? Because as you know, India has destroyed all its fossiliferous rocks. So is there in this subcommission any special category for, say basalt with is so highly metamorph, igneous, so hardly will contain fossils. So do you have a category in?
Ma'am, there are two different things. Fossiliferous.
Go to the mic.
Fossiliferous rocks in natural sites, you mean to say, or for the monuments?
Natural sites.
Yes, we have a lot of places which have been designated as IUGS, heritage geosites. We have, in fact, UNESCO Global Geoparks, which are coming up, which document a lot of such sites in China, which are fossiliferous. And many parts of the world, they document. And in fact, India is also trying to get it's this Dhar area from Madhya Pradesh as a big UGGP from India. So some projects are in pipeline, and in fact, Government of India is trying to, like, we geologists, we pestered them so much that a geoheritage bill should be introduced, where we talk about preserving these sites, because once lost, they will never form again in our lifetimes or in millions of years. So there's a special bill which we want should be contested, and it should be passed, and it should be forced into being, so that such sites are not disturbed. And we need to preserve them. Very valid. Yes, madam.
Well, in the first part of your talk, you mentioned about some cultural attributes associated with rocks. So, for example, if one wants to display power, would one always use quartzite?
Oh, it depends. Depends on the area. For example, laterite for the forts. Laterite for the forts in this part of the country. Alvar quartzite was so predominantly present in the Delhi ridge, so it was used for fortification in that part, because the transportation is not that easy. It's only very special stone like Makrana marble, which had very special attributes. It's a calcitic marble. And the Victoria Memorial in Calcutta is also made up of this marble. There is a very interesting story, not story, it is basically a factual documentation in one of the INSA volumes. When the proposal of building this Victoria Memorial came, the stones from Carrara were brought to India and from Makrana. So the Makrana was found to be much tougher and less prone to weathering. So the memorial was built using Makrana.
So
Carrara is a height stone, good for sculpturing, but not for monument building, because the stone bows very easily.
So it's essentially physical availability.
Yes.
Okay.
Yes, of course.
Yeah. Excuse me. Towards the end, you were showing the petroglyph with the maze and the similarity or the virtual identical one in Wales, did you say?
No, it is, yes, from UK. It is not like that. It is different, because they have interpreted it as two coiled cobras.
I see.
Yeah.
But it looks very, very similar, right?
Similar, but they have different design, because that is like a mesh, and this is like from the center you can go to the outer peripheral area for discharging the rainwater or sacrificial blood, as I said. This is what has been written in the records.
I see.
We'll
Yeah.
have one quick question.
Something very different from what we interpret, like.
Thank you, ma'am, for drawing our attention. I'm a local Goan, and laterite is a everyday for me, but it's so important. Laterite is found all over. There are two kinds of plateaus. One is local lowland plateaus, where even this institute sits on a plateau, or airport sits on a plateau, and highland plateaus like Karst Plateau is an example. I would like to draw your attention because saga of laterite. I'd like to draw attention to another chapter which can be there in this saga in August gathering. Research from Goa University and research from Kolhapur University has found that these laterites are high in endemic plants. These the porous rocks. They have the deposition of organic matter, and only during the 4 months of rainy season. Otherwise, they are dry and barren. And only these 4 months they bloom into the wonderful different plants, and the endemicity is so high that the endemicity of these plants on this plateau is equal to or more than the evergreen forest in southern India. And there are more endemic plants on the plateaus in the northern Western Ghats than the forest. And this is one of the findings which I'm also a big part of that group, wherein we are trying to tell the government that these should be also preserved.
And I would like to include this in our proposal, because this will add in value to our proposal, that why these laterites are also important for the endemic species of plants. And this whole Western Ghat is already a natural site, UNESCO natural site.
So a Karst Plateau is considered
there are
as the
more questions.
Valley of Love.
Yeah, there are many more questions, but I request them to be taken up offline in light of time. I wish to thank Professor Kaur.
It's an honor, sir.
It was, some of us were complaining that we didn't get to see Goa listening to all the technical talks. This was a beautiful sightseeing of Goa, and broader than that, through the lens of stone, looking at the culture, art, history, and heritage. Thank you very much for a wonderful.
Thank you so much.
On behalf of the Academy, we would like to hand over a bouquet. Sorry.
But I don't know what to announce. You are going to
Yeah, so for boat cruise, buses are waiting at this side. All of you can slowly go out and board in